something about 20 or 30 kilometers, very, very famous German football team, Recklinghausen, and you have to be careful. Here in Recklinghausen, you are in the epicenter of Schalke 04. So it's very dangerous to say something against Schalke 04 here in Recklinghausen, but you also have people supporting Borussia Dortmund and people supporting VfL Bochum. So that's very important, and it's also a cultural thing here, because you can talk with the people about football for past histories, future histories. You have always something to talk about, and we have a genetic meeting, and I'm sure that people here in the Ruhrgebiet think there's no sporadic cases. Yeah? It's dominant, it's an epigenic phenomenon, because they think that the DNA is either blue and white or black and yellow. So that's not my personal opinion, and I think my DNA has another color, but that's the thinking about football and genetics here in the Ruhrgebiet. And another thing is, you're here in the Knappschaftshospital, and I will just tell you what is a Knappschaft, because I think people from abroad, from other countries, don't know what the Knappschaft is. And again, a football theme, because this is a team, the German national football team of 2016, very successful, as you know, and the nickname of the German football team is the Mannschaft. So you know what a Schaft is. A Schaft, Mannschaft, is a community of people, a team of people, as Mannschaft, Mannschaft. So, and now I have to tell you what is a Knappschaft. Ein Knappe is someone who's working in a coal mine. So the Knappschaft is a community of people working in coal mines. That's a Knappschaft. And as it's a very dangerous job and a hard job, the Knappschaft was the first German insurance and social health system founded over 700 years ago. And you find these Knappschaftshospitals, especially in regions where there are coal mines or former coal mines, because they were in charge for the people working in the coal mines, and that's a Knappschaft. So I would like also to thank the Knappschaft, and Matthias Wagner will say some words after me, because they give us the opportunity to these facilities here in the nursing school, and they also sponsored all the catering for the 2 days. So thank you to the Knappschaft, and I'm very grateful that you have done this for us here. Thank you very much. So again, a warm welcome here to the Ruhr region, and I will turn over the mic to Matthias Wagner, who will say some words from the Knappschaft side. Thank you very much.
Good morning, everyone, and a very warm welcome to Recklinghausen and to our hospital. A particularly warm welcome to Dr. Tom Wahrlich, to Susanne and Henry Wahrlich, and to Professor Dr. Klebe. Dear guests, it's a great pleasure to welcome you all to Recklinghausen and to our hospital. Professor Dr. Klebe, you have just given us an excellent introduction to this symposium, and it's my pleasure to follow your remarks and as CEO of Knappschaft Kliniken Fest GmbH to extend a very warm welcome to all of you. Our symposium today takes place in our nursing school, a fitting setting for a gathering like this, and as an academic teaching hospital of Ruhr University Bochum, with our own nursing school, education and science have always gone hand in hand here. This makes our institution the ideal place for a symposium of this caliber. Knappschaft Kliniken Fest GmbH, with its location in Recklinghausen and Marl, 22 specialist medical departments and more than 2,000 employees, is a leading center of specialized care for the Recklinghausen district and the wider region. We are also part of a strong network. Knappschaft Kliniken comprises 13 acute care hospitals and 3 rehabilitation clinics, treating approximately more than 1 million patients every year. In everything we do, we carry the spirit that has defined this institution for decades, a spirit of solidarity and compassion deeply rooted in the history of the Knappschaft and the mining communities to the Ruhr region. It fills me with great pride that this symposium, one of the most important international gatherings dedicated to research on HSP, is taking place in our institution this year. Our sincere thanks go to the Tom Wahrlich Foundation, which has worked tirelessly since 1998 to advance research into HSP and to bring hope to those living with this rare disease. What this foundation has built over nearly 3 decades is truly remarkable. I would also like to extend my warm thanks to our own staff who have worked with great dedication to organize and prepare this symposium. Without their efforts behind the scenes, none of this would have been possible today. Ladies and gentlemen, our doors are open for 2 days of intensive exchange, new discoveries, and shared hope for all those living with HSP. I wish you a successful and inspiring symposium. Thank you.
Good morning, everyone. After writing emails, many emails with you over the last months and weeks, I'm very happy to see you all here and to welcome you in this beautiful red light room. And I hope we will have a successful meeting.
Stefan has made a great introduction, but I think the only one he forgot, and we have to make a big applause to himself, who did the main part of the organization here, and who's a close friend and a medical advisor for us for decades now, and it's really great that you finally found your place here, and it's just a great venue here. Thank you very much, Stefan. And there'll be 29 presentations, 22 posters for those of you who didn't hang up the poster yet. Just is next door, put it there. So a lot of talks ahead, and just we are happy that you're all here. Thanks.
I have nothing to say. I'm only say I'm 87 years old.
And you always said normally you don't want to die until something has been found against HSP, so please repeat that then. Well, okay, have a good meeting.
So good morning, everyone. So it's my pleasure today to, so I'm Christelle de Pienne from the University Hospital Essen. And it's my pleasure to introduce the first speaker. So it will be the genetic session this morning. It will be divided into 3 parts. So first, what's known, where we review what is the current knowledge in the field. Then what's new. We'll have 2 wonderful PhD students presenting their work. And then what's next, I will hand over to Stefan Zuckner for the chair, but we'll have also a wonderful presentation exploring future directions and new technologies. And without delay, because I inherited some delays already, I want to introduce Professor Antonio Arlacchio. So he's a physician scientist. He leads the medical genetics section at the Department of Medicine and Surgery at the University of Perugia. And as far as I can remember, I've seen publication from you, but we never met before. And so I'm super happy that you're here, a new face in this meeting.
It's a pleasure for me too.
And so the floor is yours. Thank you.
So I would like to thank the organizer for inviting to speak today. It's a pleasure to be here. In this talk, I will discuss hereditary spastic paraplegia as a disorder with many different genes but shared biological mechanism. My aim is to clearly connect the clinical feature with the underlying molecular pathway and to explain why long corticospinal neurons are especially vulnerable in this condition. Today, I will first review the genetic diversity of hereditary spastic paraplegia, which include many different genes and mutations. Then I will show, despite the diversity, many of these genes affect a similar cellular pathway. Finally, I will briefly discuss how focusing on shared mechanisms may help us move toward pathway-based treatments rather than targeting single genes only. First of all, with this slide, I briefly introduce the historical roots of hereditary spastic paraplegia. The condition was originally described by Strampel in 1880 and later by Lorraine in 1998. They characterize a familial progressive spastic paraplegia due to corticospinal tract degeneration. For decades, it was viewed a relatively homogeneous clinical entity. Today, we recognize HSP as a genetically heterogeneous spectrum involving more than 80 genes. This historical transition illustrates the evolution from descriptive neurology to molecular medicine, which is the conceptual framework of this seminar. Hereditary spastic paraplegia is clinically defined by progressive stiffness and weakness of the lower limbs, reflecting the degeneration of the corticospinal tract. The main pathological feature is damage to very long axon. Although the disease is rare, with the prevalence of about 4 to 10 per 100,000, it provides an important model to understand how long axons degenerate over time. Based on current medical knowledge, there is no cure for HSP, and there aren't any approved treatments that can help prevent or reverse the progression of the disease. Clinically, we distinguish between pure and the complicated form of HSP. In pure forms, as you know, patient mainly slow pyramidal signs such as spasticity and hyperreflexia. In complicated forms, additional symptoms can appear, including ataxia, neuropathy, cognitive problems, or gastroesophageal reflux. Overall, complicated forms are associated with a greater disability burden. Importantly, the same gene can sometimes cause different clinical presentation, which show that other biological factors influence the final phenotype. HSP typically follows a slow progressive course. Age at onset can vary widely, from childhood to late adulthood. Most patients experience gradual worsening of gait due to increasing lower limb spasticity. In many pure forms, life expectancy remains close to normal, although disability accumulates over time. Clinical disability is often measured using a standardized scale. One of these is the Spastic Paraplegia Rating Scale. Progression is usually slow and becomes evident over several years. Different genetic forms may show different progression rate. Long-term follow-up is essential to understand the natural history of each genotype. The key concept in HSP is that cortical spinal neurons are extremely long. This is very important. Because of this, they depend on very efficient transport system and a constant supply of energy to maintain their distal parts. If defects occur in these systems, the far end of the axon is affected first. Over time, this leads to progressive degeneration and explains why lower limb spasticity is the main clinical sign. In parallel with this physiological insight, the genomic era has profoundly transformed our understanding of HSP. The transition from linkage analysis to next-generation sequencing has led to the rapid identification of more than 80 causative genes, revealing remarkable genetic heterogeneity. This molecular expansion shifts HSP from a clinical defined entity to a complex network of pathway-related disorder. These animated slides represent only a small selection from the extensive literature available in the field. Actually, there are many more studies out there, and the field is constantly evolving. New technologies and applications are being developed every year, making next-generation sequencing increasingly important in both research and clinical settings. HSP can be inherited in different ways. Autosomal dominant forms are common in many countries, while autosomal recessive forms are more frequent in population with consanguinity and often present with more complex symptoms. There are also X-linked and rare mitochondrial forms. Studying these inheritance patterns helps us better understand the biology of axonal maintenance. At this point, we can introduce the main criteria used to classify HSP. First, clinical symptoms and signs. HSP is traditionally divided into pure forms and complex forms, as I told you before. Second, the age of onset of spasticity. We distinguish type 1, early onset before 30 years of age, from type 2, classic or late onset HSP, when the age of onset is more than 35 years, depending on genetic subtype. Third, the pattern of inheritance, which may be autosomal dominant, autosomal recessive, or X-linked or mitochondrial forms. Finally, HSP can also be classified according to the intracellular pathway involved, such as alterations in the endoplasmic reticulum, axonal transport, lipid metabolism, lysosomal pathways, or mitochondrial function. So the majority of HSP cases show mutation in the SPAST gene in the autosomal dominant pure pattern in Northern America and Northern Europe. The prevalence of familial inheritance is often higher than the sporadic form in 2-to-1, with 70% of autosomal dominance linked to pure HSP. Nations with higher consanguinity are associated with the autosomal recessive HSP form in ethnicities such as Middle Eastern and American Amish groups. Mutations in the SPG11 gene are the highly prevalent autosomal recessive, followed by SPG15, 7, and 5. To date, 3 HSP are observed to be X chromosome linked, and 4 mitochondrial genes cause HSP with a prevalence rate of 1 to 2% in the registered HSP cases. So the marked genetic heterogeneity that I told you just now make genetic testing a key component of the diagnostic strategy. The process begins with careful clinical characterization, which helps distinguish pure from complex forms and guide the suspected inheritance pattern. The first genetic step usually involved targeted NSGS panels for HSP genes. If results are negative, whole exome or whole genome sequencing can be performed to broaden the analysis. Identified variants are then filtered and prioritized using population databases, prediction tools, and phenotype correlation. When no pathogenic variants are found, copy number variants should be also considered. Finally, candidate variants are interpreted according to ACMG guidelines, leading to a current diagnostic yield of approximately 30 to 50%. This diagnostic framework has also been fundamental for identifying the major HSP genes. The major HSP gene, for sure, is SPG4. SPG4 is the most common form of autosomal dominant HSP. In many cohorts, it represents about 40% of dominant cases. The disease is linked to variants in the SPAST gene, which encode spastin. Spastin helps microtubules stay dynamic, which is important for axonal transport. When spastin function is reduced, transport becomes less efficient over time, and distal axon gradually failed, leading to progressive spasticity. SPG3A is the second most frequent form of autosomal dominant HSP. SPG3A is a classic early onset autosomal dominant HSP. It is caused by variants in ATL1, which are called atlastin. Atlastin-1 helps the endoplasmic reticulum form a continuous tubular network. If this ER network is disrupted, membrane trafficking and organelle organization become less efficient. Long cortical spiral axons are especially sensitive to this, which helps explain the selective clinical phenotype. Another form of scientific interest is SPG12. It is an autosomal dominant form of HSP caused by mutation in the RTN2 gene which encode the reticulin 2. Importantly, SPG12 contributes directly to the recognition of ER shaping proteins as a central pathogenic mechanism in HSP. Reticulin 2 plays a key structural role in maintaining the tubular architecture of the endoplasmic reticulum. This discovery reinforced the concept that the disruption of ER network integrity is critical for long axonal maintenance. Another type of scientific relevance is SPG11 for sure, as you have seen before from the epidemiological data. It is the most frequent form of autosomal recessive HSP. It often present with the complicated typical phenotype and with the thin corpus callosum on MRI. The SPG11 gene encodes spataxin, which is linked to lysosomal function and autophagy. When these clearance pathways are impaired, damaged components build up inside neurons. This affects multiple neural systems and helps explain why SPG11 is more often complex than pure HSP. SPG15 is considered the second most common autosomal recessive form of HSP. It is caused by mutation in the ZFYVE26 gene, very complicated to pronounce, which encodes spastische. Clinically, patients frequently present with a complex phenotype, often associated similarly to SPG11 with a thin corpus callosum and cognitive impairment. At the cellular level, SPG15 is linked to endolysosomal and autophagic dysfunction overlapping mechanistically with SPG11. So, as just said, SPG11 and 15 represent the two most frequent forms of autosomal recessive HSP. SPG11 is caused by one gene with a complicated name, and SPG15 with another gene with another complicated name. Both conditions are frequently associated with thin corpus callosum and share the similar dysfunction of the lysosomal and autophagic pathway. Clinically, the difference between these two forms of the disease is the fact that SPG11 often presents with earlier onset and more pronounced cognitive decline respect SPG15. This comparison also highlights a broader issue in HSP, the often difficult correlation between genotype and phenotype. The broad genetic and clinical heterogeneity together with extensive allelic series make genotype-phenotype correlation extremely challenging. As illustrated in these slides, there are multiple allelic series. As you can see, there are, you can see this circle, many genes that can cause different type of disorder of the first motor neuron, of the first and the second motor neuron, or affected only the second motor neuron. This overlap makes diagnostic classification and the selection of the appropriate genetic test particularly complex. An example of multiple allelic series is shown in these slides. These slides show a schematic representation of the SPG11, ELS5, CM2X protein and the position of different mutations stratified by distinctive phenotypes. As you can see, we have the same gene that is mutated in different neurodegenerative conditions, and even the same mutation is reported in different of these conditions. So, in HSP, the same gene can produce different clinical presentation. Some patients develop the pure form, while others show additional neurological features. This suggests that modifier genes, environmental or epigenetic factors, influence the disease expression. So this is a particular type of monogenic disease. For this reason, genotype alone does not fully determine the phenotype in many cases. Now, I would like to talk about one important point. So the cell biology of the disease for the second objective of this seminar. This figure summarizes the main cellular pathway implicated in hereditary spastic paraplegia as adapted from Martinello and colleague. Although HSP is genetically heterogeneous, many SPG forms converge on a limited number of biological mechanisms. These include ER shaping, lipid metabolism, lysosomal function, and mitochondrial quality control. What is striking is that very different genes ultimately affect intracellular trafficking and organelle homeostasis. This convergence helps explain why diverse genetic mutations lead to a similar clinical phenotype centered on distal axon degeneration. So, among the different cellular pathways involved in HSP, one of the earliest and best characterized is the ER shaping pathway. Over time, the identification of SPG4, then SPG3A, then SPG12, and then SPG31 progressively revealed the importance of ER shaping proteins in HSP. Each discovery added a crucial piece to the puzzle, ultimately establishing ER network integrity as a central pathogenetic mechanism. These milestones illustrate how gene discovery reshaped our mechanistic understanding of HSP. Specifically, atlastin, SPAST, RTN2, and REP1, I mean the gene encoding in these different types of the disease, encode proteins that maintain the tubular ER network along axon. When this structure becomes unstable, membrane organization and trafficking are impaired. This shared pathway helps explain why different genes can produce a similar corticospinal phenotype. Moreover, several HSP forms are linked to lipid metabolism. SPG5, caused by CYP7B1 variants, another difficult name to pronounce, alters cholesterol metabolism. SPG28 and 39 also affect lipid-related pathway. Change in membrane lipid composition can gradually destabilize long axon, making corticospinal neurons particularly vulnerable. Besides, some HSP forms are directly linked to mitochondrial dysfunction. SPG7 and 13 affects proteins involved in mitochondrial maintenance. When mitochondrial quality control is impaired, energy production becomes less efficient. Distal axonal segments are the first to suffer from this imbalance. In addition, ER mitochondria contact sites are essential for calcium transfer and lipid exchange. SPG7 directly affects mitochondrial quality control, while SPG3A, 4, and 31 influence ER structure. When these proteins are altered, what happens? That communication between organelles becomes less efficient, involving calcium transfer and lipid exchange. This might increase the axonal vulnerability. Furthermore, SPG11 disrupts calcium homeostasis in axon, which is essential for proper neuronal signaling. Calcium imbalance can lead to cellular stress and mitochondrial dysfunction. Prolonged alteration of calcium signaling may contribute to the axonal degeneration. Moreover, efficient axonal transport relies on intact microtubules and proper organelle organization. SPG4 directly affects the microtubule dynamics by spastic dysfunction. SPG3A alters the ER structure, which indirectly influences the trafficking, and SPG11 impairs vesicular and lysosomal pathway that may further disrupt the transport. Besides, in complex forms such as SPG11 and 15, widespread neuronal dysfunction may trigger secondary glial activation. Although inflammation is not the primary cause of HSP, I mean, is not a demyelinating disease, it can amplify neuronal stress over time. targeting secondary mechanisms may help, for sure, slow disease progression. So, although these SPG4 may involve different genes, they converge on a limited number of biological pathways. This concept is very, very important. ER shaping, lipid metabolism, lysosomal function, and mitochondrial dynamics all influence the axonal stability. The final outcome is distal axon degeneration driven by the extreme length and metabolic demand of corticospinal neurons. So, building directly on the concept of converging cellular mechanism discussed in the previous slide, the construction of the HSP-SOME by Novarin et al. Published in Science in 2014 demonstrated that HSP genes form a significantly connected network, highlighting a convergence on ER biology, intracellular trafficking, and nucleotide metabolism. Importantly, network diffusion analysis showed overlap with amyotrophic lateral sclerosis, Alzheimer's disease, and Parkinson's disease, positioning HSP within the broader landscape of neurodegeneration. So this slide is maybe the more important of the seminar because it represents the conceptual bridge of the seminar. After reviewing the genetic heterogeneity and mechanistic pathway, we now emphasize convergence toward the shared pathogenic mechanism. The extreme length of cortical spinal axon amplify subtle molecular defects. This convergence enables pathway-based therapeutic targeting and biomarker development. The key future direction is mechanism-driven precision clinical trial design. So, for precision clinical trial design, biomarkers are essential. In fact, biomarkers play a key role in connecting research to therapy, since they help us measure disease progression and stratify patients more precisely. In clinical trials, biomarkers can detect treatment effects earlier than clinical scale. For this reason, they are essential tools in translational research for HSP. Imaging biomarkers, for example, are increasingly important in HSP research, in particular in SPG4, 11, and 15. Quantitative MRI can measure spinal cord atrophy, or diffusion imaging may detect microstructural change in the corticospinal tract. This technique may serve as a support or even surrogate marker in future trials. Fluid biomarkers, for an example, the use of neurofilament light chain that is now used in many different neurodegenerative diseases, reflect axonal damage, especially in SPG4, SPG11, and SPG7. They can be measured in blood or cerebrospinal fluid. These markers may help monitor disease activity or treatment response. However, longitudinal validation is still required. Digital and quantitative biomarkers are particularly valuable in HSP due to slow progression. In pure motor forms, such as SPG4 and SPG3A, wearable gait analysis can capture subtle longitudinal change. In complex form, for example in SPG11 or 15, digital tools may integrate cognitive and motor parameters. Or mitochondrial forms, such as SPG7, may benefit from endurance and fatigue monitoring. This genotype stratified approach enhances sensitivity in rare disease clinical trials. So now, to discuss the translational implication, the third aim of this presentation, we should begin with the current therapeutic landscape of HSP. At present, there are no disease-modifying treatments capable of halting, reversing, or preventing the disease progression. Management remains largely supportive, focusing on rehabilitation and symptomatic therapy, aimed at improving quality of life rather than altering the underlying pathogenic process. In this context, symptomatic management has significantly advanced. For example, the use of intrathecal baclofen is highly effective for severe spasticity, providing better tone control with fewer systemic side effects. botulinum toxin in action allow targeted treatment of focal spasticity, improving mobility and facilitating the rehabilitation. Finally, the robotic exoskeletons represent an innovative rehabilitative approach, supporting the gait training and potentially enhancing neuroplasticity. Together, these strategies meaningfully improve function and quality of life for patients. However, even if treatment for HSP is still mainly symptomatic, better knowledge of shared pathological pathway is opening new therapeutic perspective. Both gene-specific and pathway-targeted approach are under investigation. Early intervention will likely be important to protect long axon. In this context, cellular models have played an important role in investigating potential therapeutic strategies. As you can see from this figure, cellular models of HSP have been instrumental in clarifying the pathogenic role of mutant spastin. In cultured cells, mutant spastin show aberrant accumulation along microtubules, leading to cytoskeletal disorganization. Pharmacological modulation with microtubule disrupting agents, specifically the use of nocodazole, partially rescues the abnormal distribution, supporting microtubule dynamics as a therapeutic target. Moreover, Drosophila models recapitulate key SPG4-related phenotypes, including axonal defects and locomotor impairment. Treatment with microtubule modulating agents, such as the use of vinblastine, for example, or nocodazole, ameliorate disease-related phenotypes. These in vivo data reinforce the concept that correcting microtubule instability may represent a viable translational neurology. Furthermore, gene therapy strategies in HSP are most directly applicable to autosomal recessive forms, such as SPG11, 15, and SPG7, where loss of function mutation allow gene replacement using AAV-based vector. In contrast, dominant forms such as SPG4 and 3A require gene modulation strategies, including allele-specific silencing or dosage correction. Although still preclinical, this approach represents a key step toward genotype-driven precision medicine. Besides, antisense-based therapy are particularly relevant for autosomal dominant HSP forms, such as SPG4 and 3A, in which the expression of mutant proteins drives disease mechanism. Allele-specific silencing or transcript modulation may reduce toxic protein effect. RNA-based approach may also be explored in selected SPG11 variants involving splicing alterations. These strategies remain in early preclinical development, but represent a promising precision medicine direction. In addition, small molecule strategies in HSP aim to modulate converging cellular pathway rather than single genes. In ER-shaping forms, for example, in SPG4 and 3A, microtubule stabilizer and ER stress modulators are being explored, as you can see from this figure. In SPG11 and 15, enhancing autophagy by agents like rapamycin, for an example, represent a rational approach. In SPGA, modulation of cholesterol metabolism by statins is a reasonable method. Mitochondrial stabilizer may benefit SPG7 and related form. These pathway-based strategies allow therapeutic convergence despite genetic heterogeneity. In summary, HSP include multiple SPG forms, such as SPG 4, 11, 5, and 7, each genetically distinct, yet mechanistically interconnected. Despite the difference in inheritance pattern and clinical complexity, these subtypes converge on shared pathogenetic pathway, and this is one of the main points of this presentation, including ER modeling, endosomal dysfunction, mitochondrial impairment, and lipid metabolism alteration. This convergence supports the concept of HSP as a network disorder rather than isolated gene defects. Importantly, network diffusion analysis, as you can see before from the HSPsome, showed overlap with other neurodegenerative disease, for example, amyotrophic lateral sclerosis, Alzheimer's disease, or Parkinson's disease, positioning HSP within the broader landscape of neurodegeneration. So, recognizing this shared biology enables pathway-oriented therapeutic strategies that may apply across multiple SPG forms or other related neurodegenerative diseases, paving the way for precision neurology approach. In conclusion, hereditary spastic paraplegia is transitioning from a clinically defined rare disorder to a model system for understanding axonal vulnerability and neurodegeneration. So we are entering a transformative era where mechanism-driven strategies may finally translate into disease-modifying therapies. So finally, I would like to sincerely acknowledge all collaborators, clinicians, researchers, and laboratory members who have contributed over the years to advancing our understanding of HSP. This work reflects a truly international effort spanning molecular genetics, cell biology, system biology, and translational neurology. I'm especially grateful to the patients and families who participate in our studies. Their trust, resilience, and engagement are the foundation of every scientific advance and the reason we remain committed to transforming discovery into therapy. Finally, I thank the organizers for the opportunity to present this work and to contribute a collaborative scientific dialogue that will define the next era of precision neurology in HSP. Thank you.
Thank you very much for this overview. So I think we have time for one or two questions. Do we have questions in the room? If we don't have questions right now, I think we can just have the next talk and have the questions together. Is it okay for you? Because we are 5 minutes late already.
Even because part of my presentation overlap.
Yeah, exactly. I think we can discuss, you know, which gene we should remove from that list, and then have a discussion together. So I will then introduce, while you install your computer, I will introduce the second speaker of this session, Stefan Zuckner, who is a professor of human genetics and neurology at the University of Miami. So he's a very efficient and successful gene hunter. He has this Genesis database that allows to find new rare diseases, describe new rare diseases. But today, I've asked him to discuss another topic which is close to my heart, which is gene curation. I've seen on the ClinGen website that there are many disorders that have this gene curation ongoing, but HSP is not part of the list. But you are on the website, so the floor is yours.
Okay. Sorry?
Oh, should I do that?
It's getting warmed up still.
Write the list.
Oh, here. Okay, it's great to be here again. Thanks for the invitation. Crystal discovered other activities of mine. So I thought I'd share some thoughts and reflection on this topic of gene curation. So known HSP genes, things that genetics still has to answer, I think. In HSP, as we just heard, we have many genes now that are known. By some counts, maybe over 80 HSP genes. If you count in genes or diseases that have spasticity as part of their phenotypic spectrum, is probably even a much larger number. So one question you could sort of reflect on is all these different genes, Mendelian genes, do they all carry the same evidence? Are we really convinced that each of them is really solidly recognized as a disease gene. They might be on testing panels, but I think we rarely go back and sort of review what's the actual evidence. Just because something is published doesn't necessarily mean it's really true. As scientists, we have to question things all the time. So about 5-6 years ago, I got I became aware of this effort called ClinGen. It's a NIH-funded, somewhat US-centric initiative, but also very international, actually. That is around for over 10 years, and they try to answer exactly these questions, mainly by reviewing literature. They try to find, they establish frameworks, and they're pretty rigid about them, to explore the question, is a gene, what's the validity of a gene to be called a disease gene? And which genes, therefore, should be on testing panels. And then they go further and do the same for variants. The framework they have is acknowledged by the FDA. So that was another attraction for me to work with this group, because when it's acknowledged by the FDA, it seemed like it's an easier than sort of jump off pad to gene therapies and companies and potentially future approval of therapies. Because when you think about it, any future therapy will need to take into account, like you don't want to treat somebody who has a mutation where you cannot be absolutely sure that this is the causative change in that person. So you don't want to treat the wrong variant, so to say. So that's very important to consider. And there are about 60 working groups that cover many large spectra of diseases, but as Crystal just said, curiously, HSP is not part of this. So when I got involved, we established a working group which is mainly based on volunteers for the CMT, Charcot-Marie-Tooth disease, which are peripheral neuropathies. It's quite similar in concept, this disease group, to the HSP group. CMTs are also very heterogeneous. It's over 100 genes now. And there's still many to probably be discovered. It's a bit more common than HSP. It's not quite as rare. So again, standardized approach that every member of the working group has to sort of learn. And then during COVID especially, we had endless Zoom meetings, and it was actually a great experience to bring together all the evidence from the literature for each single gene, CMT gene. So we did this for 70 CMT genes overall, and it's kind of a point system. And in the end, you come to a conclusion and you say, okay, this is a definite gene, this is strong, moderate, or the evidence is actually limited for a gene. And then you can also decide that a gene is disputed or even refuted as a disease gene. So here are the genes we looked at for CMT. As I said, this took us several years. And maybe somewhat surprising, like 2 genes we basically refuted. And I'm sure there are gene panels out there that still carry these genes. So when you look at them, one of is MET25. The story is that over 10 years ago, even longer, 15 years ago, a very large pedigree from Costa Rica was studied, and there was very strong linkage data, and the group really tried to find the underlying gene, and they found one variant in this gene MET25, which ultimately they thought this is the gene, and it was published. It got a designation as CMT2B2. But then over a long time, no second family or case ever appeared. And then about 9 or 10 years after this, another publication came out from the same group, actually. And they said, well, actually, it's not MET25. It seems to be this new gene, PNKP, which was published with a somewhat more complex phenotype, which also included ataxia. When they went back to their patients, they actually noticed there were elements of ataxia, cerebellar ataxia in these cases. So MET25 was even acknowledged by the original group that this is not a disease gene. And the other story is KIF1B, was published some 20 years ago, 25 years ago almost, to cause a linked locus called SIMT2A. And this was actually on the cover of Cell. So a pretty important impact. I suppose there was a mouse model. There were two small families from Japan. But there were no additional cases that ever really appeared or were published. But in fact, mitofusin 2 is a gene that was right next to KIF1B, was found to explain almost 20% of CMT2 cases, and those linked with CMT2A, all of them. So mitofusin 2 is the real gene, and KIF1B is actually not a disease gene. Yet, when you look in OMIM today, KIF1B is still there. It's called CMT2A1. So this is how this persists. It's very hard to take this off the literature. So another interesting concept that Klingen introduced is what they call lumping and splitting. Okay, first I thought that's going to be somewhat rare, probably. When you look in OMIM, you will notice sometimes there are multiple phenotypes that are linked or associated with one gene. So it turned out of all these 70 CMT genes, we looked at half of them, we ended up either lumping or splitting. So it is an important concept. And what does it mean, lumping or splitting? Sometimes it's straightforward, and sometimes we literally spend like an hour discussing it. Like really people who knew the field discussing 1 hour if they should lump or split. So the idea is really you would split something when there are distinct pathomechanisms, really distinct phenotypes, or different inheritance patterns like recessive or dominant. And lumping generally means there is a spectrum. There's some sort of continuity between these patients. So for instance, early and late onset would be a spectrum, you could say. But it's just not always as straightforward. So some decision making has to happen at some point. So here's an example. This is ATP1A1. It's a dominant CMT2 gene, or at least it was first described as a CMT2 gene. So missense changes, all missense changes cause pretty pure type of CMT2. Then pretty quickly a family was described that carried a pure HSP type with a unique different missense change. Okay, that remains to be the only case, I believe. And once a gene is published, as you know, people pay attention, and we were contacted by other groups. They saw a very different phenotype. In early onset, more like a neurodevelopmental phenotype, but with missense changes in the same gene. So turns out all those cases were actually de novo, which is interesting. So when you map them out on a protein model like here, you actually see there is some clustering happening there. So that's good. That could explain that maybe a different disease mechanisms involved here. Nevertheless, they both missense, they are dominant, obviously, and yet we decided to split it. So they should be considered two different diseases. Okay. Now, here's a little practical thing when you think deeper about these problems. If you decide to split out the HSP phenotype in this situation, there's only one reported case. It surely will end up being as limited evidence. Okay, so that's maybe not what you want. So there is sometimes this bit of a drive to tag it on if possible, so it gets a more definite answer. Okay, so a lot has to do with literature review, and of course a review of individual variation, but it's the gene concept when you talk about validity of a gene as a disease gene. But very much linked to this whole problem is, of course, the validity of the variants themselves. So I call this sometimes the Titan problem. So titin, as most of you know, is one of the largest proteins in a human genome. It's humongous. It causes different disease phenotypes. The point is, it is so large, this gene, that I bet nearly everyone in this room has some variant in titin. And that's just by chance. That's just by chance. So it's going to be very different if there's a random rare missense change to decide, is this really causing disease or not? And it's so big that you can't, it's very hard to model and so on and so forth. So this general concept of the titin problem is that as the testing space gets larger, and now it's pretty routine to test whole exomes, although it probably is smart still to pull out virtual panels. And if not only for the reason that you really don't want to, just mathematically, the bigger the spaces you look at, the more stuff you find, and then you don't know what to say. So anyway, more genes testing, we will see more of these variants of uncertain significance. There's some papers that estimate this to be a very significant problem. When you do large panel testing in a lot of people, you find a lot of VUS. So much so that there are now calls in the field, and I think some labs practices actually, they do not report out the VUSs anymore. So you cover up the problem in a way. So that's certainly a big issue for clinicians, I would say, because a patient is sort of left in limbo. They have something there, say in SPG4. The phenotype seems to fit, but you're just not certain. Is this the reason or is it not? So, and maybe a bit more sort of thinking about first principles here. If you take any gene, here it's mitofusin 2, that is certainly linked to disease. Yes, there are variants in there that are pathogenic or likely pathogenic, surely. There are surely, there are variants there that ought to be benign. Every gene has benign variants. So this is clear. And maybe in some genes this is just that. But arguably there is got to be some sort of middle field where the linkage is not so clear. Maybe the penetrance is low, or it has a sort of later onset. Such variants often come out as VUSs. You just not know what to call them. And that's what we deal with here when we talk about this variant-focused VUS problem. So generally in medical genetics, in the last 10, 15 years, labs have tried to really standardize how they look at these variants and determine pathogenicity. And in the US at least, and I think internationally also, the ACMG-AMP criteria have gained a lot of sort of appreciation. It's a human-made system that gets quite sophisticated and detailed to quite rigidly, in a way, figure out is something pathogenic or not. And it's also pretty much a point system. So, and this gets further refined sometimes, or adjusted to specific diseases even sometimes, or specific genes, I should say. So over the last few years, we have seen a rise of another approach to this, which is sort of machine learning or AI methodology. And in the extreme of sort of this large language model, AI-driven approach, these approaches to this problem, they're much more black boxed. So this is software that basically tells you this is pathogenic. And it can maybe, the better ones can tell you why it thinks it that way, but ultimately it's not always so satisfying or convincing, let's say. These systems are based on the idea that when you think about DNA, it's a language with 4 letters, right? Or the protein space is a language with 20 amino acids. So technically, you can imagine if LLMs can learn human language from reading the internet, maybe they can learn genetic language by reading DNA. And so maybe they can look at a lot of DNA and then start reasoning and see things and patterns that humans have missed so far or don't understand. So, and this is obviously in development, this field, and there are different approaches to it, like alpha missense or Maverick. Maverick, something we developed in Miami. They pretty much trained on data that humans have rated before. So they learn what humans think is pathogenic, and then you let them look at other things, and they decide if it's pathogenic or not. Something different is Evo2. That's a system where they basically feed this algorithm millions of genomes. Many of these genomes are actually from bacteria. And they basically try to really teach it DNA language, like you would teach reading to a 5-year-old or so. So we'll see. There's, as you know, there's a lot of movement in the AI field. These worlds are not necessarily so compatible, actually. And it's going to be quite interesting to see what happens. I probably think the most likely is we'll first see more and more of a convergence before, or maybe never, the machines take over. So since we did this in Miami, I show you a slide here on the Maverick. This was really trained on a sort of an LLM. And I was actually shocked how well it does. It's already published 3 years ago. I thought this is not going to be so good, but it turns out it does a good job. It's still the only system, I believe, that can differentiate between dominant and recessive effects. So that's very useful for these things we use it for. And it's actually even quite good to look at genes that are not known disease genes and make good guesses if they could be disease genes. So we use it almost every day to have a preliminary list or ranking, I should say, of genes. And just to give you an idea how well it does when you try to compare it, here's a long list of tools that people use routinely to try to rank variants and genes. And the top blue lines are actually Maverick. So it really outperforms out of the box pretty much all the traditional tools. And other things, when you gain some confidence in a system like this, what you can do is sort of in silico, you can predict every single potential missense change in, say, spastin. And so here, so spastin is a dominant gene, so you should not pay attention to the middle line, just to the middle box, just the upper and the lower. And the upper is the dominant score. So a high score is pathogenic, and the red dots are known pathogenic changes in spastin. And you see that quite reliably, it ranks the red dots, the missense variants that are red with very high scores. So if it does this to the known missense, very likely the gray ones have never been observed in the wild yet. They're probably also quite accurate. So you can have scores in advance even, before you even observe a variant. Okay, so to wrap this up, there are different approaches to improve EOS. This is probably not a complete list by any means. I think what really is still the number one, and probably will remain the number one, is genetic evidence. Genetic evidence. So observing multiple cases with different or the same variant, unrelated cases with mutations in the same gene and the same phenotype, that's still key. So sites like ClinVar, where clinical labs upload their observations, or we run the Genesis project, and there are many other databases like this where you can match, do sort of gene or variant matching. That, to me, that's still the best evidence. Animal models are fantastic, but they can also be misleading. So animal models, of course, are important. I think specific assays can be fantastic if they're good. But with an assay, you have to think that it really measures something very specific, usually at the biochemical or molecular level, and it has to link all the way up to a phenotype. And that sort of leap of faith is not always there. So, but assays can be great. And of course, increasingly maybe protein modeling, which has become much easier, also thanks to AI. The assays, when you trust them, you can actually scale them up into something which is called MAVE. These are massive undertakings where you do mutation saturation of a gene. In a single tube, you can generate all the possible variants in this gene, and then you apply it to an assay, and you have a complete readout for all the variants in a gene, and you basically predict their effect size in a certain assay. So has that been for HSP? I checked last night. I think there has not been a single MAVE experiment in an HSP gene yet, but I might be mistaken. So 1 minute. I know behind. So very important question. Should we do more of this in HSP? That was the right answer. So rhetorical question. But I have to say, after 5 years of doing this for CMT, I felt a little burned out. That's also true. It's too many Zoom meetings. You know, when you have like 5-hour Zoom meetings in a row. But just in recent months, I'm starting to warm up a little bit to the idea that maybe we should do at least a gene curation for HSP, just to be on the list, right, Crystal? So I might reach out to some of you for volunteers, and it could be, it's a fabulous work actually for all the younger colleagues to do this. And also, as you might have heard, we got very successful in the US recently to get funding for a brand new network of clinical research centers for HSP. We call it the SP-CERN. Darius is here in the room. He will speak later. He's our fearless PI. And so we have funding there. There is also a genetics center is funded. And actually, this is sort of part of the work that the genetics group is supposed to do. So I think we have to do something in this regard in terms of gene curation as a broad topic. And yeah, we close to publishing this. And for all the friends of the German language, and as a joke from last night. Okay. So, thanks.
Thank you very much. So the bottom line is more actions, less meetings. Or so I will ask Antonio to come, and we have 5 minutes. We are already late, but I have Henry's agreement to have 5 minutes more delay to have questions. So do you have questions in the room? Otherwise, I start with the gene creation. Sorry, it's my passion at the moment. Oh, there is, so I can't ask my question.
Very nice talks, Jürgen Winkler. My name Erlangen. 50% still unknown. I mean, it's great for geneticists to define genes, and then there are certain neurodegenerative disorders where there was initially a belief that everything is genetics. Who should I ask more than you both geneticists? What's beyond genes in HSP?
Beyond genes is the key concept of Beyond genes is the main point of the presentation. So the fact that there are a convergence of similar cellular pathway, even from different genes, and that cellular pathway should be, by my point of view, the main point in which we should point a sort of precision medicine. This is the critical point. Even if we have many types, many genes, and many types of HSP, the fact that there are few mechanisms related to the disorder, that should be the future in terms of potential curation of the disease. So in this context, all the technical applications that are proposed by Stefan, by my point of view, should be directed with this type of cellular mechanism, and this is the critical point.
I'll follow up with a quick question, Darius from Boston. And actually, it's a question for both of you. I noticed in your introduction, and I think we all do this, we call HSP a neurodegenerative disease, but I think it's only part of the truth. Some childhood onset forms clearly have a neurodevelopmental component. I think you showed the thin corpus callosum and SPG11. This is something that is present long before symptoms occur. So it's probably developmental. So my question is, another dimension of thinking about how genes cause disease is how they're expressed in development or throughout time. Is that something that you have started to look at for many of the HSP-related genes? I think it's something that we as a field have ignored for now.
Yeah, I agree with you. I mean, it's difficult. This is, I told during the presentation that is an atypical monogenic disease because many genes is now demonstrated that can cause even related or different neurodegenerative disorder. For example, the presence of thin corpus callosum is demonstrated in different type of HSP. So this it means that maybe other type of factor, we don't know, maybe epigenetic factor, maybe environmental factor or something like that, at least in some type of HSP might interfere in some way with the development of the disease.
Yeah, some sort of vulnerability that is there and then leads to an early degenerative process or so. Yeah. For the other question, I would also add that I see HSP in a spectrum, like the motor neuron disease spectrum, right? I think we all agree, ALS. And we're doing better than ALS, okay? But it's so curious that we cannot answer these. Why do people get ALS? In 9 out of 10 cases, we have no idea. And that's probably, if we knew, we probably could answer your question better. There is something we are missing, yeah.
So many questions. I'm not sure I can allow all of them.
Hi, Jonas Sauchi from Brazil. To Stefan, I'd like to know how the field is moving in CMT regarding this curation on ClinGen. It seems like a much more scientific community task, like you see limited evidence for some genes or even refuted, and how, when you find limited evidence, the field is moving, try to answer these genetic gaps to really move forward or refute. So how's it going on CMT, and how can we learn from that when we start in HSPs?
Well, one thing is clear that the evidence is mostly drawn from peer-reviewed literature. I mean, you can draw from other sources, but I think that's still the best source. So we need to keep publishing. We need to also consider publishing allelic series. And time is another factor. We often oh my God, we haven't found evidence. It's already 4 years since the discovery. But 4 years is kind of nothing in the big scheme. So sometimes it takes 10 years to get the evidence. So, but I would say it's very important that we're not giving up on publishing, even though it's maybe a routine thing in a clinical lab. Maybe clinical labs should partner and publish their results also from time to time. That would be a great service.
Cahar O'Kane, I work with Drosophila, so I'm a bit of an outsider for the clinical work. But you mentioned earlier model organisms, and I think Stefan also mentioned even a lot of work with cell biology and I guess iPSCs, and there's a lot of beautiful work that you described. But as an outsider, a gap that I feel aware of is that so we see lots of nice ER phenotypes in Drosophila, axon ER phenotypes, and we look at morphological phenotypes of ER and look at physiological phenotypes. But people ask me, what happens to ER in patient axons, and I've no idea. So I think that feels like there's a gap between, say, all the things we talked about already, model organisms, cell biology, in vivo genetics, knowing what goes on in real cells in patients, which I guess you have to have an understanding of to be able to think more rationally about therapy. I don't think it's unique to HSP, but I'm just wondering, are there any ways in which you could try to bridge that gap?
Yeah, the closest we have, as you know, probably is eye neurons derived from iPSCs, but it's in vivo in humans is very difficult, right?
Yeah, I believe that the translation in humans, by my point of view, will create a really very problem, and maybe many things will change drastically, even if we use the best method in laboratory. This is my opinion, but.
So I'm afraid I have to interrupt this interesting discussion. In the interest of time, we need to move to the next speaker. I'm sorry. Thank you so much. So the next speaker is Enera Salazar Villacorta. She has a double affiliation in Spain and in London. And she will present part of a PhD project. And I have to change the...
Okay, perfect. Okay, so nice to be here. Thank you for the invitation. So I'm going to present part of my PhD. So it's a work I did while I was in the Institute of Neurology in UCL. And this is a collaborative work. So I was part of it, but thanks to the work of a lot of different groups, it's the reason that we bring it here today. And I left a question mark there because I think it links very good with the previous talk. So because this, what I'm going to present now, it's the work on very few families, but I hope that in the future we could find more families and further support this work. Okay, so, okay, it's not working this. Okay. Was working before? Maybe, or I can do it like this, no problem. Okay, so this is the outline of the talk. So I'm going to give a brief introduction, and then I will summarize the recent advances in childhood HSP genetics. I'm going to try to convince you that FITM2 could be a novel cause of HSP, and then I will give the conclusions of the work, and I will mention some current projects in HSP genetics. So, as we heard before, the genetic architecture of HSP is highly heterogeneous, with more than 80 genes described causing this phenotype. And the most frequent implicated genes in children HSP include SPG11, SPG15, KIF1A, ATL1, SPAST, and CYP7B1. Okay, thank you. Okay, so as the first slide mentioned, there is a huge genetic heterogeneity on HSP. There is a broad phenotypic spectrum. I think this is the lumping that was described before, and these splittings. And because it was also difficult for me to describe this, because HSP genes can cause different phenotypes, but also overlap with other neurological disorders. So sometimes in a child it's really complex to define the main phenotype. So also we have a high rate of misdiagnosis, because initially these children could look like having a cerebral palsy, and this is mainly because they show an evolving phenotype with a slow phenotypic evolution. So, as mentioned before, there is a variable NGS diagnostic yield, around 30–60%, and is significantly lower in pure HSP. So, over the last 5 years, we have had different advances in childhood HSP genetics. So, I have selected some articles that were published and further explaining the genotypic heterogeneity and also expanding the phenotypic spectrum. So over this session, I will talk about FITM2, and the next speaker will talk about SPAST. So FITM2 gene, it's a gene involved in lipid metabolism, and it encodes FIT2, that is a protein that acts at ER lipid dephosphatase. So FIT2 activity, let me see, okay, here. So you can see here the FIT2 protein, that it's involved close to BCL2, that it's another gene that most of you will know that also causes HSP in the lipid droplet bud gene, and it also involved in maintaining the curvature of the ER, so it helps with ER homeostasis. So cells lacking FIT2 show a normal lipid droplet formation, lipid metabolism defects, and ER stress. When we delete FIT2 encoding gene in C. elegans or Mos musculus is lethal, and the complete deficiency of FITM2 in humans causes the Siddiqui syndrome. So for Siddiqui syndrome, it's an early onset deafness dystonia syndrome that was described in 2017 by Zazo-Seco et al., and there are 5 families reported in the literature. So the main characteristics of this syndrome is the autosomal recessive inheritance, the hearing impairment that is present in all the reported patients, ichthyosis that it's also present in most of them, dystonia that often is early and severe, neurodevelopmental delay or intellectual disability, and poor overall growth. So first of all, I'm going to talk about the first family that prompted this work. So this is the second child of a consanguineous family that had a premature birth and normal development, and around 9 years started to have difficulties with coordinations and balance. She showed normal hearing and not any skin lesions. So this is the exam of the child when she was 16 years old, and you can see she showed mild dysmetria on the finger to nose, and mild dysdiadochokinesis is not the best video, I may say, to show that, but it was present. And then she showed also nystagmus and slow saccadic pursuit, and a clear spastic gait with catches, extensor, Babinski-Hoffman sign, and hyperreflexia. Okay. So we performed multiple investigations to try to elucidate the case, the cause, and the first MRI on her early teens was reported as normal, and then from mild mid-teens, it was shown to have cerebellar atrophy, predominantly of the vermis. There were done multiple tests that I have not included here, but all of them were negative, and we found a compound head FITM2 variants that, knowing about the function of the gene and that was acting closely to BCL2, we thought that it could be a good fit for this family. So what we did is look at our internal database and submitted the case in GeneMatcher, and we found the second family. So the second family, it's a family that has 2 affected siblings. Both of them had a premature birth and normal development. Only they shown strabismus that needed surgery before 10 years. And then from 10 years, they started to present a slow and steady gait and needed assistance to walk. And on the exam, it was present a spastic gait and limb ataxia. And over their 20s to their 40s, they progress significantly with, and they show a severe tetraparesis and severe dysarthria, and also severe limb ataxia. And around their 40s, that is when we did the last examination, they were wheelchair-borne and dependent, and then they show a tetraplegia, anarthria, and as you can see, the SARA and SPR scores are almost in the limit of these scales. So this is the video of the last assessment of these patients. I shortened it, but as you can see, they have a severe, this is the second sibling, so a severe tetraplegia and dysmetria. They also had slow saccades and difficulties with upwards gaze, and also this diadochokinesis, and you can see the contractures and the difficulties on the movement. On the exam, there was also mild rigidity, which was also interesting to see. Okay, so in both families, we found one variant that was the same, that is the glycine 100 arginine, and then each of the families have a different variant. So family 1 had a Framshift variant, and family 2 a nonsense variant. We perform segregation of the variants, and it segregates, and then we perform homozygosity mapping to see if the common variant arose from the same common ancestor, but we couldn't find any region of homozygosity shared between them. So here you can see the structure of FIT2 protein. So this protein has 6 transmembrane domains, and there are 2 very important histidine residues that are really important for the catalytic function of the enzyme. So as you can see with the first variant, the framesheet, this framesheet leads to a premature stop codon and causes the loss of one of these histidine residues. So and it's classified as likely pathogenic. The second variant it's similar. It's a nonsense mutation that also causes the loss of the second histidine residue, and it's also reported as likely pathogenic. And the third variant that was common in both families, it's a BUS, and it's a missense mutation, and it's supposed to be deleterious, but we wanted to investigate this further, and is what I'm going to show you now. So this part of the work was done by US collaborators, and they express and purify the FIT2 protein, and they analyze by single particle cryo-electron microscope analysis. So this is the density map of, can you see when I, no. Okay, don't worry. So this is, like this? No. Okay, the density map, and what they did is they combined the density map with AlphaFold predictions, and they then have better insight about the protein. It's true that I'm not sharing here all the results we have on this, but if you want to read further are on the paper. So as you can see, and I mentioned before, it has 6 transmembrane regions. One of these transmembrane regions have the 2 histidine residues that are really close to the luminal region. So as you can see, the mutations are mapped here, and both of the first mutations are thought to inactive the enzyme. And what about the second mutation? So structurally, here you have the wild type and the prediction that the G100R will cause in the protein, and you can see that in G100R will clash with different amino acids that are close to the enzyme that is changed, to the amino acid that is changed. So this was also investigated in the different positions, the different rotamers of the amino acid, and in all of them it was non-stable. So we did some further tests to evaluate this variant. The first one was checking if the mutant was showed reduced FIT2 proteins level in patient's fibroblasts. We performed mRNA quantification, and there was not a significant change. But when we checked the protein levels in the patient fibroblasts, it was really strikingly reduced compared to the controls, and the protein that we got from this analysis, it was of full length, so it arose from the G100R allele. And with these experiments, we hypothesized that G100R substitution destabilizes the protein and causes its degradation. And then we wanted to assess if, even if the protein is reduced, it's still active, it's changing the activity. So what we did was overexpress wild type, the mutants, and a catalytic inactive mutant in this gene, these cells that are specialized to produce proteins. And we showed that the mutant, the G100R mutant, was present at lower protein amounts than the wild type or the catalytic inactive mutant, and that the assays of the microsomes that were isolated for acyl-CoA dephosphatase activity revealed that G100R exhibited less activity than the wild type protein. So with this, we conclude that the G100R does not really reduce FIT2 activity, but reduces the amount of expressed protein. So what we wanted to further analyze this allele, and we did 2 experiments in different cellular models. So we expressed FIT2 wild type and the mutant in FIT2 knockout cells, and the complementation, so the adding of FIT2 G100R generated lower levels of FIT2 protein than the adding the wild type protein. And this was further confirmed in fluorescence-based assays that where also the expression of the protein was lower, and the add-back of the FIT2 wild type rescue the ear morphology. And also, I don't know if you can see in the images, but there are some dots that are more bright. So these are the ear walls that, and when we add back the G100R, it could partially rescue this phenotype. So we wanted also to quantify if what we were seeing, it could be also measured, and we measured 2 stress ear stress proteins, BIP and ATF3, and again we saw that G100R only partially complemented the UPR induction phenotype. So with this, we made the conclusion that FIT2 G100R variant results in less FIT2 enzyme than the FIT2 wild type, and that this reduction in FIT2 protein levels results in ear dysfunction in a human cell line. And we did a replication also of this experiment, but in yeast. So the ortholog for FITM2 in yeast is SCS3, and it has different phenotypes, including the inositol auxotrophy. So we ask ourselves if the mutant proteins could rescue this phenotype, and the expression of wild-type human FIT2 rescue the auxotrophy in the genes that were deleted for the FITM2 ortholog, and the G100R only partially rescue the inositol auxotrophy. So this and other experiments that I'm not showing here make us have the conclusion that this variant shows reduced function compared to wild type, only partially rescuing the deleting SCD3 yeast phenotype, indicating that lower FIT2 protein levels impair FIT2 activity. So these are the conclusions of this work. So we thought that biochemical analysis of the G100R variant together with data from patient fibroblasts, culture mammal cells, yeast indicate that the protein retains activity but is expressed at lower levels than the wild-type protein, consistent with this allele being hypomorphic. And this is a simplistic way to present this work, and I think obviously this will expand further in the future, but our results identify a previously unreported hypomorphic FIT2 allele in combination with a null allele associated with a complex HSP phenotype. And this phenotype contrasts with the previous reported one causing deafness dystonia syndrome. And I wanted to also add this slide here because another group has published recently a third family. So this is the summary of the Siddiqui syndrome reported cases. These are the two families that we are reporting, and there is another family also showing an HSP phenotype. So I think we will see more cases in the future, or hopefully we will see them, and we could define FITM2 as a new causative gene for HSP. So these are current projects in HSP genetics that we are undertaking, and I suppose some of the first one, the IOM projects, I'm not involved in, but are part of the group I was collaborating when I did this work. And if the other ones that are more my PhD projects or part of my PhD, I will be very happy if you have any insight about them or any further patients. And thank you very much. I want to thank especially the patients and families that were involved in this work. These are the people that was involved in this work, and as I mentioned, it's a collaborative work that took quite a lot of time and effort, and these are the current groups I'm working with now. So thank you very much, and yeah.
Can you hear me? Yeah. Thank you very much. I just been informed that there will be an alarm testing today at 11. Hopefully we'll have the lunch break at the time. I had one question for you before you go. Have you considered that this specific variant actually does not lead to an hypomorphic effect, but it's an enzyme, right? So that it could actually produce something that is wrong? Like, do you plan to do metabolomic studies to see if there is a substrate that should not be there? Or
It's a very interesting question, and I think it's something that definitely needs further assessment. We have not done it, but yeah, it's interesting to do it. And
because all these mutations, they, as you said, the KO is lethal in different organisms. So probably all the mutations that you see are compatible with life. So they are all hypomorphic in some ways. So this specific one maybe have a different effect. I have to introduce the next speaker, and maybe we have the question afterwards. So the next speaker is Manon de Goutin, sorry. Now it's easier. Manon de Goutin, so she works in Bordeaux with Cyril Coiset and Giovanni Stevanin, and she's a resident in medical genetics, and she will present also part of her PhD project.
Just to say some what about the alarm testing? So at 11 o'clock, your cell phones will do a horrible sound. Yeah. So your cell phones at 11 o'clock will do a horrible sound. It's just a test. So don't worry, it's just a test. It's all in Germany and in all the German parts.
The floor is yours, Manon.
Thank you.
You will be interrupted. So.
Yeah, it will be an experience. Okay. So I'm really pleased to be there today to present my work about the phenotypic spectrum associated with biallelic SPAST variants. So let's start with a brief introduction. As many of you know, pathogenic variants in SPAST are well known to be responsible for spastic paraplegia type 4 or SPG4. It's the most frequent form of spastic paraplegia with dominant or sporadic inheritance, and it most often presents as a pure spastic paraplegia, but some complex forms exist with cerebellar ataxia, neurodevelopmental disorders, or seizures related to, notably, to missense de novo mutation. And a hallmark of SPG4 is its high inter- and intrafamilial variability, up to incomplete penetrance in some families. And typically, we observe two peaks in the disease incidence in the first decade and between 30 and 50 years. The SPAS gene codes for spastin, an enzyme involved in microtubule cleavage via an ATPase activity, thanks to its AAA domain at the C-terminal extremity of the protein. And in fact, microtubule cleavage allows for better elongation and stability of the microtubule network through the incorporation of GTP tubulin notably. And spastin is also involved in diverse other cellular processes, such as lipid metabolism, through interaction with the endoplasmic reticulum and lipid droplets. Spastin exists in two different isoforms, the ubiquitous M87 isoform and the M1 isoform, which is enriched in the central nervous system. And the difference between the two is related to the presence of an N-terminal domain here, responsible for interaction and shaping of the ER, and the other domain are shared between the two isoforms, so the microtubule interacting and trafficking domain, and microtubule binding domain and the AA domain necessary for proper microtubule cleavage to bind and catalyze the cleavage. The majority of pathogenic variants are loss of function mutation, while missense variant clusters into the AA domain. So our investigation started with a specific family with Mr. A, here, 55 years old, came to our clinic for a consultation because he was experiencing walking difficulties with a familial history of SPG4 in their family with a known nonsense mutation in other relatives. And our clinical examination revealed a pyramidal syndrome of the lower limbs, so very probably SPG4. But during consultation, he was accompanied by his wife, who was 50 years old, who is also his first-degree cousin, so consanguinity in the family. So she was also at risk for SPG4, and when we asked her, she told us that she had some pain in legs and cramps, and upon clinical examination, we also found a pyramidal syndrome of lower limbs, so also probably SPG4 in her. And we looked back to the pedigree and found that they had two sons who died 15 years earlier, at 15 years and 20 years old, from a neurodegenerative encephalopathy with spastic tetraparesis without genetic cause identified previously. So we took samples of Mr. and Mrs. A, and as expected, they were both carriers of the familial nonsense variant. And this made us wonder about the genetic status of their two sons. Fortunately, we still had DNA from both children in the lab, so we sent it to the lab for Sanger sequencing. We showed that, as we suspected, both children were homozygous for the familial nonsense mutation. We also performed quartet exome sequencing, which did not identify any other genetic cause for their symptoms. So we were very interested to know if this homozygous variant could be responsible for this severe phenotype in the two children. So we turned to the literature to know what was known about biallelic SPAST variants. And at this time in PubMed, there were very few descriptions of biallelic variants in SPAST. De Bott et al. Described two brothers with pure spastic paraparesis with an age at onset around 30 years and with parents who stayed asymptomatic until an old age. Cruz, Camino, and Hall described a brother and sister with developmental and epileptic encephalopathy with spastic tetraparesis and with heterozygous relatives presenting with pure spastic paraparesis. And in addition, the S44L variant has been known for many years as a common hypomorphic variant, non-pathogenic in the heterozygous or homozygous state, but a factor of severity when located in trance of a pathogenic mutation. And this variant could explain part of the intrafamilial variability observed in the family. So very few evidences and not a lot of clinical data about the impact of biallelic pathogenic variants in SPAST. That's why we wanted to describe more precisely this phenotype. So through national and international cooperation, we reunited a cohort of 9 patients from 6 families with homozygous variants in SPAST. Diagnosis was made by targeted Sanger sequencing in the 2 children of Bordeaux, spastic paraparesis NGS panel, and exome sequencing in the other children, and we described the phenotype retrospectively. On the genetic side, we identified 5 different variants. 2 were nonsense variants, already described as pathogenic on ClinVar. We also identified 3 homozygous missense variants, predicted to be pathogenic in silico, located in functional domains of the proteins and affecting conserved amino acids. One is located in the AAA domain, whereas the 2 others are located in the N-terminal domain and MIT domain. Clinically, the patients presented with a broad phenotypic spectrum with 2 different sets of symptoms. 5 patients presented with a pure spastic paraparesis with early onset in the infancy and without neurodevelopmental abnormalities, seizures, or brain MRI abnormalities. And their heterozygous parents were all described as asymptomatic. And on the other side, a severe phenotype of complex spastic paraparesis with severe to profound intellectual disability in 4 patients with very early onset and progressive spastic tetraparesis and with some heterozygous parents described with classic form of SPG4. And if we describe more precisely this severe phenotype, during the first months, motor acquisition were normal in some patients, while others presented with neonatal hypotonia. From 6 months to 18 months, spasticity begins, and seizures appear in some of them, up to developmental and epileptic encephalopathy with infantile spasm in 1 patient. During the first years, they presented with motor and cognitive regression. They lose independent gait or never acquire it at all, and they experience severe language regression with some of them who develop microcephaly. And by adolescence, in our 2 patients, they become bedridden with severe spastic tetraparesis, profound intellectual disability, and early deaths occur between 15 and 20 years of age due to inhalation pneumonia. More recently, a paper by Nolasco and al. Described a cohort of 5 individuals from 3 families with biallelic missense variants located in the AAA domain. Similar to our severe group, they noted a severe phenotype with profound intellectual disability in 4 patients and a patient with a milder phenotype with borderline IQ in 1. And in 2 of the 3 families, relative heterozygous for the pathogenic variants were symptomatic for SPG4. Here we are represented schematically on the proteins variant identified in our cohort and in literature, with missense variant in blue and nonsense variant in red. As is in heterozygous SPG4, missense variants are enriched in the AA domain, but we also observed in other functional domains of the protein. And here, variants responsible for pure spastic paraplegia phenotype are shown in green, and those associated with complex spastic paraplegia and profound intellectual disability in yellow. And as you can see, biallelic loss of function mutation are always responsible for a severe phenotype, while there is no strong genotype-phenotype correlation for missense variants, particularly according to the functional domain. Missense variants in the AA domain are responsible either for pure or complex phenotype. However, here I framed variant responsible for SPG4 symptoms in heterozygous relative of homozygous patients, and we can observe a strong correlation with the severity in homozygous patients. So overall, we observed that the presence of SPG4 symptoms in heterozygous relatives seems to directly correlate with the severity and intellectual disability seen in their homozygous children, with like a dose effect of the variant. The 153 C2G variant is interesting because it's a nonsense variant early in the protein located in the N-terminal domain. So in theory, it causes a total loss of the M1 isoform, the most common isoform in the brain, without affecting the ubiquitous M87 isoform. This N-terminal domain is specific, is particularly involved in interaction with the ER and lipid droplets, so alterations of this function could be sufficient to cause a clinical severe phenotype in patients. And here it's like the resume of our warning. It's to highlight this slide. Yeah. Je pense que mon téléphone est resté du coup. So rather than viewing SPAS-related phenotype as isolated clinical entities, our perspective is that they actually represent a continuous spectrum of severity, which could be driven by an allelic dose effect. We suggest that the clinical gradient is directly tied to the mutational burden. At the mildest end, Hypomorphic variants leave a residual spastic pool sufficient, and even at homozygous states, such as the S44L variant, but could modulate the phenotype related to other pathogenic variants. Further, other hypomorphic variants that drops the functional pool of spastin could manifest as a apparent recessive SPG4. Further is classical dominant SPG4 driven by a single strong pathogenic variant where haploinsufficiency or dominant negative effects triggers the classic SPG4. And finally, at the extreme right of our model, the homozygous state of this strong pathogenic variant leads to a severe infantile neurodegenerative disease, as we described in our cohort. So from our point of view, this gradient highlights a direct correlation between the variant copy number and the severity of the phenotype. And based on this, we could hypothesize the existence of a critical threshold effect regarding overall spastic activity for symptom onset. So to conclude, biallelic variants of SPAST are responsible for a phenotypic spectrum ranging from classic spastic paraparesis to infantile neurodegenerative syndrome. It's an important information for genetic counseling, particularly in families with consanguinity. And finally, total loss of the M1 isoform could be associated with a complex spastic paraparesis phenotype. And I would like to thank all the patients and their families for their trust and participation in this study. And a special thanks to my colleagues in Bordeaux, and as well as our national and international collaborators from Paris, Manipal, Montreal, and Alger. And thank you for your attention. And I'm happy to take any questions.
Thank you so much. We have time for 2 questions, and Anira, please come back to the stage. Is there any questions? No more like sound, please.
Hi, Tim from Kiel. You said on your last slide, homozygous severe variants. What about compound heterozygous severe variants? Do they need to be homozygous?
There is only one patient with compound heterozygous variant described. It's this patient in the Spanish cohort with borderline IQ and milder phenotype. So it's difficult to say, but if the heterozygous variant are responsible for SPG4 phenotype in relative, is in the 2 branch of the families, it could be responsible, in my opinion, to severe phenotype. But we don't have a lot of patients with compound heterozygous.
So, any questions? No? So we have a cover break now till 11:20. So the Probealarm was more precise than the German railway company, but...
And thank you so much to all speakers of this morning session again.
Den können wir nochmal aufhängen. Ich hab den ja als Folie dran gemacht gehabt.
Oder Sie sollen den nach vorne einblenden, sagen. Ja.
Très bien. Yes, yes, you can.
Ich muss doch nochmal ein Update hochladen.
Okay, dann ich wollte.
Stick nodig? Okay. Oh, and then Ingot net zo'n connector. Die hebben?
For an HD. Ne.
Darf ich? Ja, super.
almost finished the PhD, hopefully, and yeah, coming back to work.
Where are you going to go next in Spain?
So I'm going to work in the
Okay, we want to start the next session. Just give it 10 seconds. All right, while you still find your seats, the next session is on genomic technologies, a topic very dear to my heart. We have 3 great talks lined up, and we will start with Alexander Heuschen, who really doesn't need an introduction. He's truly a leader internationally for implementing new genome technologies. He's the director of the Radboud
University Medical Center.
University Medical Center. And anyway, I'll leave it there.
Not the director there, so it's surely not. That's a job no one wants.
You know.
Like director of the whole thing is you have to become a manager and politician, and I'm luckily not there. So yeah, I run a research group there, but I'm also the chief innovation officer of our department to indeed bring technologies, latest technologies to diagnostics. The slides on? Yes, perfect. So, I mean, first of all, full declaration, I'm the real outsider here. I guess it's fair to say I've never really worked on HSP, so bear with me. Don't ask any difficult questions. There's a lot of other experts in the room. I'm just a genomics person, a genomics guy. I'm fond of technology. And what fascinates me most is if we improve technology, then we actually do not only help patients and their families with HSP, but many, many other rare diseases have the same trouble and the same genetic and diagnostic odysseys. So with better technologies, we can help all rare disease patients and their families, and ideally not only in our center, but on European level and ideally on global level. So I'll speak about these latest developments. We particularly use one long-read genome sequencing technology from a company called PacBio. And if I may sound excited, you hopefully believe that that's not the only reason that we got some reagents for free for some of the experiments and projects that I'm showing here today. But I'll always show that for full disclosure. So if we discuss rare diseases, HSP, you're not alone. We heard it's around 80 different genes discovered so far for HSP, probably several others pending. But collectively, all rare diseases that we face are at least 8,000, some of which are ultra rare. But the reason that there's 8,000 of those makes them collectively a very common healthcare problem. And this may even still be an underestimate. We came to the lumper and splitter discussion, and then it comes down to nosology. Where does a disease start and where does a new disease start again or another disease end? So there's other estimates that even estimate that there's 15,000 rare diseases collectively. And collectively, 1 in 17 people is estimated to have to deal with a rare disease in their lifetime. And that means even for a small country like the Netherlands, there's roughly 1 million rare disease patients. For now, we think that the vast majority of rare diseases have a genetic component, or in fact are even genetically caused in a Mendelian or monogenic fashion. 3/4 of rare diseases are childhood diseases, and in a significant proportion of those childhood diseases, it's even a very severe childhood disease, and children can die from it. And even in the most modern labs in the world, we still face an absolute tragedy that genetics is not up to speed. We're simply not good at genetics, still not today, because in a lot of our laboratories, we still have to offer a portfolio of 10 to 15 different test modalities to give the best possible answers with the knowledge of today to all our patients across the spectrum of rare diseases. And a lot of those tests, and we saw that in some of the previous talks already, so far happen iteratively. So test 1 fails, second test is done, third test is done, and that is of course painful for everyone involved, and particular patients and families that have to wait until they finally get a real molecular answer. And this complex structure, the heterogeneity of the disease, but also this strange test modality scene, often leads to wrong diagnosis, either clinically but also molecularly. It's estimated that in up to 1/3 of patients, the initial diagnosis may be wrong. And I know there's patients and their families in the room. Maybe several of you also have experienced that. Throughout your life, you've not only seen one doctor that you trust, but also you have seen 10 other doctors. Some were helpful, some not so helpful. And that also is a tragedy on its own right. Not every rare disease patient meets their best doctor initially, early on in their rare disease career, if you say it like that. And then probably the problem that bugs most of us dramatically is, even with the best tests available at this very moment, still in half of our patients, we simply still lie to our patients. We don't give them the right answer that they deserve. And I think that has to really change dramatically, and I think that we share that passion that we have to move forward. I work in a big university medical center, and 15 years ago, or even 20 years ago, human genetics was always a very niche discipline in a medical center. Because genetics had almost nothing, very little to offer. There were always internal medicine doctors, there were surgeons, those were the real doctors, and they were considered serious. Rightly so, they're really good, but geneticists hardly had anything to offer. Occasionally a doctor that was responsible for a rare disease patient, after some MRI or some lab measurements, or maybe a muscle biopsy, consulted the geneticist, and the geneticist offered one test for a single gene, and in the vast majority of cases that was of course wrong. Then another test was done, and another genetic test, and it continued to be wrong. So let's say 15 years ago or so, genetics usually only gave an answer in 5% of cases, and really heavily relied on diseases that are recognizable clinically. Thanks to the advent of next-gen sequencing, and of course that was also mentioned in the previous talks, largely thanks to exome sequencing, that has changed completely. We don't need a dedicated genetic test for disease A or B. We can just sequence all genes at once. That also means we only need one doctor who has at least a very strong suspicion for a given rare disease, thinks, hey, this could well be genetic, and we just very fast in the entire odyssey do a genetic test that is rather complete. Conceptually, I think we never wanted to do exomes. We always wanted to do genome sequencing, just read the entire DNA of a given patient. And of course, that had led to really changing medicine in a way and putting human genetics much more at the forefront of medicine and medical care in big university medical centers. And a lot of that has been driven by the increase in throughput and coming down of price of sequencing full genomes. So if you look at our latest instruments from last year, we can now sequence overnight 64 genomes, so the full DNA of 64 patients for around €200 reagent cost per genome. That alone was impossible to think of 10 or 15 years ago. So that's wonderful. That's the best test we ever had in human genetics. But there's also a little tragedy to that. If we would only do this type of genome sequencing, we would still do a disservice to all our patients that we currently see, simply because what we now call a short-read genome, or standard genome, is not good enough. It's the best test we ever had. It's great. It's implemented in healthcare in many countries, but it's not good enough. In our own center, we still have 10 different tests that sometimes have to be added on top of genome sequencing for a specific disease, for a specific patient, to still get all the answers out of the genome. And there's many reasons why these short-read genomes are not enough, and I certainly don't even understand all reasons myself, but at least give you some of my ideas why we think short-read genomes are not good enough. And one has to do with the complexity of the human genome. We all carry a lot of variation in our genome. The vast majority in terms of number are so-called single nucleotide variants, so single letter differences in the DNA code. So where you have an A and I have a T on one specific position. But we also have 1 million insertions, deletions, so tiny pieces of DNA that are more or less. And we have so-called STRs, repeat expansion diseases. We all carry 1 million of those. And we also carry 25,000 so-called structure variants. So whenever more than 50 nucleotides, 50 DNA letters are affected, we call that a structure variant. And we only have very few, 25,000 only. But if you now look systematically how short-read genomes are able to detect all of those, it's fair to say that for these substitutions, short-read genomes are really quite good. Probably we can detect 90–95% of the ground truth, so the truly underlying variants in a given genome. But the tragedy starts to kick in once we look at these more complex variants, when more bases are affected. Probably indels, in particular repeat expansion, short reads can only pick up 60% or so. And the real tragedy comes once we look at structure variants. Probably there, the best method for short read sequencing only finds 40% of the ground truth, the real variation that is in the genome. So this is already showing some of the tragedy. While we call it whole genome sequencing, in the title I put the W in brackets, because there's many holes if I read such a genome. But the tragedy is even bigger, and should be of a bigger concern, if you just count up how many bases in an individual genome are affected by these variant types. So logically, the larger variant types, while they are fewer, can affect thousands of base pairs. And if you sum up how many variant bases an individual genome carries, of course, substitutions, it's 4 million variants only affecting 4 million bases, and we're quite good in finding those. But if you look at the structure variants, there is only 25,000, but because they individually can be very large, affect thousands or sometimes 100,000s of nucleotides, the sum in any given genome is certainly more than 20 million base pairs affected. And this is now the variant class where our detection methods are the poorest. So I think that, at least in a large proportion, explains why our standard technologies and our diagnostics in that way is not good enough. So in a way, short, brief summary, short read genomes miss a lot of variant bases in any given genome. And that is now about to change. And this is changing by new technologies called long-read genome sequencing. There's two technologies in the market. On the left, you see PakeBio's technology, a US company. On the right, you see Oxford Nanopore's technology, or the machines they built, a British company. And Ingo will give you much more details about that. I have never used ONT, but in a way, or importantly, conceptually, both technology can do something very similar. Instead of reading 100 or 200 bases at a time, as short-read sequencing does, these can read 100-fold longer pieces of DNA. Up until 3 or 5 years ago, the downside was it made a lot of mistakes. So you always had to choose to read short reads very accurately or long reads with a lot of noise and a lot of errors. That has now changed fundamentally. Long reads now reaches an accuracy that short reads had only before. So we now can read 20,000 letters at a time with short read accuracy. And for the first time, it's also individual DNA molecules that we read, different from the short read technologies, and I won't go into details why that is, but that also allows us to sequence not only the DNA, but we can even distinguish so-called methylated versus unmethylated cytosines. So it's basically the fifth base that is encoded as epigenetics in the DNA, and we only start grasping the relevance and potential importance for rare disease just for DNA modifications such as methylation now. So with these technologies around, we started investing in that, and by now we have 6 of these big machines. Unfortunately, they will be outdated again in 2 years or so, so we have to buy 6 other machines. That's part of our business somehow. But what these long-read technologies really do, I'll try to summarize here in one cartoon. So imagine the DNA that we all carry. Our genome is 6 million letters. And so far, we tried to arrange our genomes as a jigsaw puzzle. But those puzzle pieces were very tiny. And it seems that our genome is very complex. Imagine you do a jigsaw puzzle of a beautiful landscape here in Recklinghausen. Sorry, I'm from near Ruhr area, so actually I like this region of Germany a lot. Imagine there's blue sky. If there's blue sky, then sometimes you have 2,000 or so puzzle pieces that are all blue. Those are the ones that you hated as a child because they were so difficult. They're undistinguishable. So now imagine you do the same puzzle, but you make pieces 100-fold bigger. Then once of a sudden, almost every piece of the puzzle from the sky has something, a little tree or a little bird in it, and therefore makes it much more distinguishable. And that, in fact, is now what happens for long-read genome sequencing. We can just read much larger pieces of DNA. And with that, intrinsically, we have fewer gaps where the short reads simply fail to map at all. We can get continuity. So we not only look at one variant here, but we also know it's on the same chromosome as this variant and the next variant, and we'll come to the benefit of that. but it's also much better in complex regions. So we are better in detecting repeat expansions, and we can distinguish homologies, so extremely similar regions of the human genome with greater distinction. And most importantly for the rare disease field, and I think actually also for HSP, is our sensitivity to find so-called structural variants or repeat expansions has improved dramatically. And I think the field predicts that that will lead to a lot of new discoveries in the rare disease field in general. So in the remainder of the talk, I just brought, I think it's a total of 3 studies to convince ourselves, and maybe that also helps to convince some of you, that we need to use these long-read technologies in a real clinical setting. So how we did that is going back to mutations, variations in DNA that are already known to cause disease but are very tricky to detect. We did a quick study on that. Then, of course, clinically, but also scientifically, probably the most interesting and engaging studies we're doing is going to patients that so far remained undiagnosed, despite all the tests have been done in the past. If you now apply long-read genome sequencing to those, what is the percentage? Are there a few percent of patients where you now give a clear answer thanks to the better technologies? And then ultimately, in a way, again, a bit boring, but that's what our clinicians and on a prospective study, not to just jump on an innovation, just light-footed as a cowboy like me may do, you really have to do some thorough testing and blind yourself towards the outcome and then show that the newer technologies are better. So, how good are these long reads for known mutations already? And I'll just summarize that on 2 slides. So what we did is, in this phase of that study, we went to our diagnostic experts and we just kindly asked them, give us 100 samples, DNA samples from patients that all carry very difficult to detect mutations. Largely mutations where short-read genomes fail or where we a priori already know that we need a second and a third test to detect them at all. And here's the summary of that. And actually, people that know me know I'm extremely optimistic. I'm even surprised by how good it already worked, and this is all using tools from 2 years ago. We find 83% automatically with the tools available, and for 10% of variants, we had to optimize tools a little bit or visually inspect the data, so it's clearly still visible in the data. So that is fantastic, because usually you need a mix of 10 different technologies to find all of these variants, and now you have a single technology. But, unfortunately, as with a lot of new things, you also lose a few things, and we found 2 more systematic issues still with the data. So there is some so-called cytogenetic aberrations, including so-called Robertsonian translocations. So cytogenetic aberrations that are balanced, where you don't lose or gain material, but you have breakpoints in the centromere or near the centromere or other repeats. Those are still structures that are so difficult to detect by any sequencing method that we still fail to detect them now. We think we have a few new bioinformatic tricks that we can apply here, but I don't want to overpromise that yet. And to our surprise, we had one second category, and those are repeat expansions. While long reads and Stefan and several others here in the room have brilliant data from the last 2 or 3 years, it's really exploding, and long reads really show dramatic benefit for repeat expansions in general. There's a small set of repeat expansions where at least this technology particularly fails or has tremendous difficulties, I should rather say. And those are very long GA-rich repeat expansions or GA-rich repeats in general. And just by chance, we included several cases with Canvas syndrome, so their hallmark is RFC1 repeat expansions. And we also had one patient with Friedreich's ataxia, again an AG-rich repeat, and we failed to systematically find those. So that still needs to be improved, and there's some hints. We have 2 ideas how we can further improve upon that. But just to put it out there, it's not perfect yet for these specific variant types. Second study, and unfortunately it's not for HSP again, but it is for other severe neurodevelopmental diseases. So it's largely young patients with rather severe intellectual disability and other developmental problems. And there, we and many others have studied de novo mutations in greater detail. We know that very often both parents are healthy, and the disease is a really de novo occurring mutation in that one egg cell or the one sperm that was used to make the new generation. Just the high-level summary, and I'll show you more details on the next 2 slides. So we selected, and it's in particular Lot Snyder's block, a very talented young clinician in our center together with my postdoc Bart, who selected 100 undiagnosed cases that underwent arrays, exomes, and the vast majority even short-read genome sequencing. For now, I think we have a clear-cut diagnosis in 7 out of the 100, but very strong candidates in 12 additional ones, including one new repeat expansion. But going back one step for those that wonder, why do you at all do these trios? I mean, first of all, they're important in disease, and in particular in the outbred population, we still think that a lot of the undiagnosed cases indeed have a de novo mutation that has been missed in the past. So that's the clinical and the scientific rationale. But whenever we test a new technology, we also like to run these trio sequencing, simply because de novo mutations in the child are so extremely rare, they're an excellent test for any new technology. Because if the technology makes a few false positives in the child, they will once of a sudden also appear as seemingly de novo mutations. So if your technology is not accurate, and long reads were really not accurate 3 years ago, it's a great test. You would drown in false positives and you would hardly be able to find de novo mutations. And good news, I can cut that short, the data is now so clean and beautiful that we can really use it very effectively to find de novo mutations on all variant levels. So in a way, my prediction for a lot of the undiagnosed cases is we may have overlooked de novo variation, and in particular de novo structure variants, and I think those are the best keys to new discoveries. And particularly for the 98% of the genome that does not encode directly for proteins, the so-called regulatory regions of the human genome, my prediction is that at least in severe sporadic disease, the vast majority may be de novo structure variants that hit not a gene, but the regulatory landscape. I think we'll see a lot of new paradigms and new understanding coming just from those type of discoveries. Just to give you one slide on the real data of that study, so this is after a few filters, the de novo calls we see in 100 trios. And that fits what we know from literature very well. So it seems to be a very clean data set. So we found on average 67 substitutions de novo, 4 indels that are de novo, and on average 3 structure variants that are de novo calls. The beauty now of the long read data is, different from short reads, we can immediately face those. And this is just replicating biology that many others have published upon. We can now look how often is a de novo mutation happening on the paternal chromosome or on the maternal chromosome. That's intrinsic in the data, very easy to filter. And we just replicate that 80% of de novo point mutations are all paternal. It's also important for the more general public. We always ask the mothers not to be too old when conceiving children, but in fact it's also the fathers. They actually carry over more and more de novo point mutations to the offspring if fathers are older. So this new trend that some of the VIPs give fatherhood when age 70, I think it's a bit of a daunting trend, and we at least as scientists, we should tell and educate the public that old fathers, for many reasons, are also not great. And we can discuss that in greater detail. All fathers are loving fathers, don't get me wrong, but we have to educate. It's not blaming mothers to be too old when conceiving children. That's the important message here. And then just for those that want to see a bit more real data, here are some. The bold ones are the ones where we have firm new diagnosis based on the data. Some are not really new just because of long reads. It's also the gain in knowledge. So Christel's one of your favorites, or 2 of your favorites are there, the RNU genes. When we originally analyzed the genomes of those patients, those papers and manuscripts were not out. By now revisiting data, even if they come from long reads, we now find stuff that short reads could have found already. Lastly, one more study where we try to explore how much extra can we find in undiagnosed cases is another study of 100 genomes. And this is a cohort we called It Must Be Genetic cohort. And of course, we largely, I mean, Stefan has the same. For almost every genome we sequence, we think there must be a genetic basis in it. But let's be honest, there's some cohorts or some samples and patients where we're even more convinced it's a true monogenic disease. So what we did is we asked all the expert doctors on the campus involved in our rare disease meetings and said, okay, give us only this one patient that is undiagnosed where you bet your monthly salary that it is a true monogenic defect. And we asked for those where you had a single heterozygous mutation and a recessive gene that fits very well. Think of Usher syndrome. We asked for those where you had a lab measurement that confirms it's this one specific gene or locus or 2 genes. So those we included, and here's just 2 examples of that. So we had very severe immune cases, CGD, And there's only 5 genes published, and we even had functional evidence that there's ROS production deficits in the patient. So it's very clear-cut diagnosis where the immunologist really said it must be these 5 genes, including NCF1, but NCF1 has a pseudogene and has very strong homology elsewhere in the genome. This specific same DNA we tested here was sent away to Barcelona, where I set up a specific test for NCF1, but they couldn't find the bloody mutation. Now we know it's there. It's a so-called gene conversion from the pseudogene. So basically a copy of the pseudogene jumped into the real gene locus and therefore destroyed the functional gene copy. And this phenomenon will happen more and more, but we may have been largely blind, especially in regions where homology is so high. Second case, we had one patient with clinically suspicious GLUT1 deficiency. GLUT1 is a sugar transporter in the brain, and we can even measure that enzymatically. So in liquor measurements, there is an enzyme defect for GLUT1 in that patient, but all the mutation testing done so far, Sanger sequencing, exome sequencing, all of that failed. Now we know why. Long reads discovered a translocation that just broke between the gene and one of its regulatory elements. So that still leads to the same non-functional gene copy, but it explains why we missed it by all the standard tests. One more study, and there we didn't do it just in our own center, but we involved many colleagues. In fact, several colleagues from your field. So Mattis and Rebecca, for example, were involved in these studies. And a big European consortium, Liedewey, was also involved in a project called SOLVE-RD, Solving Rare Diseases. And again, there we applied long-read genome sequencing. And as part of that study, we had 93 index patients that were largely put forward from 3 ERNs, European Reference Networks for Rare Diseases, including the one for rare neurological diseases, including HSP patients. And what we found in such a cohort, 12.9% of patients finally get a diagnosis, and an additional 4% have at least a very strong new candidate that requires further follow-up. And as Stefan said, we need to continue to publish, because that's one way of sharing this knowledge and new insights, even if you're not 100% certain about a candidate. I think you have a duty to publish it and share it in one way, and publishing is one way of sharing it globally. You just have to be careful how you report those cases. We found also reminders for ourselves in such a study, in particular large genes may be quite strongly enriched for structure variants, especially if they act in a loss of function mechanism. Then deletions or translocations that disrupt the gene or its regulatory landscape may be quite strongly enriched. If you work on Schinzel-Gideon syndrome, one disease dear to my heart, it's only caused by de novo point mutations that are gain of function. So for that disease, I dare to put my annual salary even, that nobody will ever find a structure variant. But for other diseases where the mechanism is different, I think they may be strongly enriched. And there's also something, and I have that on the next slide, we found 2 patients with muscular dystrophy from our colleagues from the neuromuscular network that both had inversions breaking the DMD gene. But in one of the 2 cases, and that's the lower one, the inversion not only broke the DMD gene, but another gene called NHS. And that's also a known disease gene that causes Nance-Horan syndrome. And in retrospect, the referring clinician, Alessandra and Rita from Italy, said, hey, in fact, our patient has a cataract. So in fact, this is a subtle, very much more heterogeneous phenotype where we probably have only one patient in the world who really presents with a genetic defect that disrupts these 2 genes. Probably there's never, ever a second patient like that. So keep that in mind whenever you work also with HSP patients that are very, very specific. Maybe they indeed have a merger of 2 phenotypes and therefore make it more difficult to understand the underlying basis. I just extracted from that same study, there were some candidates in there from the HSP families that were included. This was a case included by Rebecca Schüle. And what we found in this case, it's father and son that both carried a single exon deletion in REAP1. And to my understanding, there's experts in the room that know that gene. I think Stefan published several papers on that gene, so it's always difficult to talk about something you don't know. I think it's a classically more autosomal recessive gene, but there's at least one other report that exons in frame may be pathogenic also in dominant state. So we classified this as likely pathogenic. We don't have full evidence for everything, but it matches the report from the paper Rebecca confirmed. And again, I think there may be new mechanisms out there that we may have overlooked, and only by sequencing the gene or the entire genome more accurately, we will find some of the missed mutations. So just to sum up, for that part of the study, Just wanted to show everything in bold here are HSP patients. So we found another structure variant that impacts a gene called AMRC9. I don't even know whether it's impacting that gene itself or something else in the region, but that's yet another candidate. I think the structure variant itself and the inheritance of that is very strongly suggestive. So again, as in line with Stefan's remark, we need to sequence more, we need to publish more to really jointly gather the information out of those data. What I wanted to highlight is that some of these data we didn't just analyze ourselves. We organized what we now call hackathons, and in Solvd we called it solvathons. So the whole concept, and we were very privileged to host the long read and OGM solvathon in Nijmegen 1.5 years ago. And what you do is you bring these new data types, but you also invite disease experts, like your crowd, biological experts, bioinformaticians. You all lock them up in one room, give them 50 new data sets of undiagnosed patients, you give them a bit of Red Bull and some chocolate, and you hide the key for 48 hours, and everybody's only allowed to come out if there's at least 5 new diagnoses. And this magic really works. So if you've never done a solvathon, I propose your next meeting next year should include at least 20 data sets of some of the undiagnosed patients, and I'm sure you'll all solve them once you're together. And it's sometimes there's one clinician that stands up, but I remember this and that fact. What we had in Nijmegen is that we invited the mother of a patient, and she said, more recently we were concerned our patient not only has the developmental problem, but now he had recurrent infections over the last few months. By adding recurrent infections to the phenotypic spectrum, we found the genetic diagnosis. Another instance was that the clinician was in the room of a patient from Singapore. And then we found mutations in a gene that was described in the literature with very strange-looking feet. But the clinician had to admit, I never looked at the feet of the patient, but she had the WhatsApp number of the mother. So one WhatsApp in the solvathon itself, and the mother replied within 5 minutes, and the picture of the feet showed up on the screen, and it was identical, nearly identical to the feet from the paper. That's how you together, in a hackathon or solvathon, solve cases. Bringing all the joint brain power together in one room is still a magical thing. All right, what I tried to show you so far is that we did several clinical utility studies for long reads. Known mutations, that's a prerequisite. If technology is not up to speed there, we really can't use it. And then several clinical utility studies, summing out how much extra we find. And those, I should say, are rather conservative estimates with the knowledge we have today. I'm still convinced that there's much more in the data. For example, we only start looking at repeat expansions that are new at this very stage, and I think there's much more promise even in the data as they are now. So hopefully this convinces more and more to move to long-read genome sequencing as a first-year test. To convince our own management, we had to do one more trick. And basically, as I said earlier, we had to run a prospective study or a prospectively designed study. And that's what we did. So we basically mimicked 1 full year of germline diagnostics, where on the left, we just do what our diagnostic does, usually exome sequencing, since then genome sequencing, but also sometimes a mix of 10 different tests, versus just a long-read genome sequencing on the right. We selected retrospective samples, but in a prospective design. That means we went to the full year of 2022, calculated how many diagnoses we made, and selected 1,000 samples from that represented our yearly diagnostic referrals. So those are diagnostic categories or diseases that are referred to us for germline testing. It represented our average diagnostic yields across all those diseases, and it represented the use of different workflows. And then we installed a team of 3 postdocs, 2 of my postdocs, 1 postdoc from Lysenko, and said, okay, imagine you're a diagnostic expert now, make as many diagnoses as you can. If you fail, you're fired. No, we didn't say it. You don't do that. We're quite nice to our people. Just for those interested, these 1,000 samples represent close to 300 different clinical indications. It's 748 singleton index patients, and in some cases trios. And on the lower right, you see the tests that are originally requested in 2022 across all those samples. And then the most important table from such a study is shown on this slide. What is the comparison? How good or bad did our postdocs do by analyzing a single test compared to standard of care? And the great news is we found 96% concordance. So both tests were either accurate in giving a diagnosis, a likely diagnosis, or not providing a diagnosis at all. Some people were very disappointed by the lower right corner. Everybody expected you now should give a diagnosis in almost everyone. In fact, we're still happy that we have negative cases that remain negative. Imagine all of those cases we find 10 variants of unknown significance. That would completely overburden the system, and all our diagnostic experts would leave tomorrow. So in a way, the new data does not introduce a lot of new biases or variant lists that are unmanageable. In fact, this shows it's all feasible right today. In the 1,000 samples, we also missed things. That's always happening if you introduce something new. I'm surprised by seeing that we only missed 2 cases. One with a mosaic mutation that was found just by chance in deep exome sequencing, and one other case where the coverage was very low in the long reads. But we also upgraded variants and cases in this way. In total, 28 cases had either only a likely or no diagnosis before, but have a firm diagnosis or were upgraded to likely diagnosis compared to the standard of care tests. And the biggest chunk in there are cases where we upgraded 2 variants to certain diagnosis by phasing. So that means we have a sample where the genome is sequenced, we find 2 variants in a recessive gene, we think it fits the phenotype extremely well, but we ask to get the parental DNAs or other family DNAs to be sure that they inherited on both alleles, so that both alleles are destructed. And I always thought in Nijmegen we have exceptional contact to our clinicians, the clinicians have great contact to the families, and we get family DNAs within weeks. Even in Nijmegen, that does not happen. Sometimes that delays the entire process by months or years. So long reads don't desperately need the maternal and the paternal DNA. From the long reads itself, we can distinguish that one variant happens on the maternal and the other on the paternal level. We call that phasing. So that, in fact, that simple trick already will speed up diagnostics quite dramatically. And imagine even families where father, for whatever reason, has moved on, is not in the picture anymore. Also, all those families are much more amenable to more firm diagnostics from now onwards. Also, the opposite happened. We had 2 cases where we found 2 hits by the standard of care testing. We think it may well be the gene, but in fact, they're happening on the same chromosome. So that gene should never be reported. So there also the long reads can help dramatically. What else is in the long read data? I briefly mentioned that we now can find methylation. Just by chance, we have 3 cases that had pathogenic mutations in genes, or on the right-hand side for Down syndrome, for a syndrome where an episignature had been published in the past. We can now look up the methylation status and really have this more molecular phenotype on top, and we've now replicated that in larger series and even described one full episignature based on long-read data now. So that was all these retrospective samples from 2022, but then we got criticized, hey, by 2024, your diagnostics had also improved and you did genome sequencing instead of exome sequencing. We now, or in fact, Lee Senker and her team went through all the data from 2024 and modeled what would have happened if we would only have done long-read genome sequencing now. And the pie charts don't look dramatically different. I mean, that in a way, again, is good. We're not changing everything, but we're improving a little bit, and that is an annual increase of 2.5% of firm diagnosis. As we see 15,000 index patients in Nijmegen, that's close to 400 patients or families that get a better diagnosis than before. But of course, we can dive into that, and in fact, this 2.5% gain is in fact 1% loss and 3.5% gain in total. And of course, I want to zoom into a lot of positive things, but I think it's also important to stress where we're not good at. Again, those are not surprised. Oh, that was animated before, sorry for that. We still lose repeat expansions. Just by chance in 2024, we did a lot of FGF14 testing, so Stefan is all to blame, that we had to run through our diagnostics of a lot of missed ataxia patients. So we for now anticipate that we have a very hard time detecting all of them automatically here. So that is 1% loss, largely repeat expansions, mosaic mutations, mitochondrial heteroplasmy. Those are the 3 big issues that we still face. That would, on an annual basis, give us 1% loss, but a total of 3.5% gain by the better test. So if you're interested, the preprint of that study is out, but potentially even more excitingly, we moved already live. So our management was convinced by this data that since October, this is the only test we offer for germline testing for 2 large disease categories. So all patients in Nijmegen that come with severe neurodevelopmental disease and with genetic eye diseases, aka blindness, now only undergo long-read genomes, and we stopped all the other test modalities for those tests. Ambition for year 1 is to offer that for 5,000 samples, and this is just the start. We hope that we really push that as a generic first-year test across all 25,000 samples we see a year, because that will really consolidate tests, automatically speed up, and ideally even make more diagnoses, as some of the research studies now imply. So in a way, for the first time, at least conceptually, have a genome read to near completion, near perfectly. We're still not perfect, but it's near perfect, better than ever before. And as such, I think we have a duty to really bring that to the front line of testing, and in Nijmegen, that would be 25,000 tests a year. Replacing all the other tests, becoming faster in turnaround, and as such, look at genomes much more completely by all variant types, as I alluded to throughout the talk, which itself will gather some extra yield and potentially, and that's of course what we are still driven by, new discoveries. Conceptually, I think there's two big changes coming to our field in general. I think we should move away from variants in isolation. So when we discuss gene curation, I think it's really intriguing to think not about variants in isolation, but about complete alleles and haplotypes. Probably some of the non-penetrants and the bits that we don't understand may at least in part lay in understanding alleles to bigger completion. And what I didn't realize, maybe such a much better genome test is also of great benefit for a negative result. If you have to write a negative report now by exome sequencing, you always have your safe harbor statements in there, what else may have gone wrong. For the first time, we now have certainty that we see both alleles, and we with certainty can tell a doctor, it's really no mutation in that one gene or in that gene panel even. And that, again, conceptually will be really different. If I still have 1 minute, I'll finalize. I don't know, I couldn't see them. Oh, you should, huh? I'll just close with one family case here, why this is important and why it matters. And I'm just a biologist. I don't have the privilege to usually work with families and patients directly, but here I had the privilege to really spend a full day with one very remarkable Dutch family. And they are so remarkable because of what they witnessed in the past. They witnessed full tragedy by losing their 2 daughters due to a terrible brain tumor, a very specific form, ATRT. After the birth of daughter 1, it was already clear by histology of the tumor that is a tumor driven by biallelic mutations in SMARCB1. So we know the mutation, the responsible gene, and in 1/3 of cases it's even suspected that there's a germline predisposing factor for SMARCB1. So all genetic testing failed to find a germline mutation in SMARCB1 in that family. Then they got a second child, and she also had a brain tumor, again, diagnosed right after birth, and unfortunately it was already metastasized, so the family took the very difficult decision not to even treat the second daughter, and therefore she passed away 2 weeks after birth. From that moment, it was clear there must be a mutation, but again, even genome sequencing failed to find the bloody mutation. Luckily, they saved some cells of the first daughter, sent it to us, and we applied, in fact, a mix of 2 long-read technologies, optical genome mapping and the long-read sequencing I mentioned, and we finally found the mutation. It's a so-called retrotransposon insertion in the intron of the gene. So one of the most difficult mutations you can imagine. We now know that is the germline mutation that disrupts transcription for that gene, and that's something we published. But much, much more important than the publication, because we always criticize what can you do for families once you give a diagnosis. You only do a diagnosis, you can't treat. Surely here we can't treat. Both patients already passed away, but still it was so important for the family. First of all, having clarity, taking away uncertainty, what the root cause is, was very, very important, and both parents explained that. But in this case, my colleagues in diagnostics were even able to custom build a prenatal assay. Only because of that, the mother dared to get pregnant again at the age of 40, and luckily, in pregnancy week 10, we could offer a prenatal test and show that the new baby did not carry the mutation. So since 3 years now, they have a healthy baby boy. And I had the true privilege to spend a full day with that family, and we even recorded a little video. If you haven't cried today, this shows why we have to do our work and why we have to get up every morning to work even a little bit harder. I'll finish here. There's many people to thank in Nijmegen, our colleagues in Maastricht, many collaborators in Solvardy. But of course, I also thank you for the kind invitation, and I'm very happy to engage in questions now or later. Thank you very much.
Wow, super inspiring. I suggest, because we're sort of late, we do a question session in the very end. We have 3 very similar, or not similar talks, but same topic. So the next speaker will be Ingo Kort. Great pleasure to introduce him. He's from my alma mater, the Hereditäre Aachen University. And Ingo is the director of the Center for Human Genetics and Genomic Medicine there. And he will very well complement this talk because he will focus on the other technology, the Oxford nanopore.
Yeah, thank you very much. Will you change the presentation or should I? Do it myself. Okay. I can. Right. Right. Where's the pointer?
Nothing works here.
Okay, I leave Crystal with that. So,
That's not.
And I can already start. So the pointer is somehow gone. We don't know, but I'm sure we will fix this in a second. There you are. All right.
Which one is it?
It's
This one?
this one.
Yeah.
Yeah, perfect. So, ladies first. Yeah, I'm very glad to be here, and thanks for inviting me. Also, the Walik family. It's really great to see you here, and it's difficult to speak after Alex, of course. This is yeah, but the good side is that he already convinced all of you that long read sequencing is really the way to go and will bring us further. And he also showed nicely that we are different. We have a lot of variants in the genome, 4 to 5 million, so that's the rough estimate. And all of us here in the room are looking for one single genetic variant, right? And which one is the right variant? If you look on the entire genome, that's always the question, and this is maybe one of the central questions in human genetics. And also the downside is that if you look at our understanding on the human genome, it's still a bit disappointing. You see, we know a little bit. These are here the 3 windows which are enlightened, but that means we have a lot to do, and maybe long-read sequencing is also a way to move forward here. And again, these are the 2 technologies. Alex was talking about PacBio on the right-hand side, so it's still working with fluorescently labeled dyes, and the principle of nanopore sequencing is fundamentally different. So it uses biological pores, which are embedded on flow cells, and so whenever a DNA strand is pulled through these biological protein pores, you have changes in the currents, and they correspond to the nucleotides which enter the pore. And that means you can also detect DNA signals, but you can also see methylation changes because there are slight differences in the DNA. So that also means that on the fly you have a pattern of the methylation, which you can also record. And so I would like to start with 2 examples from what we know. So the protein coding regions, and these examples are both a bit linked to HSP, but the phenotypes are also more complex. So the first one is on the so-called trick-related disorders, and this is a large family of diseases, and they are due to mutations in a complex which encodes a chaperonin. And this complex is composed out of 8 different subunits, and they form barrel-like structures. And whenever a protein enters this barrel, it's getting folded in the inside. It's because of the environment within this barrel, right? And so it prevents also aggregation, and this is also something that we observed in some HSP subtypes. And so the phenotype which is caused by mutations in these genes is rather complex. It can lead to brain malformations. However, some of the patients that we know were also referred with kind of a complex HSP disorder. And I think, again, here we have a broad phenotypic spectrum. And you see that besides also developmental delays, we have a lot of patients with pyramidal signs and also signs of ataxia. And also in these cases, sometimes the long reads help us to interpret the data. Here is one case which I would like to show you. So normally the disease is inherited in a dominant fashion. That means most of the patients have de novo mutations. However, also in this case, we found one very clear mutation on one allele, but on the other allele, there was another variant, a missense variant. And in this case, we were not sure whether, first of all, this is kind of a modifier or also causes kind of a recessive mode of inheritance here. However, we didn't have parental samples, but could, as Alex also demonstrated, phase these two mutations. Could say, okay, one is on the maternal allele and the other one is on the paternal gene copy. So that means from the long reads, we have now a global picture of what this patient has. So this is sometimes really helpful, especially in cases where you don't have parental samples. So the second example is the so-called IST1 related disorder, or at least we think that this is a new disorder. So this is also called for additional patients because so far we only have one single case, and whenever you might have mutations in that gene, we would be definitely interested to team up. And it started here with also a complex HSP-like disorder, also accompanied by hypomyelinating leukodystrophy. And this patient is now, in the meantime, it's a progressive disorder. He is severely disabled. And we identified mutations by the long-read sequencing technologies, but also in this case, retrospectively, it would also have been possible to solve the case by short reads. So actually, the patient has two variants in the IST1 gene, two clear loss of function alleles on the maternal and the paternal copies, and you can also see in fibroblasts that the IST1 protein is absent, so clearly this is a loss of function. This is an MRI picture showing the hypomyelinating leukodystrophy in this case, and IST1 is a member of the endosomal sorting complex, and it's also in the complex together with spastin and other genes which are linked to HSPs, so I think this is also demonstrating that this is a broad spectrum. So whenever you might have mutations, I'm really interested. And yeah, now coming back to the dark sides of this house, of this building. And the downside is we're still not good in interpreting the non-protein coding variants. However, we are getting better. Christelle is the best example. She could solve a lot of cases with mutations in the RNU genes recently, and this shows that there's also a lot to discover in the non-protein coding regions of the genomes, and long-read sequencing can of course help here. Alex also showed nicely that episignatures might also be a way to solve additional cases. What is an episignature? It's a unique DNA methylation profile, which can also serve as a biomarker. So it's mostly something that you find in when a gene is mutated that is somehow involved in methylation processes, and then on the fly you record by the long-read sequencing also the entire methylation data. And so you can also look beside the mutation or the variant that to show whether this could also point to a disease. So in this example, we had a patient with a global developmental delay and a microcephaly, and identified a missense variant in the gene called KDM6A. So missense is sometimes tricky, can be pathogenic, can be benign, and so also the bioinformatic predictions were not clear here. But the episignature gave additional hints, because you can see here the comparison between controls on the upper left side and the patients that very clearly cluster with our proband, and that means in this case we could be more convinced that this variant is really causing disease, because it clusters to, in this case, kabuki patients which harbor mutations in the KDM6A gene. So it can be helpful to also have the long read and also the methylation data from one single test, and this was also where we have to move to have less tests, but one test that shows you all the things that you need to know. And that means if you look at the genome-wide methylation data, you can also see changes in regions of the genome, every region, and I think this is also currently studied in HSP samples as well in patients, whether there are also specific methylation patterns, episignatures. I think this is currently really not well studied. And again, we also observe in many of the neurogenetic disorders that repeat expansions underlie the phenotype. And what we know from more recent literature is that not only the length of this repeat expansion is important, but also the composition, the architecture of the repeats. And this is something that you can more easily resolve by long-read sequencing technologies. And this is an example from an RFC1 patient, and you can see here that these motifs can change within a repeat, and this can also determine a bit the phenotypic variance that can also determine whether at all this repeat expansion is pathogenic or not. So with long-read sequencing you can also resolve these issues, and I think we can also learn a lot about genotype-phenotype correlations here. And of course there are additional machine learning approaches which help us. This is maybe the second shift in human genetics beside the long-read sequencing, that the variant interpretation gets better and will be better in several years. Also, we heard about EVO2 and these models which will help us to really address the unsolved cases. So yeah, long-read sequencing is not only suitable for humans, and this is an example from the neighboring Gelsenkirchen Zoo, and we got a blood sample from the polar bear Antonia. She recently died after a long life, and it's a dwarf polar bear, and currently it's unclear why this ice bear is so small. And we now have the long-read data available of the ice bear. The downside is that an ice bear genome is even more difficult to understand than human genomes. Also simply because we don't have too many samples from ice bears as comparison. So in humans this is really good that now we're moving towards large genomic analysis and know about genetic variation. So maybe this is also, Alex, something for your next Solfathon, and you will only get out of the room if you can solve this case with your team. Maybe we can discuss this. Yeah, so I think with long reads we're moving forwards to an era where we really can advance the understanding of genomes, genomic variation, and still it's a long journey towards the understanding, but we keep going. And I think it's really important to stress that finding the genetic variant which is causative is the first step towards targeted therapies, and this is why we do this. And yeah, I'm happy to discuss this with you. Thanks for listening, and thanks also to my team in Aachen. Yeah.
Thank you, Ingo. I think we will do a question after. All right, the last speaker in this session is Liedewee van der Wandel. Liedewee did her PhD training at University of Antwerp and is currently in Miami. And she's a very talented postdoc and will tell us her experience on long-read analysis.
Thank you. So, good afternoon, everybody. As Stefan said, I'm currently a postdoc in his lab, and I'm hoping third time's the charm today. It's the third time about long-read sequencing. And I, kind of in my day-to-day, I just try to solve patients, and currently I'm trying to focus on how to do that with the long reads. So it's been touched upon already today. There is a very persistent diagnostic gap in HSP, and there are, of course, several hypotheses how we can still break through this. I think there are multiple genes that show significant phenotypical heterogeneity, and then non-coding variants or maybe oligogenic inheritance. But what the long reads will specifically be good for is the structural variation or the tandem repeats. So in Miami, we have the Genesis platform where we analyze genetic data, and where we also are making a lot of new long reads right now. So we have about 200 patient-derived long reads to analyze, and about a quarter of those are actually from HSP patients. We try to all kind of do it together because we can learn from analyzing CMT patients or ataxia patients and try and apply that to the HSPs as well. So Professor Heuschen and Professor Kurt already kind of explained this. Why do we think the long reads will be so much better? So with the short reads, if you want to detect, for example, structural variants, you're kind of relying on bioinformatic tricks with read depth or maybe split reads. Whereas with the long reads, we're hoping that you can really capture the structural variant in one read, have a full readout, and be able to characterize it. For the tandem repeats, on the other hand, once your tandem repeat becomes larger than your read length, which used to be 150 base pairs, you can't really see the accurate length, but also you can't see the sequence composition, which is something that we're hoping to resolve with those long reads. So talking about the structural variants first, Professor Heuschen also mentioned this. So previously with the short reads, we were only able to detect about like 4,000 structural variants per individual. This was from the 1,000 Genomes Project. So when we try and do this with long reads, I think initially everybody tried to look at, okay, let's just only do healthy persons first and kind of get a feel of like how many structural variants actually should there be. So we see about a 5-fold increase. This is data from the Olivus project, for which Stefan is also one of the leaders in there. So these are the phase 1 data, so the data is in different phases. For now, this is only African American data because that's what the NIH decided on focusing first. So only in 1,000 long read patients, we see that we have these 25,000 structural variants per individual. We also see a very stark increase in these mobile elements that we're able to detect now, so the ALU elements or the retrotransposons, of which Professor Heuschen also showed an example, which we weren't really able to properly place before. A kind of competing consortium, let's say, is 1,000 Genomes, which is ONT-based and not hi-fi based like all of us. And they kind of see the same number of those 25,000, except that they already have data about the different ancestries. And as you would expect, the African ancestry has a higher amount of diversity also in their structural variants. But the number of like 25,000 is kind of what's valid. So what do we do now? Like now we have a kind of an idea of like, okay, all of these structural variants are in healthy people. So as we would use NOMAD, you're trying to look in patients. So in the Genesis platform, we had a patient, actually 2 high ONT data from the mother and the son that were shared by collaborators, the Ravenscroft lab in Australia. And there they identified the in-frame deletion that actually deleted 2 exons in the SBTN1 gene, which is known to have an autosomal dominant inheritance for HSP. So that was a really interesting solved case, and I wanted to highlight this because I think, as we talked about earlier, This is one of those cases where if we publish this, it can actually really confirm a very rare form of HSP. So SPTN1 can cause a lot of different diseases, including peripheral neuropathy, epilepsy, etc. But only one mutation has so far been identified with HSP, but it was highly recurrent, the arginine 19-tryptophan. No other variants were found so far, but so this patient would actually confirm the pathomechanism of the arginine 19-tryptophan variant. So SPTN1 encodes alpha-2 spectrin, which as a protein forms a heterotetramer. So first it forms a dimer and then a heterotetramer. And the arginine 19-tryptophan mutation is located at this tetramerization binding site. And it's actually those exact same spectrin repeats that would be deleted in this patient, kind of confirming the pathomechanism there. Another example, it's a CMT patient, not HSP, but it's very similar. It's a patient from collaborators in Iowa from the Shy Lab. And it's kind of the same story, had HB, is also a very rare cause of neuropathy, but this patient might confirm that again. And it's also a sporadic patient, which Professor Kurth highlighted earlier, that because with the long reads, you can see those haplotyping, and this is what it looks like in Genesis, so you can really clearly see the two different colors with the missense variant being located on one allele and the deletion on the other allele. So we do not need the parent samples, which is often also a case in late onset patients. So I'm hoping that this would be kind of like everybody's first guess, like what to look for. We have SVs that affect exons in known disease genes. I think the second step would be, of course, to do that in not known disease genes, but as touched upon earlier, it becomes harder and harder once we look at intronic SVs, or maybe SVs that are affecting enhancer regions, promoter regions, and then lastly, those intergenic SVs. For that, I think the control databases that I showed will be very, very important. And of course, as touched upon earlier as well, those prediction efforts across the genome, of which Brain Magnet from the Barakat lab that was recently published, I think is an awesome study, as well as Alpha Genome, of course. And then one other thing I think that is very interesting to follow up on is maybe to think about what we're actually doing with the data. Because at this moment in time, maybe the long reads, when we're aligning them to the reference genome, you're aligning them to a reference genome that was built with short read technology, which maybe doesn't exactly make sense. So we have these de novo assembly methods where instead of aligning your long read to the reference genome, you just kind of let it align by itself and see what it does. And it's starting to show that that's also a really interesting way to look at the structural variants. And then maybe possibly, once your structural variant actually becomes too large, once it passes that threshold of the long read length again, you need other technologies such as optical genome mapping. So switching gears a little bit right now to the tandem repeats, I hope everybody has already read this excellent review by Crystal, which I love. So the kind of general things to keep in mind for the tandem repeats are, of course, that they anticipate, so they get worse with the generations. A lot of them have somatic instability, which was also recently very eloquently shown in Huntington disease. And then they're also known to have very significant founder effects, so we really should take care of the ancestry data when we look at control data. And then previously, of course, without proper linkage, it's really challenging to identify them in short reads. I think in part that's also because how we're classically trained to look at them is you just think of them as like, okay, we have a tandem repeat, and once it crosses this kind of length threshold, that's when it gets pathogenic. I think that is true about a lot of the repeats, such as the known coding CAG ones, but what we're learning from the most recent ones is that sometimes you have this motif change that's necessary, and you really should be looking at the different compositions of the alleles. So for that, my colleagues at the University of Miami, Matt and Isaac, they devised this kind of algorithm to calculate what is called the longest pure segment. So it's literally what is your longest pure stretch of your repeat in your genome, whereas in the top allele, maybe it might still be the green composition, in the other one it might be the pink that's taking over, and it's really that one that we should be taking into account when we're looking at our patient alleles. So they did that for the entire All of Us phase 1 data set that I showed earlier, so the 1,000 African American data sets, and they analyzed the full Adado catalog, which is kind of telling you where in the genome all of these tannin repeats are located. So those were 1.7 million loci. And they just analyzed these LPS patterns, and they were thinking, okay, can we not only use this to look at patient data, but what if we can use this to look at sort of pathogenic hallmarks, like how we use a nomad conservation score to identify a new gene. And they were actually very successful, I would say, in pulling out these pathogenic alleles, loci you see in red. So if you look at every single 5' UTR CCG repeat expansion, which is what you see here, or locus I should say, sorry. If you look at the pathogenic ones, you actually see that those are the most variable ones just in the control population. So thinking about this is kind of directly is opposite of what we do with a nomad, where you want a gene to be constrained. With the tannin repeats, it's actually you want it to be as variable as possible in the control population, and this might underlie some kind of propensity for it to become large in your patient as well. They're currently extending this because, as I said, those ancestries are important. So they're currently working on 12,000 data sets and also a bigger catalog of TRs to fully categorize this. Why did they look at this kind of classification? Okay, what if we look at all the 5' UTR CCGs in the genome? It's because, and this is again from the review from Crystal, if you look at the tannin repeats, it's often that it's kind of like their motif that dictates the phenotype-genotype correlation, and not necessarily only the gene that they hit. A very eloquent example of this, I think, are the OPDMs. So they all have 5' UTR CCG expansions in a lot of different genes. So initially in 2019, OPDM1 was identified, and now we have 6 of them, and only OPDM5 was previously identified in a European ancestry. OPDM is a myopathy, so it's oculopharyngeal distal myopathy. It's also a rare disease, which is why I'm trying to show it today. And I'm actually happy I included this example because it also shows that Professor Horschen wasn't lying about the solvathons. They were really awesome. I was there. It was really fun. And we had this family A, that was actually solved right there in Nijmegen. It was the first time I was looking at the data, and so Solvardi, through Solvardi, we were able to generate data for the 4 of them. And using actually a structural variant analysis, doesn't really matter how we found it at this point. We found another CCG expansion in the 5' UTR. So at that point, I know, okay, 5' UTR expansions, CCG, the phenotype matches, this is good. We just need to find other families. And so the Cortese lab in London, they actually, independently from us, also found it through short read data, through association studies, and they had 2 more families kind of confirming this. So what we then did was, okay, let's look at from what's the pathogenic length, comparing it to the control data, as I showed earlier. And actually, most of the patients in the control population have a CCG-CTG motif and not a pure CCG1. So actually not a lot of people have that, so we are able to kind of make this benign versus pathogenic range. And then additionally, I couldn't include the data here because I'm kind of short on time, but we also found that one of the parents was non-penetrant, but that was because his pathogenic repeat actually got fully methylated and therefore silenced. So just again as a reminder to include this when you're looking at it. So because most of the people in the control population actually have a CCG-CTG motif, this particular locus wasn't included in the previous analysis of all of the 5' UTR CCGs. So we repeated it, and I think everybody can agree that this is lovely, that TBC1D7 is the most variable one in the genome, kind of confirming what we saw earlier. So I'm hoping that this kind of showed you that methylation is really important to take into account, not only when you look at segregation in your pedigree, but also when we're comparing it to the control data, because it might be that there are actually a lot of people in the control population that have a larger tannin repeat, but that actually gets methylated. So there might be like this kind of intermediate range where the repeat is still pathogenic. Tying to that, I think we now know that the pathomechanisms might be motif-specific, with, for example, the CCGs having this gain of function window, and then the GAAs like RFC1, like FGF14 is loss of function tied. So we really need to think about this when we identify a repeat. What's the motif? Do we think it's gain of function, loss of function? And does it actually matter what gene it's operating in? And then additionally, some of them might have both, like for example, FMR1, which is also a CCG that has a gain of function window and also a loss of function window. I think this also shows that there are actually a lot of different loci in your genome that can lead to the exact same expansion and therefore the exact same gain of function mechanism. And it might become even harder to make these kind of links between genotype, phenotype when we're not sharing enough patient data. So I think a lot of these will be even more rare, unfortunately. And then lastly, the population context really matters, so we should really take into account those ancestries. So with that, I'd like to thank everybody at my current lab at the University of Miami, the previous lab I was in Antwerp, which also allowed me to take part in SolveRD, and then of course all of our collaborators. Thank you.
Wonderful, wonderful. So it's a very technical session for this kind of meeting, but I hope you got the sense that in genetics, everything is we kind of start fresh. That's how I see it. So we a little bit over time, let's just give us a couple minutes to ask some questions. I have one for you, Alex, right there. And now I know you like the future. So this is all amazing. You said this is near perfect or so, he said. Is there beyond that even, you think?
I mean, we like that term a bit, just conceptually, to really trigger the field, to really think in a way that we haven't done before. And I'll just add one example why we think that is important, and that is you brought up the AI tools that can help us in interpretation. For the first time, I think we can look at variants not in isolation, but in the context of the rest of the genome. So for the first time, you could potentially, and ACMG already has it in their guidelines, in a Bayesian way, look at variation. So you could look at a missense variant, let's say in your favorite HSP gene in an SPG patient. But for the first time, you would have evidence that there's nothing else in the genome that is more convincing than that one private missense, and that in theory can, if our tools are good enough, upgrade that missense. So the absence of other events is something very powerful that we were never able to utilize. And only if you read your genome near perfectly, we could do that. So that's for now a conceptual thing. If you ask Evan Eichler, of course we all need T2T quality, ideally combination of 2 or 3 technologies to finally close all the remaining gaps. I mean, he's right, but I'm not sure how much extra that will yield, and probably only time will tell. For now, I'm so happy with the current both technologies, I should say. I mean, there's very similar data, so it's good that both technologies are around. And for now, we can just push them into clinic, and I just do this single test instead of doing 10-fold the amount of money and do 10-fold the amount of patients less. So we're already happy by moving with this in the clinic. It's so much better than anything we did that it's a major leap forward.
It's just the cost factor, right? Okay, more questions.
First of all, thank you for the fascinating talks and really great future. But if I go 2 years back, I would have heard that the genome is it, and that we get all the information about the intronic variants, and they probably need a transcriptome to interpret it. I don't hear anything like that anymore. How do you solve that?
Who wants to talk about it?
Bad luck that I got the microphone. Yeah, no, you're absolutely right. I mean, this is the way to move forward. Transcriptomes can solve additional cases. I think we're also moving towards proteomes, which might also do a very good job here. Well, it's always the argument that, well, we're talking about HSP, why do you look at the transcriptome in the blood? But still, I think this is a good measure at all. Yeah, it's also some kind of an indication. So I think it's, in the future, it's a combination of the different things which come together. I think you're coming back to your question, why is it not done in routine? It's still because it's costly, and in the routine clinics it's not really possible to do all this. I mean, you could, of course, transcriptome is not very expensive any longer. However, still, at least from what we do, it's challenging to really say. So the changes that you find are not always so convincing. So my guess would rather that we go for a data integration via all the new machine learning tools which can implement such things, but from my point of view, the genome is still a very, very good ground to find the right variant.
I have one question for Leaderway, so to equal it out here. So if you had a wish, or what do you think is the biggest, in your daily work, finding new genes, even if there's new data, what's the biggest impediment?
What's the biggest? I mean, there's so many.
Distractions in Miami, the beach.
No, I think talking about that, I think it would be awesome to move towards this kind of integrated data set indeed, where you have transcriptome. It would be lovely if it's a long read transcriptome at once from induced neurons from a patient to complement long read. I think that would be the most ideal way to go about it. But I think at this moment in time, for me, it's really trying to matchmake the different patients. I think having only 200 long reads, I mean, it's a very privileged position to be in, but at the same time, I'm solving one patient and then you're there at N of 1. So I think at this moment in time, it's still there to try to make the links, which would become easier with maybe artificial intelligence to do across big data sets.
Right, fair enough.
I have a question and a comment about artificial intelligence and what these machine learning methods can do. So I'm not a specialist, but as far as I understand them, they just work, they are trained. And you train them with what you know. And what we want to actually find is probably what we don't know. And I'm not sure how good these methods would be to find, I'm not sure that these methods are there yet to actually, so talking about repeat expansion, I think it's a very good example where they actually don't work that well. Especially for the new motifs. They can't predict if new motifs would be pathogenic or not. So it's both like a comment and a question. Do you think it will get better? Or like with Maverick, do you plan to address that gap specifically? Or what is the next step?
Let's just say it will get better. We'll see. I mean, all of us, it's going to be crazy, I think. For better or worse, in 5 years, we'll talk again. Okay, next is lunch. Very important.
Yeah, that's right. So we have a lunch break till 2 o'clock, but before we would like to take a photo, and as the sun is shining outside. So everybody, Nicole Jeschonik will show you where the way is, okay? And we restart at 2 o'clock after the lunch.
modified spastic paraplegia rating scale intended for application in children age 5 and older.
Items 2 and 3 assess gait quality
Hello, hello. Thanks for coming back in time from the lunch break. I hope you had some really nice lunch and some really good discussions with old friends, or you met new people. My name is Beate Winner. I'm from Erlangen, Germany, and I will guide you through the next session. And now, since we heard about the whole spectrum of all HSPs, this session will focus on only SPG 11 and SPG 8. And Elena Rogali will be the first speaker. She has been working on SPG 7 for all the time I can remember. So I'm really looking forward to her talk.
Thank you very much. So I will get started, and we are all here today to, of course, to think about hereditary spastic paraplegia, and especially how we can think of improving the symptoms of the patient and maybe one day treat this disease. Now we all know the main symptoms of this disease is the degeneration of the long cortical spinal neurons. These are extremely long neurons with extremely long axons, and I think it's very important, therefore, that we understand how this distal part of the axons that are the first one to degenerate are maintained. And there are a lot of questions in the field, basic questions that are still largely not completely answered. So how, for example, do we supply this distal region of axons with functional organelles? For example, mitochondria or endoplasmic reticulum that you know plays a very important role in the disease. So how is this supply of organelles regulated? How is the quality of these organelles that are very far away from the soma ensured? And how is the biogenesis of the protein that, of course, don't have such a long half-life matched with this long commuting and half-life of axonal organelles? And also, how are the metabolic function specific for this organelle when they are in the distal part? Because I think we can probably all agree that probably the axons is a very special compartment that is doing, where organelles might be doing different things than in the rest of the neurons or in the soma or dendrites. So today I'm going to talk about mitochondria, which of course, as you know, are very important organelles. They're very much enriched in neurons and in axons. They are, of course, famous because they produce ATP, but in the neurons, they are also extremely important for calcium buffering, and they are also host many of the metabolic reactions in our cells, and of course also in the axons. And we know that mitochondrial dysfunction is linked to HSP, and indeed the very first gene for hereditary spastic paraplegia, the very first autosomal recessive gene was cloned is SPG7, which is, I'm going to talk about it today, and is encoding for a mitochondrial protein. So this is the beautiful mitochondrial M-AAA protease that you see here, model with alpha fold. And you see, is this a big proteolytic complex, is an hexamer, and can be composed even of a homoligomeric subunit of a protein that is called IgG3L2, or of heteroligomeric subunits of IgG3L2 and SPG7, or also how we call it paraplegin. This part here you would have, I don't know whether you see you see it? You would have the membrane, and then you have an ATPase domain, and below the proteolytic domain. What is interesting is that recessive mutation in SPG7 give rise to hereditary spastic paraplegia, very often with an ataxic component, so it's often a spastic ataxia disease, but mutation, and almost always, with some exception, heterozygous mutation, so dominant mutation in FG3L2 give rise also to neurological diseases, mainly characterized by ataxia, but also by a complex phenotype like progressive external ophthalmoplegia or optic atrophy. So what do we know about this complex? We know that it is located in the inner mitochondrial membrane. It is important for the quality control of protein in the inner mitochondrial membrane. That means that it degrades or mediates the turnover of, for example, subunit of the respiratory complexes that are in the membrane, or proteins that are regulator of other important complexes. For example, this protein here, EMRE, is a regulator of the assembly of the calcium uniportin, MCU, and therefore this regulates the calcium homeostasis. But it could also proteolytically process proteins. And this proteolytically processing, for example, for a small component of the mitoribosome, MRPL32, allows this protein to be folded and therefore then to assemble, and therefore allows mitochondrial translation. So you see already here that this complex, if it doesn't work, you have a lot of different pleiotropic function. So in the years, we started actually this project. I started this immediately after the gene was cloned. So the gene was cloned in 1998. And then we started to generate the very first mouse model for SPG7. That was also, I think, the very first mouse model for HSP. And we were lucky because this mouse model is a late onset phenotype, but it reproduced the disease phenotype. And now we have a new model that we call the enhanced mouse model. This model has mutation not only in SPG7, but also in IFG3L1, which is a third orthologs of this complex, of these subunits of the M-AAA protease, similar to FG3L2, that, however, is expressed in mouse, but not in humans. So by mutating both of them, we have a genotype in these mice that much more resemble the human genetic, let's say, status. And these mice are quite nice because they have a phenotype that is a bit more accelerated. So already at 1 month, we see that they tend to be smaller, especially the males. They have a prominent phenotype that we can measure motor phenotype already at 4 months. We have a spinal cord, and also what is important, an ataxic or cerebellar phenotype, similar to the human patients. And I show you a few images just to give you an idea. So you see that these mice are smaller. They have a pronounced kyphosis. This is the growth defect that they show. And this is an example of the motor impairment that so takes for them more time to cross a small and thin beam. And this phenotype seems to be progressive with time. But what is most important is that they lose axons very slowly. But before these axons degenerate, we can start to see what is happening in them. And what we see is the appearance of these abnormal mitochondria that become large, swollen, and ultimately really accumulate together with a lot of other material into these axons. And these axons then degenerate. So they have also proteomic changes. These are examples from the spinal cord. Something is very similar that we see in the cerebellum. So for the many components of the mitoribosome are decreased. So you see that this is probably linked to this defect in processing of this protein, and therefore defect in assembly a functional mitoribosome. We see some changes at the level of the proteome. They're very minor, but we have to consider that this is a very heterogeneous tissue, the spinal cord, with a lot of normal axons and axons that are abnormal. But what was interesting is that we see a lot of changes in solute carrier in the mitochondria, so indicating that there might be a lot of metabolic rewiring into these mice. They also accumulate a lot of nuclear encoded component of the respiratory chain, and this is in line with the role of this complex in the turnover of this protein. And indeed, if you measure on intact mitochondria how these mitochondria can respirate, and you give them substrate for complex 1, so here pyruvate, glutamate, and malate, you see that there is a decrease in this mitochondrial phosphorylating in respiration, and also a decrease in the maximum ability of this mitochondria to respire. So we have an interesting phenotype here, and the question now is, now that we have a model that represent rather well what happens in the patients, can we identify druggable pathways to improve this axonal degeneration phenotype? And the way how one can think in this direction is, of course, you can imagine that if there is a process that leads to the axonal degeneration, we could try to identify these let's say, death pathway and try to block them. Or we could also try to identify pathways that are present into these axons to try to counteract the degenerative process and try to stimulate them. So I will show you something that we did in the past on the first, it's not enough, I thought it was an alarm. Okay. And yeah, so what we did in the past with the first strategy and something that we are trying to do now. So the first thing that we were thinking is we were realizing we have mainly a complex 1 deficiency in these neurons. So is it possible that we have some problem with NAD metabolism? This has been actually shown and seen in many mitochondrial diseases because complex 1 not only is important in the respiratory chain, of course, but is also important for regenerating NAD+. So many NAD+ depletion in mitochondrial is a hallmark of many mitochondrial diseases, and we know that there is an important enzyme that has became very famous in the axonal degeneration field, SARM1, which is an AD-consuming enzyme. So SARM1 is implicated in the salvage pathway, so it's able to degrading NAD and producing NAM, is one of the many enzymes that can do this. But what is really interesting about SARM1 is that normally this enzyme is in inactive state, but upon an increase in the ratio between NMN, nicotinamide, nucleotide, and NAD+, this enzyme becomes active and now becomes to consume very dramatically NAD+, and to produce cyclic ADPR and nucleotinamide. And cyclic ADPR and DPR are very strong stimulator of calcium channels, so this leads to massive calcium influx in the cytosol, calpain activation, and then axonal degeneration. And the reason why it is thought that this is an important factor in axonal degeneration is that if you have an injury to an axon, for example, you have what is called Wallerian degeneration, so the axon is cut. This enzyme, NMAT2, that is this enzyme that I showed you before that is important for the production of NAD+, has a very short half-life and goes drastically down. So you have a reduction of NAD+, an increase in NMN, increase of this ratio, and the pathway is activated. So by now there are a lot of studies that have very convincingly shown that if you delete SARM1, this is very beneficial to protect axons from Wallerian degeneration, so that the axons distal to the cut will die later. And also in many neuropathies of genetic and toxic origin is beneficial, and SARM inhibitors are being developed with the idea that they might be very beneficial in many diseases characterized by axonal degeneration, like HSP, but also ALS, for example. And we know that there are missense mutation in SARM1 that are also involved in ALS. So the question was, what happens if we block SARM1 in our mouse model? So we crossed our SPG7 knockout mice with SARM1 knockout mice. These mice are healthy, they have no sign of neurodegeneration. And what we observed at the beginning was very encouraging, because when we tested our mice on this walking beam, so the red mice are the knockout alone, and the blue are the double knockout, the black are the wild type. You see that the double knockout mice behave really like the knockout or even better at early time point, but later they also start to become, to take more time to cross this beam. However, when we tested them with a different type of test that is more important more than for coordination, more for grip and strength, so with the rotarod, we didn't see any effect of removing mouths in these animals. I show you just how this looks. So you can see this a wild-type mice, and oops, no, sorry. This is not working as it should work, because, sorry, now I don't know. This is a knockout mice, I think, and then the other one, should be a double knockout mice, exactly, that you see now it works. It works really much faster and much better. Okay, what is happening? So we looked, of course, in the spinal cord, and in the spinal cord we didn't see any effect. So the spinal cord was affected exactly in the double knockout like in the single knockout. You see again, swollen mitochondrial degenerating axons. We quantified all of these, we didn't find anything. However, in the cerebellum, the effect was completely different, because now we saw that while in the knockout alone, we have abnormal mitochondrial in the granule, in the cytosol of granule cells. You can see here, respect of a normal mitochondrial here. This is the dendrite of Purkinje neurons. In the double knockout mice, these mitochondrial effect were rescued. And when we looked, what was even more impressive at the neuroinflammation that is very strong in the cerebellum of these mice. So you see that they accumulate a lot of activated GFAP, and also they have activated microglia. This neuroinflammation was, at 28 weeks of age, was suppressed in the double knockout mice. So we also looked at the proteomic, and so we compared. Now here you compare the proteomic changes between a knockout towards wild type in the cerebellum at 28 weeks, and you see many proteins are changing. In orange you have the mitochondrial protein, but you have a lot of other pathways that are changing, that are related to ROS and glutathione metabolism, to calcium, lysosomal biogenesis, and many other. And of course you have the mitochondrial OXPHOS subunit and mitochondrial related pathway. But when we look instead at the comparison in the SARM1 knockout background, we only see the mitochondrial alteration that are linked to loss of the M-AAA protease, and we don't see all of these other proteomic rearrangement. So this would indicate that indeed we have a sort of a delay in the manifestation of the phenotype in the cerebellum. So this now is actually already published, so you can read the paper, and it was of course quite surprising, but it was not good enough, because first of all we don't know why we have this effect so specific in one population of neurons and not in another, but of course we want also to cure the degeneration of the long spinal axons, and it doesn't seem that this is the way to go, at least not completely. So now what I want to show you for the last few minutes is something that we are thinking at the moment, and I have only very preliminary data to show you. So the idea is, what about stress pathways that are activated in neurons upon the insult, that in our case is the loss of the M-AAA protease, and that might promote survival? Can we stimulate this pathway and can therefore promote or delay even more the degeneration of the axons? And here I have to introduce an important player of these stress responses, which is another protease in the mitochondria, which is called OMA1, which is activated preferentially upon stress. And it can be activated by several types of stress. Like, for example, if you have accumulation of protein that are misfolded, especially in the membrane of the mitochondria, if you have loss of mitochondrial DNA, if you have loss of ATP or reduced ATP level, if you have therefore OXPHOS dysfunction or oxidative stress or impaired mitochondrial translation, and also loss of the M-AAA protease, this protease is activated, and what this protein is doing is cleaving specifically certain specific substrate. We still don't know the entire range of the substrate of this protease, but the one that we know are extremely interesting. The first one is OPA1, that by the way, is also is a protein that is involved in the fusion of the inner membrane of mitochondria, is also mutated in optic atrophy. And when OPA1 is cleaved by OMA1, this leads to inhibit mitochondrial fusion and therefore leads to fragmented mitochondria. The other one, where I want to focus today, is DELE1, because this is an important molecule that communicates to the cytosol and actually then later to the nucleus the fact that there is mitochondrial dysfunction. And then now more recently, there is, has been identified also a component of the import pathway, DNAJ-C15, as a target of OMA1, and this blocks mitochondrial protein import when there is stress. This makes sense. There is stress, you don't want to overload the mitochondrial with protein. So what about DLA1? So DLA1 is a very important protein because it is a very small protein that normally is imported into the mitochondria. However, when there is stress and OMA1 is activated, OMA1 cleaves DLA during the import to the mitochondria, and DLA can retrotranslocate into the cytosol. And when it retrotranslocate into the cytosol, it binds this kinase HRI, which is a heme sensitive kinase that is one of the important kinases of the so-called integrated stress response. So what HRI is doing is phosphorylating F2 alpha, and this phosphorylation blocks CAP-dependent translation, but allows CAP-independent translation of transcription factor and other protein, but especially of this transcription factor, ATF4, which then is translated, can go into the nucleus, and can induce the expression of a series of stress-induced genes. Among the stress-induced genes, there are chaperones and proteases that try to resolve the stress, redox enzyme, and enzymes that rewire metabolism towards biosynthesis of amino acid and also towards synthesis of glutathione, so to basically cope with the redox stress. What is important to say is that what is happening here is also that among the genes that are also induced, there is also a phosphatase, or a component or a regulatory component of a phosphatase, GAT38, and these together with the PP1 then dephosphorylate F2 alpha and restore translation. So this is a stress response that at a certain point is terminate itself, or that maybe fluctuates into the cell. So why I'm telling you this is because this pathway is druggable, because we have molecules that inhibit the integrated stress response, or a molecule like cefin-1 that inhibit the phosphatase and therefore maintain this response active. And there was a very interesting paper from Francesca Maltecca last year on brain, where she used the cefin-1 treatment on mice that are lacking FG3L2. Now so the partner or SPG7, these mice are extremely sick. Indeed, in our mice facility, they're not even born, but she could obtain a few of these mice by outbreeding them with CD1. You see that they're extremely small and they die at 14 days. And when she gives cefin to these mice, she can prolong the survival of these mice for a week or so, but the mice also are quite bigger, so you see an effect. Now, so of course these mice have a very severe phenotype, so she saw quite some effect in the cerebellum, but it's not really clear, it's not really the nice model to see what is the effect. So are they simply bigger and therefore living longer? In which neurons is this beneficial? And therefore we thought we would like to know what is the role of this integrated stress response in SPG7 deficiency. So do these mice, our mice, show activation of the ISR? This is not very easy to see because our mouse model is not so severely affected. So in each tissue we have only a few neurons that are indeed or axons that are affected, but we perform metabolomic analysis on the cerebellum of these mice, and we see some evidence that this pathway of amino acid biosynthesis that are clearly, normally activated upon the integrated stress response are upregulated. So you see that normally what is clearly seen is upregulation of serine metabolism, and then serine goes into the transsulfuration pathway, and this leads to the production of cysteine, and then taurine, hypotaurin, and glutathione, and we performed some untargeted or semi-targeted, I should say, metabolomic together with the group of Christian Fretz, and we saw an accumulation of this metabolite, or some of this metabolite, in the cerebellum of the SPG7 knockout mice. So the question at this point was, if this is the case, what happens if we suppress this response in SPG7 deficiency. And we are lucky enough that the DLA1 knockout mice have been generated in the institute in front of us by Thomas Langer, we therefore got these mice. Again, these DLA1 knockout mice don't show any phenotype. We crossed them with SPG7. And what we saw, or what we are seeing, because these experiments are full ongoing, and I'm just showing you the very preliminary data, these mice are quite sick. So these mice become now suddenly extremely small. I think you can see here. They lose weight. They don't gain weight like the other, but they also, around 12 weeks, they start to lose weight, and actually now we are forced by the mouse ethical committee and to kill these mice so quite early. And I want to say this age is an age in which we don't see a lot in our SPG7 knockout mice. And when we tested these mice now on the rotor of the mice, these are female, the experiments are still ongoing, so still very little animal, but you can see that this double knockout, they really do not perform well at all. I just want to show you what happens, because this is a DLA1 knockout mice that is pretty normal, and this is, if I can start the video, mouse that doesn't want to go, but you will see now that it will start. And I think you can appreciate they have extremely pronounced kyphosis. They are extremely, extremely tiny, and they really have difficulty to cross. And this is a mouse is 6 weeks of age, so immediately 2 weeks after weaning, so very, very young mouse. So I don't have more data to show you, because we are now looking at the brain of these mice, of course, but not only the brain, but also other tissues. Because it could be that this response is what protects the patients to have symptoms in other tissues. But we think that this stimulating of the mitochondrial ISR could be actually a way to go, or something to test that we would like to do in the future. And with this, I finish my talk. I want to thank the people who did the work. So Carolina Montoro did the work together with Thibaut Molinier in the with the SARM, developed the new mouse model and also crossed it with the SARM1 knockout mice. And now Rejusha Parail is doing, looking at this DLA1 SPG7 double knockout. And I really want to thank my technician, Esther Barth, that is with me since years now. And she's doing always beautiful electron microscopy and doing a lot of work to help us there. So thank you for your attention.
Thank you very much, Elena. And in the interest of time, we agreed that we will have all the questions at the end of the session. So Linda Nemeshka will be the next one to move from SPG7 to SPG8 and also change the model. So looking forward to your talk.
Just hier, also genau, einfach aufmachen. Okay. Thank you for the introduction. Thank you for having me today. I'm very happy to share with you what we've been doing. So there was this discussion, who's the real outsider? I think it's us, because we're the only worm people here. So thank you for bearing with me. I will tell you today about our work in C. elegans on the function of WASH-C5 and what that tells us about SPG8. So I quickly want to remind you that SPG8 is caused by dominant autosomal mutations in WASH-C5. And as we heard this morning, there are certain pathways in the cell that tend to be always affected when you have these SPG genes. And SPG8, WASH-C5, is actually one of those that is involved in endolysosomal processing. So it encodes the WASH complex member strumpelline, and strumpelline and the WASH complex together function in the sorting of endosomal cargo. So if I click this for you, you can see that down here, these pink dots depict the WASH complex. And if you think about endolysosomal function, you have the sorting from the endosome either into the degradation pathway towards the lysosome or you can go, very simplified view, you can go via the recycling route and then get cargo back to where it's belonging. So you can imagine that this sorting function of the wash complex is very important for the function of this endolysosomal system and what happens to the cargo. We know all of this, and we know from several different model organisms and cell culture that if you have mutations in strombelin, that the system is anatomically and functionally very impaired. What we do not know is how that actually contributes to the pathology of SPG8. And this is where we come in, because we decided that we want to go a very different route than anyone else, and we would like to model this disease, or the loss of WASH-C5 function, in C. elegans. This is C. elegans. It's rather tiny, as I've depicted up here for you. It's just 1 millimeter. They grow very fast. You have 3 days from egg to egg-laying adult, and they live on like small 6-centimeter agar plates seeded with bacteria. So I think we can all agree that it's a very fast and very easy to maintain and very cost-efficient system, which is a great thing to have in the lab if you have limited space and means. So this is our favorite lab member, so to speak. And it has the second advantage that is very easily genetically manipulated. It's been as a genetic model organism around labs all around the world for quite a long time, and there are a lot of different techniques that you can use to basically introduce any kind of genetic modification that you want in very different means and techniques. What is very important for my talk today is yet another feature of this worm. It's actually a proper organism. I worked in cell culture, I really like cell culture, but what it's lacking often is traceable phenotypes. So this organism has the same complex tissues that mammals have, and I've just marked the muscle cells, for example, up here for you, or the various types of neurons that are in this organism. So that allows us to actually trace what we do genetically and incorporate the outcomes of these genetic changes by having a look and scoring the different phenotypes. And in case of SPG8, we are looking at locomotion as a readout for neuromuscular defects. And having said all of that, we think this is the biggest advantage of our system, so I will focus for my talk on the phenotypes that we found and that we analyzed. First, I have to show you that WASH-C5 is actually also present in C. elegans. And if you look at this, on the top panel, you see the human protein structure. Very important for its function is here in yellow, the spectin repeat domain, where most of the functional interactions happen with this protein. Also, the interactions within the WASH complex are happening in this marked in yellow spectrin repeat domain. And if you look at the lower panel, the C. elegans protein looks similar to the human one. It's a tiny bit shorter, but it actually does have the same domain structure. And if we model it in AlphaFold, we also see that it has a general overall structure that resembles the human one. So what we did was we introduced several mutations that would lead to a loss of function in this protein. And for the sake of time and simplicity, I'll be talking today about this one. It's called EF159. And it's a CRISPR-generated loss of function mutant. So we have introduced a very early stop codon into the WASH-C5 open reading frame, which leads to a truncated protein. You see it's a tiny stub of a protein here of 16 amino acids. So it's lacking all of the functional domains and definitely also lacking this important spectrin repeat domain. And when we now look at the phenotypes that are produced in worms that do not carry any wild-type alleles, but only this very short stub of a protein here, then we actually see nothing, at least when we look at baseline traits. So what I'm showing you here is a lifespan assay, and if you look at this Kaplan-Meier curve here, you see that the mutant worms live exactly as long as the wild-type worms. Next, in C. elegans, a very important measure for fitness of the organism is the brood size. And as you can see on this graph here, we've checked the brood sizes of quite a lot of single worms. These worms are hermaphroditic, so they produce their own sperm and they self-fertilize. So you can analyze the brood size of each individual, and they are usually very consistently around 330 offspring per hermaphrodite, and you see that stays pretty much the same also in our mutant. Next, we looked at basal locomotion, and what you see here is a vector graph of a worm that I filmed in liquid, and you can see that it does something that is called trashing. So basically it forms these C shapes from one side of the other, and if you follow this counter up here, you see that every time the C changes from one side to the other, we count half a body band. And in the end, we have a certain amount of time that we're filming the animal, and then we calculate the rate of movement or the locomotion as body bands per second. So what you see there is a young, healthy wild-type worm, and if I film young, healthy wild-type worms and young WASH-C5 mutants, you see again that there is no difference in the spatial locomotion in these young adults. However, since SPG8 is also a neurodegenerative disease that gets worse with age, we also looked at worms that are aging. So again here, this is the data, this is the data from the young adults on their very first day of adulthood, and I told you there is no difference here. If we look at adults on their 4th day of adulthood, basically that would correspond to at the end of their reproductive period, you see that there is also no difference between the wild type and the WASH-C5 mutants. That changes a little if we look at the, say, golden ager worms, so day 7 adults. You can see that both the wild type but also the mutant actually show a bigger variability in their trashing behavior. So meaning that the rate of movement that they show in this liquid becomes more distributed in these two populations. And I think what you can also appreciate, this trend is much, much more pronounced if you look at our mutant. So there are some animals that perform even better than wild type, but the majority or the much bigger proportion kind of drops out at the bottom. So we are generating with age this portion of low performer worms, as we call them, where some of them actually don't move at all anymore, which is rather rare in the wild type, and where quite a big portion of those actually shows less than 1 body band per second. And that's the threshold where you can really see also with the naked eye that the movement starts to get slower. Nevertheless, this is also not significant. So we do at this point not really see a difference in the movement. We just see a higher variability and a higher tendency towards less movement. So we decided to challenge these worms a bit to maybe really get the phenotype to become visible. So what we did was we did a locomotor stress assay. What that means is that the worms are put through a rather harsh regimen of swimming, and swimming actually means stress for these worms. They have to constantly move, their synapses are under constant stress, there is the whole neuromuscular system is under constant strain, and if we put them for 10 minutes into the water and let them trash, or into the buffer, not water, buffer, water would be even worse, and let them stress through the swimming, and then put them back on solid ground onto their plates. And then the next day, after a certain recovery period of 24 hours, we stress them again through the same rigid regime of 10 minutes of swimming. And right after that, we score. And what we're looking for is failure to complete this assay. And failure is defined as either the worm dies under the stress or the worm stops movement. And if we now look at these same time points again, then we see that we do actually have quite an effect on these worms. So let's start at the front here. These are day 1 and day 2 adults, so meaning they were naive worms that had their first bout of swimming on day 1, and then on the day 2, they had the second round. And afterwards, if we score them, almost all of them, I think this is one worm that actually showed failure, almost all of them that we looked at actually performed quite well, survived these 2 rather hard swimming routines, and actually were not counted as failure. We see the same picture if we look at our just after reproductive age worms. They were naive worms that had the first round of swimming on day 4, second round of swimming on day 5, and again, in this case it's a wild type worm, but I think it's pretty sure it was just one worm under all those worms that we tested that actually did not perform well in this assay. Now we're coming to our golden ager worms, where we have the first round of swimming on day 7 and the second round of swimming on day 8. And you see now also the wild type worms start to get more more susceptible to this stress, and we have a certain proportion of the wild-type worms now as well that starts to show failure, but we have a much higher portion of our WASH-C5 worms that collapse under the stress, where we see failure in these worms. So either they die or they show up as paralyzed when we do our scoring. So that shows us that even though the basal locomotion is fine in these worms, if we apply stress to this neuromuscular system, the robustness is missing. So we really have a much higher vulnerability under locomotor stress in our WASH-C5 mutants. So we asked ourselves if it's just this locomotor stress, so is it this rigid regime of the swimming, this constant movement that is actually stressing the system very specifically, or can we induce this actually also through other stresses? And I will just tell you very briefly, heat stress is not a problem for these worms. They seem to even perform a little better than our wild-type worms, but oxidative stress is a big problem. So what we did is a classical oxidative stress assay in C. elegans, where you put your worms onto agar plates, so we're back onto solid ground. And these agar plates have been supplemented with some compound that generates ROS. In our case, we use juglon, which is a compound from the black walnut, and it's a naturally occurring ROS generator. These worms are placed on there. They will stay on these plates for 4 hours, and we will every hour check again for failure. And again, failure means we find dead worms or we find worms that are paralyzed. Difference to the assay before, here we assess dead and paralysis separately. So I will start with the death. If we put these young adults that coped very well with the locomotor stress onto juglon plates at a medium to slightly higher concentration, so we have them at 240 micromolar. And we see that after 4 hours, we have a very significant decline in our WASH-C5 mutant worms as compared to wild type. So here's the log rank score, and you see this is highly significant, and we have a lot earlier onset of death, and we have a lot more dead worms at the end of our assay. So our mutants are really sensitive not only to locomotive stress, but also to oxidative stress. And in a further assay, we actually now checked for paralysis, and I would like to explain that a little better to you. So what you see in this upper panel are stills out from a movie that have been montaged one after the other, and this is an N2 young adult that has been 2 hours on a juglon plate, and you see that this worm moves in this sinusoidal wave pattern, the way that these worms move, and is very reminiscent also of their swimming pattern, and this worm here is not affected by the juglon. What you see in our lower pattern here is WASH-C5 mutant that shows a very stereotypic kind of paralysis where the posterior part, so the tail of the worm, stays stable. You see here, it doesn't move at all, and it very slowly swings the head from side to side. It also takes much longer than you see up here. So this is very characteristic. This loss from the proximal end of movement is very characteristic for neuromuscular stress in this in C. elegans. And if we quantify that on a bit lower juglon levels, we can see that after 4 hours, the proportion of worms that have been paralyzed under this juglon stress, so under this oxidative stress, is again much, much higher. So that points us towards the juglon being indeed a factor that stresses this neuromuscular system. And to sum up what I just told you, so what we've seen in our worms is that the loss of WASH-C5 function in C. elegans does actually not affect any overall fitness traits that we know of or the basal locomotion. Indeed, we see that WASH-C5 shows an age-dependent locomotor defect that is much more pronounced under repeated physical stress. And we see that also the same mutants are showing severe neuromuscular sensitivity or reduced neuromuscular robustness under oxidative stress. And to sum this up, I hope that I could convince you that at least for C. elegans, WASH-C5 is not a gene that leads to a loss of locomotive function, but instead it leads to an increase of neuromuscular robustness and to vulnerability that is very susceptible to different kinds of stress. And I want to thank my group and the group that actually hosts the C. elegans facilities and that shares them with us. And a very special thank you to the Tom Wallig Foundation that has been supporting this project, and especially to Susanne, who's a fantastic person to talk to, always encouraging and always very patient. So thanks a lot.
Thank you, Linda, and we will move directly to the next talk by Eleni Taispra and Isabel Hall. And with that, we will move back to SPG 7, but also to switch the model to human cells again.
Okay. Hi, I'm Isabel. I'm a PhD student in Evan Reed's lab.
And I'm Eleni. I'm a PhD student in Patrick Shinner's lab.
So we're collaborating on this project to establish an in vitro model of type 7 hereditary spastic paraplegia. So we'll be talking about the experiments that each of us have done to establish this model. So for some background, you will have heard some of this today, but SPG7, or type 7 HSP, is caused by loss of function by allelic mutations in SPG7. The modeled global prevalence is around 0.22 in 100,000 people. It's later onset, so median age of diagnosis is 39. And complex phenotypes are often observed, so typically presenting with cerebellar ataxia and optic neuropathy. On the gene itself, so SPG7 encodes the protein paraplegin, which is an inner mitochondrial membrane protease, and it functions in a complex with its paralog, AFG3L2. So together, this complex regulates mitochondrial proteostasis and protein quality control. And on our model, so the system we're using is an SPG7 knockout system. So the cells are not expressing SPG7, they're depleted of paraplegin. And that was generated using CRISPR-Cas9. So guiding the Cas9 enzyme to exon 4 in SPG7. And then we took forward clonal cell lines with frameshift mutations in that exon, as this is predicted to cause nonsense-mediated decay of any mRNA transcribed from the gene. And we then confirmed knockout with qPCR and Western blot. So the experiments will use a couple different SPG7 knockout lines, so with slightly different edits to that exon 4. On the neuronal system, so we're using I3 neurons. This is a very established method of deriving glutamatergic cortical neurons from iPSCs. I3 refers to integrated, inducible, and isogenic. So it's a transcription factor-based approach. It relies on forcing expression of neurogenin 2, or NGN2. This is a master regulator of neurogenesis in humans, and in the iPSCs we use, it is expressed under a doxycycline-inducible promoter. So by exposing the iPSCs to doxycycline, you then force high expression of NGN2, which stimulates the differentiation. So you can see that morphologically here, and we can also confirm that by looking at loss of markers of pluripotency, so loss of stem cell markers, like NANOG and OCT4. You can see this disappearing over the 2 weeks, and gain of neuronal markers such as tau and MAP2. So onto the phenotypes we've tested to validate this model. So some of these are based on existing literature. One of the things that's come up in cell lines that are derived from SPG7 patients is a reduction in neurite complexity. So we looked for that in 5-week-old neurons using shoal analysis, which entails imaging fixed neurons and then placing equidistant concentric rings around the cell body, which project outwards, and then counting the number of neurite intersections with each ring. You can plot that. The upper trace there is the wild type, and the 2 lower traces are different SPG7 knockout lines. What you can see is that the number of neurites projecting from the soma is already slightly higher to begin with in the wild type, and then there's a more rapid drop-off in neurite complexity, so faster drop-off in the number of neurite intersections in those two SPG7 knockout lines. So lower neurite complexity as predicted. We also observed a small decrease in the total axonal length in the SPG7 knockout neurons. Another morphological phenotype we looked for was axonal swelling. This is a very typical precursor to axonal degeneration in vitro. However, at the time point we looked at, so again 5 weeks, we did not observe a difference in the frequency of axonal swelling. Axonal swelling is observed in mouse models of SPG7, but that's months into development, so we expect this may just be due to this being an earlier time point. Another morphological phenotype, again based on existing literature, something else that has been observed is an increase in fragmentation of the mitochondrial network in cells derived from SPG7 patients. So we looked to confirm this with super resolution microscopy. So that's TOM20 there. It's an outer mitochondrial membrane protein that's shown in red, and then tubulin is shown in blue just to outline the neurons. Visibly, there was no difference at the two time points we looked at, so 2 and 3 weeks into a differentiation. Could not identify a difference in the fragmentation of the mitochondrial network. We quantified various features to confirm that. So the average size of the mitochondrial particles, the number of branches and junctions, the maximum branch length, among other features. We did not identify any difference, at least at these time points, in the mitochondrial network. We also performed some functional testing, so looking at electrophysiology on multi-electrode arrays. So the I3 neurons, they tend to show spontaneous electrical activity from around 2-3 weeks into differentiation, and you see more established patterns of synchronized activity from about 4 or 5 weeks in. And so one way you can look at this is with multi-electrode array plates. So this involves growing the neurons on plates of electrodes, and that allows you to look at activity on the network level rather than in individual neurons. And that's what that looks like in real time. So I'll show a couple of representative raster plots. So each raster plot is showing the activity in one well or a plate of neurons over a 15-minute period. In our setup, each well contains 16 electrodes. So within each row here, you're seeing the activity at one electrode. Each black line in a row is showing that the activity picked up by that electrode has surpassed a threshold, at which point we would say that action potentials are being fired by the neurons on that electrode. The orange lines are denoting events that we call network bursts. So these are just events in which the activity across the well is very synchronized. So synchronized activity across at least a third of the electrodes in the well. The general trend we observed was that in the wild type neurons, this is around 5 weeks into differentiation, you tend to see these blocks of network bursts, which are interspersed with not silent periods, but periods in which there are no network bursts, as you can see. Whereas in both SPG7 knockout lines we looked at, you tend to see much more regular, consistent network bursting. So there appears to be some dysregulation of that activity. One way you can quantify this is by looking at the coefficient of variation in the duration of the interval between the network bursts. Because that network bursting is much more regular in the SPG7 knockout lines, that coefficient is much smaller, as you can see there. So some dysregulation of that activity.
So another phenotype that is quietly observed in patient cell lines is reduced mitochondrial memory potential, and this is what we wanted to assess in our model as well. So just to give a bit of a background on that, the memory potential is the electrical potential difference across the inner mitochondrial membrane, and it is generated by the proton pumps of the electron transport chain. The memory potential is important for a number of aspects inside the mitochondria, but it is crucial for ATP synthesis, so energy production. And in the case where the memory potential is not functioning properly, then it is a major indicator of severe mitochondrial dysfunction. So the way that we assess this is by doing a live cell imaging experiment. So we grew our neurons up to day 21, and then while the cells were still alive, we stained them with 3 dyes. We used hooks in blue for the staining of the nucleus, then mitotracker in green for the staining of the mitochondria, and then TMRM in red for the staining of the memory potential. And then we image the neurons, and what we already see from the microscopy images is that the signal of the memory potential is decreased in the knockouts compared to the wild type. We then quantified those images. We used mitotracker as a way to normalize against TMRM, and after quantifying, we saw a significantly reduced mitochondrial memory potential in our knockouts compared to the wild type. We then wanted to do a bit of a more unbiased assessment of that, so we did a FACS assay. Once again, we stained the cells with TMRM and mitotracker green for normalization. The blue peak is our unstained control, the red peak is our wild type, and then the 3 other peaks are the SPD7 knockout lines. And after quantifying the FACS data, we do once again see a significant decrease in the memory potential of our knockout lines compared to the wild type. Another phenotype that is observed is reduced mitochondrial DNA copy number, and here we assess this by doing a ddPCR in day 21 neurons. So in healthy cells, the mitochondrial DNA copy number is around 1,000 copies, but what we observed was a decreased number of mitochondrial DNA copy number in our knockout cells compared to the wild type. So when the mitochondrial DNA copy number is decreased, it can be associated with reduced mitochondrial content and function, increased cellular stress, and energy deficiency. Then we wanted to assess the levels of some key mitochondrial proteins. And first we checked in TFAM protein, which is responsible for packaging the mitochondrial DNA. In the case where TFAM is reduced, then we have a reduction in mitochondrial DNA copy number and increased expression of mitochondrially encoded genes. So reduced TFAM means less protection from mitochondrial DNA damage. We also checked the ATP5A, and when ATP5A is reduced, then we have less energy production, which can lead to energy stress. ATP5A also helps to maintain the memory potential in reverse mode, and its reduction could be linked to the reduced memory potential phenotype that we observed. We also checked the levels of VDAC protein. So VDAC is responsible for overall the mitochondrial health, and its reduction is linked to global mitochondrial dysfunction, affecting energy production and inner mitochondrial structure, as well as the outer membrane transport. So to assess that, we did a Western blot probing for these proteins. We did first at day 0 time point to see if the damage is present even before the differentiation. And what we observed was decreased levels of TFAM in our knockout lines compared to the wild type, as well as decreased levels of VDAC. We then did the same thing in day 14 neurons to see how that progresses during the differentiation. And once again, we confirmed the lower levels of TFAM and VDAC proteins. So overall, what we see is that SPG7 loss leads to severe mitochondrial dysfunction, including significant loss of memory potential, mitochondrial DNA copy number, as well as decreased levels of key mitochondrial proteins. We also observe neuronal abnormalities like reduced neuroid complexity and dysregulation of electrical activity. So overall, the phenotypes that we observe confirm the development of an in vitro model of SPG7 in human I3 neurons. Our next steps include genome-wide CRISPR screens to identify genetic modifiers that are capable of rescuing the mitochondrial memory potential phenotype, as well as to identify synthetic lethal interactions between SPG7 and other genes. Those two screens will be complementary to each other and will form the basis of investigation into other therapeutically targetable pathomechanisms of SPG7. Before I finish, I just want to give some more details about the screens that are currently ongoing. So the synthetic lethality screen, we will first introduce our iPSCs with a lentiviral vector, grow our neurons up to day 7, and then collect the genomic DNA, do deep sequencing, and then bioinformatics to identify compensatory proteins or pathways and potential therapeutic strategies. Now for the FACS screen, which will focus on the TMRM phenotype, we will once again transduce our iPSCs, grow them up to day 7, and then collect the neurons and do sorting of TMRM, low and high TMRM. Then we collect the DNA, do bioinformatics analysis, and identify what genes that, when you knock them down, they are capable of rescuing the mitochondrial memory phenotype that was observed. I'd like to thank all of you for listening to us, and also our labs in Cambridge University, the Chinnery Lab and the Reed Lab. Thank you so much.
Talk. Can I please ask all speakers of a last session, Elena and Linda, to the stage, and then please speak up with your questions.
Thank you so much for the nice presentation. I wanted to ask, what kind of neurons? Do you know which neurons are in the, when you transduce them? Are there like excitatory, inhibitory, a mix of neurons in there? Because that might also impact how these neurons fire. Also, you know, do they form like networks or what kind of?
Yeah, the glutamatergic cortical neurons, they're so generally excitatory. The population is fairly homogeneous, based on the protocol that was established to derive that. And we've repeated that many times. The electrophysiology I showed, the trend has been broadly similar. So yeah, I would say that the trends we've seen with electrophysiology have been consistent over multiple replicates. Yeah, does that answer your question?
I have a question to Elena. Like you were showing the microglia that you can kind of decrease with SARM1. Did you also check for astrocytes at some point?
The problem is that, so you mean, okay. So astrocytes were also decreased, both of them. What we don't know now is whether this effect is intrinsic of the astrocyte, because of course our SARM1 is also full body knockout. So now the question is, would it be sufficient to decrease SARM1 in astrocyte to repress the ability of the astrocyte to become active? This we don't know. Maybe I can ask a question to them.
Yeah.
I'm very curious about, so it's quite remarkable the decrease in TFAM that you have. So do you have an explanation? Do you know whether you have increased mitophagy, maybe less mitochondrial mass?
Thank you. So this is something that I'm currently working on at the moment to try and identify why we see a decrease in TFAM. So we haven't checked into the mitochondrial mass, but it's something that we're actually doing at the moment. We do think that the reduced mitochondrial linear copy number could be associated to the reduced TFAM. So it's something to definitely look into more, but it's an interesting phenotype that we observed.
Okay, so thanks everybody. I see no further questions. Thanks to all the speakers for fantastic talks. And with that, I'll turn over to Jonas, and we are just in time.
Exactly. No, just the first 3 days of the differentiation. We're in a bit of different, once we replete the neurons after day 3, we remove the doxycycline as well.
All right, so moving forward. Right. So, yeah. Thank you. We have to move. Yeah, we have to. No problem. So moving forward, the idea of the event is to talk about the gaps in HSPs. We started with the genetics, and afterwards we know that we also need to refine the cell modules and also mice model for better understanding and also therapeutic purpose. And right now we go much more into the clinic, I would say, and the gaps related to that, talking about the natural history and also longitudinal data. So the first speaker of this session is Julian Alecu. He's a physician researcher, so please come here, Julian. He's actually a postdoc fellow at Stefan Hauser and Lukas Scholl's lab in Tübingen. He was also former postdoc of Darius, and he'll talk about the natural history and also genotype-phenotype correlation regarding SPG4. So the floor is yours, Julian.
Give me a second. Okay, no, thank you so much. Can you see the? Yes, thank you so much to the Bali Foundation and the organizers of the meeting for inviting this talk. It's a real pleasure to present our work on genotype-phenotype correlations in SPG4 on behalf of our team at Boston Children's Hospital, but really on behalf of a brilliant team of international collaborators that made this effort possible. So as many of you know, SPG4 is the most common form of hereditary spastic paraplegia, and it's typically inherited in an autosomal dominant pattern. However, a growing number of patients with de novo variants have been reported, and as we heard in Manon Degoutin's talk today, rare cases with biallelic variants are also being described. As we have also heard today, spastin encodes spastin, which assembles into a homohexameric complex that functions as a microtubule severing enzyme. And through this activity, spastin regulates the turnover and dynamics of microtubules, processes that are essential for neuromorphogenesis and the long-term maintenance of long axonal projections. Besides being the most frequent subtype, SPG4 is also notable for its remarkable phenotypic pleiotropy. And this is not only reflected in a pronounced bimodal distribution of the age at onset, but also, as many of you from clinical experience might know, by the presence of two very distinct clinical presentations, a childhood onset severe and complex form, and a predominantly adult onset, mostly pure HSP phenotype. And despite the well-established clinical heterogeneity for SPG4, robust genotype-phenotype correlations remain limited. Previous studies, including work from our own group, have suggested that missense variants and or de novo variants might be resulting or might be associated with more severe presentations. To date, however, the only established and repeatedly reported correlation that is linked, or the repeated genetic configuration that is linked to more severe presentations are either biallelic pathogenic variants or a pathogenic variant in trans with a intragenic modifier variant at residue 44. So this already highlights one of the several key challenges currently limiting clinical trial readiness for SPG4. First, the lack of systematic natural history data for early onset disease. Then secondly, as I have mentioned, the absence of robust and broadly applicable genotype-phenotype correlations that could support stratification of patients in the context of clinical trials. And third, the lack of a systematic characterization of the distinct clinical trajectories encompassed in SPG4. We therefore set out to systematically investigate these aspects of SPG4, and for this study, most of the patients were prospectively enrolled at Boston Children's Hospital through the natural history study on early onset HSP. And in addition, data on deeply phenotyped patients were contributed by 6 collaborating centers across North America, South America, Europe, and Asia. The inclusion criteria for this study were intentionally kept broad and focused on individuals younger than 40 years of age, a cutoff that we identified in the literature and that represents the period of greatest clinical heterogeneity. And with the relatively larger proportion of younger patients in our cohort, there also came, with that came an overrepresentation of de novo variants, which have previously been linked to an earlier age at onset. Notably, and somewhat unexpected, the overwhelming majority of these de novo cases carried missense variants. And in line with previous reports, we found that most missense variants clustered in the AAA domain of spastin, and that by far the most frequent recurring variant or variants affected the arginine 499 residue. Next, we performed a set of exploratory analyses illustrated here by the analyses of the spastic paraplegia rating scale over time. And overall, our cohort recapitulated previously described patterns with patients who were carrying biallelic variants or had an intragenic modifier variant showing an early and more severe disease course. When we further examined variant types, we observed that among patients who were severely affected early in life, there was a strong overrepresentation of both de novo variants and missense variants. And the vast majority of these patients, as I mentioned, the vast majority of these patients, independently of the mode of inheritance, carried missense variants. At the same time, a substantial proportion of patients who followed a more canonical moderate disease trajectory were also carrying missense variants. So we were wondering what differences for these missense variants might be, and so we took a closer look at the exact variants present in the cohort, and to our big surprise, we observed an almost complete segregation of the distinct missense variants in the cohort by inheritance. Variants occurring de novo were very rarely observed as inherited variants and vice versa. For some neurodevelopmental disorders, this pattern represents a well-known paradigm, where the most deleterious variants are relatively overrepresented among de novo cases because their severity and the severity of the associated clinical phenotype limits transmission across generations. Based on this observation, we hypothesized that these two groups of missense variants might have differential effects on spastin function, which in turn may give rise to the distinct clinical presentations. We therefore set out to classify missense variants. However, rather than restricting our analysis to the variants observed in our cohort and inferring deleteriousness from the associated clinical phenotypes, we wanted to come up with an independent classification system that potentially could apply or could be applicable to all SPAST missense variants, and rather use the clinical data that we had collected to validate this classification framework. For this purpose, we adopted a framework developed by Dennis Lyle's group out of Broad Institute, which provided a suitable basis for the classification of spastin variants. Their original algorithm elegantly integrates missense pathogenicity predictions, evolutionary constraint with variant burden analysis to identify functionally essential protein residues. Importantly, using high-quality protein structures, all of this is done within the three-dimensional residue neighborhood. And the underlying rationale for that is that for missense variants, the spatial neighborhood and side chain interactions of a given residue play a key role in determining the functional importance of a given residue and thereby dictate the functional impact of missense variation at this position. We extended this framework in in a couple of ways, and most importantly, we added we retained the variability in pathogenicity predictions for residues, giving us a mean to measure how variable and how context sensitive to the exact amino acid exchange the pathogenicity at this residue is. So we use an experimentally validated crystal structure of human spastin, of the human spastin hexamer, covering most of the microtubule binding domain as well as the AAA domains, where the majority of missense variants were located. And then we mapped residue essentiality across this hexamer. This is what it looks like projected onto the three-dimensional structure. Next, we collaborated with Dr. Ral Michak at the NIH to functionally annotate residues based on prior experimental work from her lab. We found that the essentiality predictions that we had previously generated closely aligned with these functional annotations from Dr. Michak. In particular, residues within the Walker domain, within the pore loops, as well as variants or residues located in an allosteric network coupling ATP hydrolysis to microtubule disassembly, were predicted to be functionally essential and therefore more vulnerable to missense variation. And somewhat unexpectedly for us, the most frequently affected residue, arginine 499, was predicted to be a context-dependent residue based on the variability in pathogenicity predictions. We therefore examined these variant-specific predictions and observed a strong divergence between different substitutions. And interestingly, this divergence was closely mirrored by the clinical trajectories associated with these variants. Patients carrying the arginine 499 histidine or leucine variant showed an early onset severe phenotype, whereas patients with the arginine 499 cysteine variant showed a slower progressing phenotype. And a similar pattern was actually observed for the age at onset, with patients harboring the predicted more deleterious variants presenting at a substantially earlier age than patients with, for example, the arginine 499 cysteine variant. To test whether these trends extended beyond arginine 499 and beyond our own cohort, we compiled age at onset data from more than 350 patients with spasticity variants reported in the literature. And indeed, we found that variants located at the predicted neutral sites were associated with a much later age at onset, typically in the late 30s, while variants at predicted essential sites had a median age at onset below 10 years of age. And variants affecting context-dependent residues showed a pronounced bimodal distribution, as you can see here in the violin plot shown in green. This pattern was largely resolved when these patterns were further substratified based on variant-specific pathogenicity predictions. So encouraged by these findings, we next integrated our missense variant classification with the aforementioned established genotype-phenotype correlations, so the presence of biallelic pathogenic variants or the presence of an intragenic modifier variant. And we used this to stratify the entire cohort. This allowed us to define a predicted severe subgroup comprising of patients with biallelic variants, missense variants affecting essential residues, as well as variants predicted to be highly deleterious at context-dependent residues. All remaining variants were classified into a predicted moderate subgroup. In addition to the patients enrolled in our study, we also complemented the data set with high-quality cases from the literature who fulfilled the same inclusion criteria. And to evaluate whether this stratification predicted outcomes that were relevant to our patients, we analyzed patient-reported quality of life as measured with the CP-Child score. As you can see here, the two subgroups began to significantly diverge from around 5 years of age, with patients in the severe subgroup showing a progressive decline in the perceived quality of life over time, whereas the moderate subgroup remained largely stable. We also found that subgroups diverge in their qualitative symptom profiles, with patients in the severe subgroup more frequently exhibiting severe developmental impairments and more pronounced motor dysfunction, often with an involvement of the upper limbs, as well as bulbar and autonomic symptoms. The subgroups also show a differential progression of motor impairment, illustrated here using age and sex-adjusted proportional hazards models for reaching key disability milestones. The need for a walking aid, which corresponds to a score of 4 on the Spastische disability scale, is shown here, and then loss of independent ambulation with wheelchair dependent, which corresponds to a score of 6 on the Spastische scale. The trajectories of spasticity and spasticity-associated symptoms as measured with the SBRS also differed significantly between the subgroups, with patients in the severe subgroup showing high scores early on in life and showing a steady progression over time, reaching near maximum levels by the 2nd to 3rd decade of life. In contrast, the moderate subgroup followed more of a biphasic trajectory, where scores remained low and relatively stable during childhood, sometimes with patients or families reporting these patients as being basically subclinical as being healthy. And their scores remained low, relatively stable during childhood, typically below a score of 10. This was followed by an acceleration of clinical progression, typically during adolescence, around the second decade of life. And importantly, these increasing SPRS scores were strongly associated with declining quality of life, linking a worsening of spasticity directly to patient-relevant outcomes. Given the aforementioned upper limb involvement in the severe subgroup, we examined the evolution of spasticity in more detail using the modified Ashworth scale. And for the severe subgroup, we observed a clear temporal and spatial pattern where spasticity first affected distal lower limb muscles, then progressed proximally, while upper limb spasticity typically emerged later during the second decade and accelerated thereafter. And to better understand early development in patients, we analyzed the timing of motor milestones, and patients in the severe subgroup showed substantial delays across gross motor milestones, with many of the milestones not being met by many patients. Differences between the subgroups were already visible for unsupported sitting, but became much more pronounced for standing and walking, with only about a third of the severely affected patients eventually achieving independent walking. In contrast, patients in the moderate subgroup generally achieved these milestones, albeit with some of the patients showing slight delays in motor milestone attainment. And beyond motor delays, we also analyzed developmental quotients for other domains, including expressive language and social behavior. And analyses of these developmental quotients also suggested that there was a developmental stagnation in the severely affected patients for these domains. Lastly, in an exploratory analyses using the Simoa system, we measured plasma neurofilament light levels in a subset of 26 patients and a larger group of age-matched controls. And we observed subgroup-dependent differences with differentially elevated plasma NfL levels in the severe subgroup. And these levels gradually declined with age and began to converge with the moderate subgroup during adolescence, but both subgroups remained elevated compared to controls. And we also compared these NfL measurements with large cohort studies, one including pediatric levels for plasma NfL, and only for the severe subgroup, only a single patient fell within the 99th percentile of the reported normal range of plasma NfL levels. Importantly, albeit a weak association, but there was a significant association of plasma NfL levels with clinical severity as measured with the SPRS. So with this, I would like to conclude and summarize that the essentiality informed variant classification of missense variants combined with established genotype-phenotype correlations helped to stratify SPG4 into two major clinical trajectories in this cohort, distinguishing a severe early onset from a moderate adolescence to adult onset disease. And importantly, the severe subgroup shows early and multi-system involvement, including severe developmental delays, intellectual disabilities, as well as a faster progression of motor impairment with an earlier loss of ambulation and differentially elevated plasma NfL levels. And with this, I would like to come to an end and thank our team at Boston Children's Hospital, as well as the aforementioned brilliant team of international collaborators, of whom I see a couple of faces. A couple of collaborators are here today. And most importantly, I would like to thank the patients and their families who made this research possible.
So you're going to move directly, questions at the end, right? So I would like to invite Emilien Petit. He's a biostatistician at the Paris Brain Institute, working with Professor Alexandra Dur's group. He has a lot of experience with the analysis of pooled databases of major natural history studies in both ataxia and HSPs. And he will talk to us also about the natural history and genotype-phenotype correlation, but in SPG7, in a study related to the PROSPAX cohort. So Julian, the floor is with you.
Thank you very much for the introduction, and thank you for having me here. So yes, I'm going to present some results about the PROSPAX study, which is a natural history study of both SPG7 and RSAX patients. So the PROSPAX study was an international multi-site longitudinal study with patients recruited in the UK and France, in Netherlands, in Germany, Italy, Turkey, and Canada too. So there were two different genotypes, both RSAX and SPG7 patients. Healthy controls were also recruited, and it was a 2 years follow-up study with annual visits, so a baseline visit, a 1 year visit, 2 year visit, and a lot of assessments were done, but I'm going to talk in this talk only about the clinical assessment, the digital motor assessment mainly, and a little bit of the MRI assessment. So we really have two very different populations in this study, mainly because our SACS participants have a very earlier age at onset during the childhood, I mean around 6 years, whereas the SPG7 onset is way later in life. So this have a direct impact on the age of participants. SPG7 are much older, and also their disease duration is lower than our SACS participants. You got here some mean value at baseline for participants. So we also saw that see that our SACS patients have higher, for example, SAR value, SPRS value, and force ideal value. Maybe some of you are not familiar with the SPAX comment. I'm going to talk about it a bit later. So there were some digital motor outcomes that were measured. They are divided in two groups. So first the APDM1, which are gait outcomes. At their gait outcome, they were measured only on ambulatory patients, and as our SACS patients are a bit more severe, there is, you can see here, very few longitudinal data for our SACS and much more for SPG7. So, and these gait outcomes were measured in two different conditions. First, free walking, and also lab conditions. So basically, just sensor put on the body that measure a lot of different things, different metric of the gait. We also have Q motor outcome, which are related to a limb coordination outcome. And for this data, we got a little bit more because the patient doesn't need to be ambulatory to be assessed with these assessments. And also, we got some brainstem cerebral volumetry. So first, I'm going to talk about this scale that was developed. It was actually one aim of the PROSPAX study. So the idea was, as we have patients that have both ataxia and spasticity, we want to build a scale that can measure the two things at the same time and that is sensitive to change. That was the main idea, was to build a sensitive to change scale. And to do that, colleague Cecil DiFolco just tried to make a combination of items taken from the SARA, the SPRS, and the force RDL to build the most sensitive to change scale. And finally, there were 7 items that were selected. So the gait from the SARA, the sitting from the SARA, the fast alternating and movement from the SARA, the walking from the SPRS, climbing stair from the SPRS, weakness of foot dorsiflexion from the SPRS, and the personal hygiene item from the FRS ADL. So this scale shows, of course, a good sensitivity to change because it was the aim of the scale, but also it shows very nice correlation with the disease duration and disease stage. So next I'm going to present what we did with the longitudinal data. The first thing was just to see what was moving. So we just build up linear mixed model so basically just slope, population slope since the inclusion in the cohort. And we see that all our 4 clinical variables are very significantly increasing. So you get the p-value above the mean slope and with the confidence intervals. And among the digital motor outcomes, there is really one that stands out from the other, is the harmonic ratio, both measured in the free walking condition and in the lab condition, which is significantly decreasing both in the pooled cohort, but also in SPG7 and RSACs taken separately. So I'm not an expert of digital motor outcomes, but harmonic ratio is basically a measure of both the smoothness and the rhythmicity of the gait. And it has been shown to be sensitive to change in other neurodegenerative disease. So we know what variables are significantly changing in the patient life, but we wanted to see if these changes were actually encoded to the patient impression, because meaningfulness is a big thing in developing scales. And so each patient at each follow-up visit were asked if his or her functional impairment due to the neurological disease compared to the previous visit has either improved, was unchanged, and worse, or worsened. So what we would like to have is what we can see, for example, in SPRS, in SPG7, is that we want to have patients experiencing a worsening with an increase of the scale, patient experiencing no change at all with no change on the scales, and patient experiencing an improved with an improving on the scale. But we see that it's not the case for most of the variables. For example, for the SARA scale in the pooled cohort, we got this big difference between worse and stable patient, worsening and stable patient, but the improved patients are nearly overlapping with worsening patient. Nevertheless, we still got these 2 digital motor outcomes, so harmonic ratio and free walking and lab, that are quite nicely, mostly the one measured in lab, anchored to the patient global impression with this quasi-linear trend. So with patients experiencing improvement that are really improving and respectively worsening. Then we wanted to see if the changes from both digital motor outcome and clinical outcome were correlated together. So as some of them were decreasing and some of them were increasing, I just inverted everything, so everything was basically increasing. So what we would like to have is a red matrix with everything going in the same direction. And we see that it's not the case. We got some weak to moderate correlation between clinical outcomes, mostly between SPAXCOM and other scale, and it's expected because SPAXCOM is built from item from the SARA and SPRS and also FORCE ADL. We got some intra-correlation, so the harmonic ratio in the two conditions, and for example, double support and the swing variation, but we have no correlation of changes between clinical variable and digital motor outcomes. A very important thing too when we are looking at longitudinal progression and thinking about how it could be used in clinical trial is to look at the sensitivity to change, and this is done by calculating what we call SRM, so standardized response mean, which is basically a signal-to-noise ratio. So we are looking how much we see patient improving compared to how noisy the signal is. And so what we can see is, of course, that SPAXCOM is the top variable, but it was designed especially for this. So hopefully we got it at the top. But we also see that the second one, at least in the pool analysis, but it's the third one in SPG7, is the harmonic ratio in free walking, which outperform all the clinical variables in the pool analysis. So once again, it's these variables that stand out from all the digital motor outcomes. But I just wanted to make a reminder that the SRM are quite low because, for example, here it's around 0.45, and that in sample size prediction, it will be 160 patients needed for a one-year trial with a 100 efficacy drug, so with a flat progression of patients under the drug and with 80 power. So these SRM are quite low, and we are not very likely to observe the efficiency of a drug in a trial when using this variable alone. So what we try to do is to combine multiple digital motor outcomes to build a more sensitive scale, and I will just talk about the graph on the right side. So just as the SPAXCOM was designed, we try to iteratively add the digital motor outcomes to increase, to optimize, to maximize the SRM. The dashed line is the SRM of the SPAXCOM, and we see that with the addition of 5 digital motor outcomes, we finally managed to outperform the SPAXCOM SRM. But it's not the case in SPG7 plus RSAX, so we think that the signal-to-noise ratio of these variables in RSAX are way too low. Finally, we wanted to look at if there were some predictors of the change. So we looked at the baseline value for all patients and to see if this was correlating with the annual slope of every clinical and digital motor outcomes. And after adjustment, we finally had only one significant correlation that was remaining. It was a correlation between the variance of the finger tap speed, and it was negatively correlated to disease duration. But in fact, when we look at the scatter plot, we see that there is absolutely no correlation in the early disease when the disease duration is low, but it's only correlated to this finger tap variance change with a very high, very severe patient with long disease duration. So in conclusion, we've seen that we can track the clinical progression and the APDM and Q-motor assessments over the 2-year period of this PROSPECT study. We also seen that the changes in clinical variables are not correlated to the changes in digital motor features. But still, we got this interesting variable, the harmonic ratio, that have quite good anchoring to the patient global impression of change, and also that have the best sensitivity to change. We also saw that SPAXCOM outperformed all clinical and digital motor outcome in sensitivity to change, but as it was built on these data, this needs to be replicated on independent data because it's very linked to the PROSPECT study. And we also saw that we could combine the digital motor assessment to build an even more responsive scale, and that there is no strong predictors for both the clinical and the digital motor outcome progression. So I just wanted to thank Sophie Tesnas-Dumoncel and other people that worked on this data, Willow Scott, Cécile Di Folco, and Charlotte Dubec, and Mathis and Rebecca for coordinating the whole PROSPECT study. And of course, a big thank to all PROSPECT investigators and participants. Thank you very much.
So again, questions at the end of the session. Thank you a lot. Now I invite the last speaker of the session, which is Marcondes França Jr. Marcondes is a full professor of neurology and the head of neurology department at Unicamp in Campinas, Brazil. He's also the co-coordinator of the iAxon Brazil, which is a network related to HSPs, and is a world expert in MRI as a biomarker for ataxias as well as for HSPs. And what he's going to talk to us today is about longitudinal data with MRI in the context of HSPs. So once it's working, the floor is with Marcondes.
So apologies for the delay, so technical issues, but we're now ready to begin. So again, thank you for the invitation. It's really a pleasure to be here once again in this symposium. And it's good to see that we have some spot to talk about imaging. I think this is an important part of, biomarkers and the investigation of potential, tools to assist us in the design of clinical trials. So in this talk here, we're going to focus on the brain. So it might be a little bit surprising because, of course, when you think about HSPs, we all think about spinal cord as the main target. But as we shall see here, we do have some interesting data coming from this approach, looking at the brain. So I have no disclosures. So some of the studies I'm going to present to you have been funded by the Spastic Paraplegia Foundation. And briefly, I'm going to try to cover in this next few minutes so an overview of what we have in terms of brain involvement in these diseases. And of course, then I'm going to talk about some data on longitudinal brain evaluation of these patients. So as I mentioned to you just recently, so HSPs are well known as a spinal cord disease, so that would be the hot target in this disease. But indeed, so we do have some evidence showing that the brain is also an important site of damage in this condition. So the first line of evidence comes from genetic data. So if you think about all the genes that we know that are responsible for HSPs, some of them are massively expressed in the brain. And this is the case, for example, of Spastische, so SPG4 gene, where we can see here in yellow that the brain is really has a massive expression of this gene. So the second evidence that supports that the brain is also involved in this group of diseases comes from the phenotype. So as we all know that we take care of these patients in clinical practice, we know that some of them have, in addition to the typical spastic phenotype and weakness in the lower limbs, we also have, for example, movement disorders. We can also have cognitive decline, which are obviously connected to brain damage. So quickly, I'm going to show you a video of one of my patients with SPG11, and I think it's quite easy to appreciate that he has a pretty severe Parkinsonism. And this obviously has connection with damage to specific brain regions that we can see, for example, here in this throat scan, absolutely abnormal uptake in the deep nuclei of the brain. And finally, I think the third point that I'd like to briefly touch upon here is that we know for many years that some HSPs, we can see some specific radiological signs which are typical of these genotypes. And of course, this is helpful from a clinical point of view. Perhaps the most known of these clinical radiological signs is, of course, the ears of the link sign combined with the corpus callosum thinning, which is typical for SPG11, but also SPG15. So in summary, we already know, we had pretty good, I'd say, insights that the brain would also be damaged in this disease. But something different comes when we think about using computational imaging. So we're now shifting from this clinical evaluation of brain data sets and now thinking about using computational tools to evaluate what's really going on in the brain of these individuals. So what is computational imaging? I'm sure that some people here are not familiar to these imaging terms, terminology. So when you think about computational imaging, I'm talking about techniques that enable to perform quantitative analysis of the brain. And for example, we basically use, when you think about the studies in patients with HSP, we use two different approaches. So the first one would include techniques that look at the gray matter of the central nervous system, especially the brain. And here, for example, this is the one that is most frequently used, called free surfer, but we have others as well. So what this kind of pipeline typically does is to perform an automated segmentation of the gray matter structures of the brain. So we're able to compute, for example, the cortical thickness for many different brain regions. We're also able to calculate, for example, the volume of the deep nuclei within the brain. So we have numbers, and each number for each patient. And if we have, for example, longitudinal data, we're able to see how this kind of measurement performs in the long term. But on the other hand, we also have other pipelines, other techniques that enables us to evaluate, for example, what goes with the white matter. And we can apply volumetric analysis, such as the one we've just seen with the free surfer. But what seems to be more sensitive, and I hope I can prove this to you with the available data that we have, is the assessment of what we call microstructural integrity of the white matter. And to do this, we typically use a sequence that we call diffusion tensor imaging. So this is a modified version of the tissue, the clinical tissue that we typically use in our routine scans. And using some mathematical evaluation of this acquisition, we can assess whether white matter is really well structured. And we can compute, for example, different metrics such as FA and the diffusivities, as you can see here. So whenever we have a good integrity of the white matter tracts, so the FA varies from 0 to 1. So when it comes close to 1, it means it's better. And when it comes close to 0, it means it possibly has some kind of damage. So in summary, these are the most important pipeline that we have in clinical practice and in research to investigate what's going on in the brain of these patients. So, and what is the relevance of this kind of approach? So why should we do this in patients with HSP? So I think the first reason to explore this line of investigation comes to understand disease biology, so to assess in more detail the genotype-phenotype correlation. For example, this is a quick example to show you different patterns, different signatures that we have in two different diseases. So SPG11, one of the most typical complex phenotypes, and on the other hand, we have SPG4, which is, even though we have seen today that we might have also complex phenotype, it's more typically associated, at least in adults, with a pure phenotype. So as you can see here in these two images, the pattern of damage in the brain is absolutely different in these two types of the disease. So much more widespread damage in SPG11 involving, for example, the basal ganglia, and much more widespread damage to the white matter in SPG11 compared to SPG4. An interesting insight that we can obtain from this kind of approach is trying to somehow correlate and compare what happens to the maps of structural damage of the brain compared to the maps of normal gene expression within the brain. So this is a publication that we were able to have some years ago, published in Movement Disorders, and the idea here was to assess how similar were the maps of structural damage in the brain, assessed using free surfer, using DTI, compared to the maps of physiological brain expression of the genes related to HSPs. And as you can see here, I think you can appreciate here, so in the left side of the slide, you got the correlation in SPG4, which I think is quite interesting to see. You can see that the regions with more severe damage are specifically those where we have higher expression levels of spastin. But on the other hand, when we come to SPG11, on the right side of the slide, it's quite different. So we have much more widespread damage, including regions where we see essentially minimal or no gene expression. Again, thinking about the physiological pattern of gene expression. So in summary, we can get some insights, and we can see that the mechanism behind brain damage in the different HSPs are possibly different. But now coming to the real focus of this presentation, I think this is perhaps the most practical relevance of doing brain MRI studies in these patients, comes with the potential development of biomarkers. As we all know, HSPs are slowly progressive diseases, and so thinking about designing a clinical trial for these patients is really challenging because clinical tools often fail to capture progression, especially if you think about short-term design of clinical trials. So thinking about this perspective, so developing potential biomarkers, we need to see what we have in the literature. And so here in this slide, you can see what we have in terms of cross-sectional data. So we do have something out there in the literature. So most of these studies were done in single center-based cohorts, so not really that big. But we do have some data, for example, in SPG 3, 4, especially 5, 7, and 11, and adrenomyeloneuropathy, which is another closely connected condition. So there are differences, of course, depending on the genotype, so the signature is a little bit different, but the overall pattern that we see in these diseases is much more severe and much more widespread white matter damage than gray matter damage, especially if we think about these late onset subtypes and the pure phenotypes. And it's interesting because in some of these studies, you're able to see not only that there is damage to the brain, but also you're able to see that some regions within the brain are, connected, are correlated with the severity of the disease. For example, this is a publication from our group from some years ago where we're able to see that in SPG 11, we're able to see that there is precentral gyrus, thinning, and that this thinning of the precentral gyrus has, I would say, quite good correlation with the SPRS scores in this cohort. So showing that this might be a potential outcome measure for clinical trial. So there is a good correlation. But of course, to validate this assumption that this might be an interesting candidate for a biomarker, we need longitudinal studies. And at this point, I shall tell you that the literature is quite limited. So we don't have many studies published out there with this kind of a longitudinal design. So perhaps one of the first one that has been published many years ago by the Italian group from Professor Andrea Martinuzzi. So he was able to recruit a kind of a mixed cohort of patients with HSPs. As you can see here, I think 31 patients with the different genotypes. And he was able to follow some of these patients. So even though the cross-sectional data was from 31 patients, the longitudinal evaluation was only from 19 of these patients, so a smaller cohort. And in summary, what he was able to see was that there was no significant gray matter changes. So volumetric reduction was not progressive in this cohort, at least. And he could not also find anything on spectroscopy, so nothing really. But he was able to see something going on in the white matter. As you can see here, I just get some of the regions and the white matter tracks that he found different. And you can see here, for example, that the white matter just beneath the precentral gyrus, it was altered in this patient and it progressed in the long term. So this was the first study. Of course, there are limitations because of the sample size was not that big and because this was a mixed genotype cohort. But even so, we can appreciate that the white matter seems to be a potential outcome measure because he was able to see such a difference in an interval of around 1 year for these patients. So thinking about this, we decided to design a study in our center. So this is an ongoing project run by Carelli Salazar, which is my postdoc student. This has been funded by the Spastic Paraplegia Foundation and FAPESP. And basically, the goals of this study were to try to somehow characterize what's going on in terms of the natural history of CNS damage in these patients. So we are looking both at the spinal cord, but also at the brain of these patients. And the initial 3 cohorts that we're focusing on are SPG4, I'm going to show you some results, SPG11, and also something going on SPG7, but I'm not going to be able to show you results from SPG7, only SPG4 and SPG11 so far. So briefly commenting on the SPG4 cohort. So we're able to recruit for this study 27 patients, all adult patients, and 32 controls. These individuals were followed for around 6 years, so we were able to see three time points, so baseline, visit 1, and visit 2, so a relatively large follow-up period for these individuals. And we employed a mixed model to evaluate what's going on in terms of brain change in these individuals. So gray matter, we employed volumetric analysis and cortical thickness as well. And white matter, we employed the diffusivity analysis that I've shown to you previously in this paper. So the first image I'd like to present here, this would be the comparison. So that would be between time baseline and time 1, and then time 1 against time 2. So these are the maps of the difference comparing patients and controls. And I think you can appreciate that there is some, increase in the areas affected when you think about visit 1 and visit 2. And these are essentially those related to the corticospinal tracts. I think you can appreciate here. You can barely see something going on in the corticospinal tract in the first evaluation, and then it appears in the second evaluation with a higher effect size. And in the end, when we applied the formal statistical modeling, we were able to see that the FA value, which one of the values derived from DTI at the corticospinal tract, indeed we had a significant change over time from baseline to time 1 and then from time 1 to time 2. And comparing the performance of this metric against SPRS in this same cohort, we're able to see that the SRMs, the same metric that Emilian has just told you, was quite similar. So we're able to capture something is really changing in the brain of these patients, but this does not seem to be more sensitive than clinical evaluation to track disease progression. So I think this is interesting data. I think this is original results, but I think we still need to do better because this would not really help a lot in terms of designing clinical trial. The scenario is a little bit different from SPG11. I would say, I think more, exciting, I would say. So this is a more recent cohort. So it's already, as I mentioned to you, this is an ongoing study. The cohort is smaller, so we have 14 patients and 31 controls, and we have so far two time points. So just 2 visits with 1 year interval and the same approach in terms of the imaging analysis and also in terms of the statistical approach. And in contrast to what we have seen in SPG4, which was quite limited in terms of the regions which were abnormal, here we had much more results. And first, we were able to see that there was significant progression of gray matter damage, and perhaps the 2 most relevant results came from the precentral gyrus thickness. As you can see here in blue, you got the control values in both time points, and you can see here in red the results for the SPG11 cohort. And in the right portion of the slide, you can see the volumetry for the caudate nuclei, which again showed significant results in these patients after just 1 year of follow-up. When you come to see what's going on in terms of the white matter, we again found quite interesting results. The first one comes from the corpus callosum. This is a very interesting region when you talk about SPG11. As we all know, this is known to be affected since the description of the disease, but there is some doubt on the literature regarding the nature of this involvement, whether it's developmental or it's degenerative or both in the end. And what we're able to see is that, at least thinking about diffusion metrics, it seems to be progressive. So there is a progression of the corpus callosum FA in this cohort, and we're also able to see a progressive decline in the FA values for the cerebral peduncle, which is also a region important for this kind of condition because it's the way where the corticospinal tracts path within the brainstem. So in summary, in SPG11, we're able to see much more damage, and progression was able to be captured in this 1 year interval. So in the short term, it was able to be captured. And looking at the SRM, you have seen that in SPG4, not a significant difference, but here it's quite different. So SPRS, the SRM was around 1. So we all know that SPG11 progresses at a faster pace than SPG4. So this is the reason why the SRM is a little bit higher. But if you look, for example, at the volumetry of the putaminal nuclei, and for example, at the FA for the body of the corpus callosum, we have higher SRM values, suggesting that this might be an interesting biomarker to be used in clinical trials, at least as an exploratory measure. So I think SPG11 might be an interesting condition to think and to include eventually imaging data as potential outcome measures. But even though these are promising and interesting results, we do have many limitations. So there are a limited number of studies. So these are essentially single-center cohorts. So the cohorts are small and the follow-up times are really short. So we need to improve these. And I think we have some experience now with international imaging consortia. For example, the TRACK-FA consortia, the Enigma ataxia consortia. We're able to get information from many different centers across the world. And this would be the best way to move forward and to really validate these potential biomarkers and eventually to find new ones. So I'd like to finish with my take-home messages. I hope I have been able to show you that the brain is also damaged in many HSPs. I think neuroimaging is able to capture such damage in a very precise and quantitative way if you use the right tools to make it. This advanced imaging approach definitely helps us to understand what's going on in the disease, so a good correlation between genotype and phenotype. And finally, thinking about the use of this kind of tool as a potential biomarker I think it's promising at this point, at least for SPG11. I think we have pretty interesting data coming out using the diffusion data set. But again, we still need to work more, and we need more studies with larger cohorts and longer follow-up periods. I'd like to thank by thanking my group in Brazil and our funding agencies, and I'll be happy to take any questions. Thank you so much.
So, Julian and Emilian, please. Since we have the coffee break, so I think we will have a space for 2 or 3 questions, depending.
So I have a question for Julian. I'm interested in these pediatric NFL values. I assume they were cross-sectional only. Because what's the interpretation? They are younger ages, they go really up, very high, and then they decline, at least cross-sectionally. What's the interpretation behind that? Because it's in other pediatric ataxias, you see the same thing, but very little longitudinal data, and at least I don't know how to interpret it.
Absolutely, excellent observation. Again, very limited data set, 26 patients. I think we had a follow-up for 2 patients only, so very exploratory in nature. I think the one big trend that you mentioned are those very high levels, at least for an HSP, those very high levels in early childhood. And I think our again, very speculative, but our take on it is that this might be a superimposed either degeneration or loss of or neuroaxonal damage that results from alterations to neurodevelopmental programs that are still taking place during that period. And that this might explain why there is such a high or comparatively high levels of NFL compared to the more moderate patients who experience their acceleration of clinical severity much later.
Any other question from the audience?
Maybe following up on your idea about decreasing NFL in particular early onset, did you check for myelination in your early onset patients, whether they have myelination at that time point or it's lacking and therefore leaking much more NFL?
Again, excellent question. I think we have some imaging data for some of these patients, but I'm not aware of whether we explicitly checked for late myelination. I don't know, Darius, if you want to.
I can comment on it briefly, and I'll touch upon it very briefly in my talk as well. But it's interesting, and I think you pointed this out as well. Even in healthy children, NFL levels are higher in the first 2 years of life, and it's an interesting hypothesis because even in healthy children, myelination is not complete until about 24 months. In many complex forms of HSP, we see delayed myelination, so that might extend a little bit. So it's an interesting hypothesis. We have not looked at a correlation between the lack of myelination post 2 years of life and NFL levels. I think the cohort is too small for that, but it's an interesting hypothesis.
Any other question? I have one question for Marcondes. You showed mainly data from brain MRI. I know it would be another talk to talk about medulla, spinal cord, but can you just like comment on what you think is more promising for a trial perspective, like if there is any, it's more sensitive or not?
Yeah, so I mean, as I mentioned, so in this study we're running, so we're looking both at the brain and the spinal cord. So in the end, what happens is that, you know, spinal cord is also affected. So if you look at the cross-sectional data, it's really interesting, but the longitudinal data, I think it recapitulates what we have seen, at least for SPG4 and 11. So it's harder for us to see a difference in SPG4. So we did see something going on in specific tracts within the spinal cord, whereas in SPG11, we were able to see a lot of difference in terms of the, you know, when we compare baseline and time one. So my general perspective, I think it depends on the disease. So SPG11, we might think about using both brain or spinal cord. Possibly for others, I think we might need to choose one of them, which seems to be better. But there is another point which I think might be relevant, is when you think about using this in a clinical trial, especially if it's a multi-site clinical trial, it's a lot harder for us to harmonize spinal cord acquisitions and spinal cord analysis. So if we're able to come up with something from the brain, especially something which is easy to implement, like volumetry or quite basic evaluation of DTI, that would be better in terms of using in a clinical trial, using in a multi-site investigation, I think.
I'm going to ask you a very quick question to all three of you. I think one thing that I always think about when I see this beautiful work is what parameter, what clinician-reported outcome measure or biomarker, imaging marker would show me a change in the shortest period of time. So it's a big question, but what I'm trying to ask is, if you had to point your finger at one thing that changes in the course of months or maybe a few years, what would that be? Because everything you've shown so far, I think, as from what and all our own data points to changes that are more in the order of several years, and that's a problem for clinical trials, obviously.
Yeah, I think it very much depends on the stage of disease, but just looking at the SRM, as you showed, for now, this is the MRI that shows the best sensitivity to change, and with the advantage that we can use it even in the pre-symptomatic phases. So I think, to me, MRI is the most promising variables.
Maybe if I might add to that, I think especially mechanistically informed biomarkers that are either immediately downstream or in the middle of the pathomechanistic cascade are probably or will probably be most sensitive to therapeutic or to changes arising from a therapeutic intervention. So I think it's essential that the clinical side and the more basic science focused researchers collaborate on identifying potential mechanistically informed biomarkers.
Yeah, I think I agree with both of you. So of course, imaging is something we've been working on, and I feel like it might be really helpful. But again, it's going to depend on the disease and the stage of the disease, the cohort profile. But I think it has a lot of advantages. So as Emilian just mentioned, something that I think might be helpful is using inertial movement sensors. This is something that many groups have been working on, trying to capture in a more sensitive way what's going on in terms of the gait and the balance of these patients. So this might be something that we need to see more detail, might be another interesting way to pursue the investigation, I think.
Now we have a bit of more questions. We have 3 people that already raised their hands. So we're gonna, I ask you to give really fast questions. Since we have the coffee break, we have time, but just for these 3, right? So Stefan first.
So quick question to Marcondes. Do you have any data from pre-symptomatic patients, MRI data?
Yeah, we do have SPG4 patients. We do have, our cohort might have like 10 patients or so. We do have only cross-sectional data at this point. But just to give you a quick insight, there is something going on. We're able to see differences in terms of the spinal cord, but also in the brain of these patients. So the corticospinal tracts, we had abnormal diffusion in these pre-symptomatic individuals. and something also in the corpus callosum. So there is something already there in these patients.
Again, pharmacokinetics, the most promising marker could be the one that is severely changed in severely affected patients. So there should be a big effect. So if you go for the severely affected disease group of patients, which are the most, where are the biggest changes where we can expect something?
Well, you know, again, we're still working on that. So this cohort that I have shown to you, so it's the SPG11 cohort, that would be a late stage and quite disabled patients. And I think, you know, diffusion definitely is the best outcome that we have based on this data. And I would definitely go to the either corticospinal tract or the corpus callosum diffusivity data. So in most of our results, I think FA and RD, which is a marker of demyelination, another marker, they are quite similar in terms of the behavior in terms of the clinical course. So in summary, I would go for diffusivity markers, especially FA, RD, looking at the corpus callosum and the corticospinal tracts.
Yeah, sorry, I'm Ralf Reimann from the Huntington field, actually. So I just would like to make a comment rather than a question, actually. Apologies for that, but I do think you guys have to really look carefully into like what you are looking for. I think there will be proof of concept markers, there will be target involvement markers to actually establish that your therapies are working, and that's very important. However, we've learned it the hard way. I mean, you have to kind of also always keep in mind that if you want to get these therapies to patients, in the end of the day, you have to convince regulators, and the feedback that we have gotten all along the way was sometimes quite surprising. So, for instance, imaging is fantastic for proof of concept, but at least in the Huntington field, the FDA won't buy imaging as a primary endpoint for a clinical trial for proof of concept of something delivering clinical meaningfulness. So I think we have to do a lot of convincing work, and we have to collect a lot of data. Maybe eventually we can convince them, but think about also what can be done in terms of actually showing clinical benefit in as small cohorts as possible. I think that's something we learned the hard way, I have to say.
All right, so with this advice, we can close the session, and thanks for the speakers. I invite you to the coffee break. Or in Boston, so I don't know if I did a mistake. I came back to okay.
Hello, everybody. I think we have 1.5 hours ahead of us, and then afterwards we have, I think, a wonderful dinner. Therefore, we have 5 talks left, and I got told that everybody needs to stick to timelines, and therefore we try hard. Respectively, the presenter try hard. First, we will hear from the poster session, a flash talk starting now more and more into clinics, and it's about clinical and molecular characterization of SPG7 patients carrying the specific P-alanine 510-valine mutation. The stage is for you. Please, go ahead.
Thank you. So good afternoon. It's a real pleasure to be here at this great conference. I would like to thank the organizer for giving me the opportunity to present our results on SPG7 patients. So it's already been introduced by the other colleagues. So just brief introduction. It's an autosomal recessive disease. The SPG7 encodes for paraplegia that it's protein of mitochondrial inner membrane. It's a metalloprotease, and it forms a complex with another important protein, the AFG3L2, that it's responsible for spinocerebellar ataxia type 28, and this complex is important for the regulation of the respiratory chain synthesis, for the protein activation, but also protein degradation and the homeostasis of calcium in mitochondria. About the clinics is a frequent cause of HSP, but also cerebellar ataxia. So in this figure, you can see our cohort in Paris is the second cause of recessive cerebellar ataxia after Friedreich ataxia, and is the first cause for recessive HSP. About the clinics, we conducted a collaborative study in 2019 with German colleagues, Belgium, Netherlands, Italian, to collect cases of SPG7 patients. So we collected 241 patients, and we characterized very well the clinic because we described a mean age at onset at 35 years old. Almost all patients presented a pyramidal syndrome, but in 65% of cases there is also cerebellar ataxia. Half of patients present also deep sensory loss, sphincter dysfunction, and there are other signs such as pescavus or muscle wasting that are quite frequent. These patients can present also optic nerve atrophy with or without diminished visual acuity. And about cognitive impairment, we reported last year a cohort of SPG7 patients, and cognitive impairment is also frequent because the definite cognitive cerebellar affective syndrome is present in 68% of patients. About imaging, the cerebellar atrophy is present in half of patients, and the degree of the severity of atrophy, you can see that is different from one patient to another. And in this cohort, the most frequent pathogenic variant was alanine 510 valine. This variant was widely debated about its pathogenicity because it's very frequent in general population, up to 1% in European population, and rising to 3–4% in UK. But now is established its pathogenicity, and we show that the presence of this variant is more associated with cerebellar ataxia onset and also with a latest onset comparative to other variants that present more spasticity. But in this study, we didn't distinguish from homozygous for alanine 510 valine compared to compound heterozygous. And so the aim of this study that I presented today is exploring SPG7 patients that carrying at least one alanine 510 valine, and we included all patients from our course of SPATAX and BioMove course in Paris, and also performed proteomics and functional studies. So we collected with Charlotte Moreau, a PhD student in our lab, 95 SPG7 patients. 29 homozygous for this variant, 66 compound heterozygous, so 18 with a missense variant and 48 with a loss of function. You can see that the variants are distributed throughout the entire protein. There are also some deletions. And about the clinics, so the age at onset differed significantly across group, being earliest in alpha 510 valin missense patients with a median of 35 years, intermediate in patients with a loss of function with 38 years old, and was the latest in homozygous patients with 42 years old. About the clinics, there is no special difference in among these three groups. Of course, the age examination was higher in homozygous group, but the severity of disease was quite similar with a SARA score of about 12, a disability score of 3, that means that the walking is impaired but possible without assistance. And the group with loss of function variant presented more spasticity, as already reported, and more cognitive impairment. And the cerebellar atrophy was most present in missense and homozygous group. And also, in collaboration with Claire Pujol at the Institut Pasteur, we assessed some functional studies. So we collected fibroblasts from 3 controls, 1 patient with alanine 510 valin homozygous, 4 compound heterozygous, and 1 patient with another variant, the arginine 585 to glutamine 587 deletion at homozygous state. And so we assessed firstly the morphology of mitochondria. So in these merged fluorescent images, we can see in blue the nucleus, and in yellow the staining for the mitochondria. And there are preliminary results. It seems that there is difference in mitochondrial density, especially for patients with homozygous variant and other SPG7 variants, but there is no evident fragmentation or tubulation. And we check also the activity of mitochondria. We performed the SEORS assay. SEORS assay is an assay that can measure the oxygen consumption rate at basal respiration, but also maximal respiration after treatment with FCCP, that is a mitochondrial uncoupler that collapses the gradient of the proton. And we can see that there is no difference for the basal respiration between control and SPG7 patients. And for the maximal respiration, it seems that the patients have more consumption of oxygen, but of course we cannot do any statistical analysis since the sample size is restricted. And after we check also the level of mitochondrial protein. So in Western blot we can see the different proteins that are involved in the 5 complex of the electron transport chain responsible for oxidative phosphorylation, so there is no difference in the level of this protein. We checked also MRPL44, that is a protein for the translation, for the synthesis of the protein, and also SOD2, but there is no evident difference in the level of this mitochondrial protein. And I present some preliminary data for proteomics. So the proteomic was performed by chromatography on PBMC from 7 SPG7 patients and 11 controls. We can see that there is a clear discrimination between control in the right part of the figure and SPG7 on the left. And when analyze the gene set enrichment, that is analysis rather than classical gene ontology, but it considered the global expression changes of all proteins that are in the dataset, so the proportion of genes that are up or down regulated in a within each pathway. So we can see that the pathways that are downregulated mainly involve the peptide chain elongation, the translation elongation, translation termination, mRNA splicing, the quality control for mRNA quality. And so there are all pathways that are interconnected, and are important for the synthesis of the protein. Among the upregulated pathways, we can find the mitochondrial protein degradation, the metabolism of fatty acid or lipids, the aerobic respiration. But these analyses are still ongoing. And also we check if there is any difference for in SPG7 group between homozygous for alanine 510 valine and compound heterozygous, but you can see in the volcano plot there is no difference between the two groups. So in conclusion, we can say that there is the patients carrying homozygous ALA 510 valine present a later disease onset compared to the other patients. There are a similar phenotype in this cohort with more cognitive impairment and spasticity in group with the loss of function variant. About mitochondria, the activity seems preserved, and also the level of the protein seems preserved, but of course we are in the fibroblast, so it are not the neurons. For proteomic, the preliminary data results reveal that there are some differences between patients and control, but no genotype-dependent difference in this cohort. So I would like to thank all the lab at ICM, in particular Charlotte Moreau, and the collaborator at Institut Pasteur, Claire Pujol and Jean Tourbeau, the Bank of DNA, and thank you for your attention.
Thanks a lot for the deep phenotyping and really great work. I think we discussed it also at the end, similar to the other sessions. And we will continue now with an electrophysiological approach by video oculographic data in patients with genetically confirmed pure versus complicated HSP. And Ludmila Novotná from Prague will present her data in her study.
Hello. Thank you for the opportunity to present our preliminary video oculographic data in patients with pure versus complicated HSP. Allow me to make a brief introduction to the eye movement and eye movement abnormalities. Vision is a dominant sensory modality in humans, so there's no surprise that there is a large portion of the brain that is dedicated to controlling eye movements. These eye movements include gaze-shifting movements, which are primarily mediated by saccades, and gaze-holding eye movements, which include fixation, smooth pursuit, VOR, or optokinetic reflex. These eye movements are mediated by several systems, including cortical systems such as the frontal eye field, mediotemporal regions, or the prefrontal cortex, several brainstem regions, cerebellum, and basal ganglia. So the next question is, why even assess eye movements in patients with hereditary spastic paraplegias? Oculomotor abnormalities are frequently reported in movement disorders and are reported in some subtypes of HSP, such as SPG7, as you might have heard in the previous presentations. But there have been, up to this date, very few systematic studies. I would like to highlight one of them that showed sloped vertical saccades as a hallmark of hereditary spastic paraplegia type 7 in 5 patients, but there is a lack of other studies in other subtypes in patients with HSP. So we had our research question. We wanted to know if there are other subtypes of HSP that could have prominent or even subclinical oculomotor abnormalities, and if those would be subtypes with primarily cerebellar involvement, or if there would be other subtypes without it that would show subclinical oculomotor abnormalities. Also, the video oculographic data could present a future objective digital biomarkers for assessing phenotype in patients, and even for some longitudinal studies if we could prove there could be some abnormalities. So here's our research question. We wanted to evaluate the frequency and types of oculomotor abnormalities in patients with HSP. And we wanted to compare their occurrence between patients with pure and complex forms of HSP. Our main hypothesis was that there would be other subtypes of the complex forms of HSP that would have oculomotor abnormalities, and that the pure HSP subtypes would show no significant oculomotor abnormalities. So let me introduce our HSP VOG cohort. We had 20 patients with genetically confirmed HSP. Those were divided into 2 groups according to their genetic subtype and also according to the clinical phenotype into the pure and complicated or complex HSP group. Our groups were pretty well balanced. We had 9 patients with pure HSP and 11 with complex HSP, 9 males versus 11 females. The median disease duration in the pure HSP group was significantly longer than the median disease duration in the complex group. The SPRS scores were pretty similar, but surprisingly were a bit lower in the complex HSP group. So here is our VOG protocol. We assessed several parameters during the examination. We tested for the presence of spontaneous and gaze-evoked nystagmus, the smoothness of horizontal and vertical smooth pursuit, and the accuracy of horizontal and vertical saccades in our patients. We also tested the elicitation of physiological optokinetic reflex in our patients, which was a more complex task, as for the physiological optokinetic reflex, intact smooth pursuit and also intact saccades are needed. We also added a more complex task, which has a cognitive component. We tested the anti-saccades. How we did it, you can see on the small black picture underneath the saccades. The patient had to look at this red dot on the screen, and there in the regular intervals there would appear a second dot, and the patient had to look to the other side of the screen, so he had to make the choice to look to the other side. The optokinetic nystagmus you can see in the second picture, which is a reflex that complements a vestibulo-ocular reflex at lower frequencies and requires a constant stimulus, which in this case was moving blue and yellow stripes. So before we delve into our results, I wanted to show you some examples of our patients in the VOG cohort and their eye movement abnormalities. The first patient was a female with SPG4 subtype that showed prominent abnormalities in the smooth pursuit. As you can see, the smooth pursuit is disturbed in the video, and you can also see that in the complementary picture underneath it, you can see the oscillations there where there should be a smooth line. The other patient was a patient with complex HSP that had an upbeat nystagmus that first started when she was pregnant, and after the pregnancy has significantly subsided, but was still present even on the clinical examination. This patient has showed prominent abnormalities in other oculomotor domains as well. Besides the upbeat nystagmus, she had prominent saccadic oscillations, and also the smooth pursuit was disturbed by this nystagmus. You can again see that on the supplementary pictures below. The first one is from a patient without any oculomotor abnormalities, and you can see that on the gaze up there are 2 straight lines that are basically similar, and on the other picture you can see that there are 2 lines and that there are these prominent oscillations that represent the upbeat nystagmus. So, without further ado, let's review our preliminary data. To our surprise, our first hypothesis was disproved because we found prominent oculomotor abnormalities even in some patients with the pure HSP, such as I've shown you in the previous slide. In both groups, we had patients with spontaneous nystagmus. With the second parameter, we had only one patient with pure HSP that showed gaze-evoked nystagmus and several patients with complex HSP. Surprisingly, saccades were the parameter where we found almost no abnormalities. There were no patients with abnormalities in the horizontal saccades, and only one patient that showed inaccurate vertical saccades. The most abnormalities were present in the horizontal and vertical smooth pursuit in both groups, PHSP and complex HSP. Also, surprisingly, there were many abnormalities in the optokinetic nystagmus, but only in the complex HSP group. In the pure HSP group, only one patient didn't have elicitable optokinetic nystagmus. But it is worth noting that even in the patients with elicited optokinetic nystagmus, there were subtle abnormalities such as lower gain of the optokinetic nystagmus, or the optokinetic nystagmus was only elicited at slower speeds of the visual stimuli. So, to interpret our results, surprisingly, we noted many abnormalities in many of the parameters, such as spontaneous and gaze-evoked nystagmus, and also abnormalities in the optokinetic nystagmus, which could have stemmed from abnormalities in the smooth pursuit. But surprisingly, we had almost no abnormalities in these saccades, as I've noted. I also wanted to note the anti-saccade task because we had a lot of patients that presented with incorrect anti-saccades. And we think that could be mainly due to a cognitive component, as a lot of our patients in this cohort showed a mild cognitive impairment in our cognitive screening. But we have to analyze these data further, and we want to correlate them with a further neuropsychological testing and its results. So, where is the problem? So far, we do not know, because we were surprised even by our data in the pure HSP group. Of course, it could be a subclinical cerebral involvement, or there could be a component of the cognition, as I've said, in the anti-saccades, or it could be a more complex disruption of the networks, because, as I've previously said, there are many anatomical structures and many networks that are included in the controlling of eye movements, and it is very hard to establish where exactly the problem is. And we also think that in certain subtypes, the problems, of course, might be in different networks or in different structures. So, what do we know and what do we not know? Oculomotor abnormalities were present in both pure and complex HSP in our cohort, and abnormality in at least one of the evaluated parameters was present in almost 3/4 of the patients. But there are several important limitations to our study, and the main one is the small sample size of the cohort, which was only 20 patients. So in the future, we would need a much larger cohort and also a healthy control group or an ataxia group, which could be interesting to compare these results to. Also, another important problem is the heterogeneity of HSP. We decided to divide those groups into pure and complex HSPs, but this might be an oversimplification, especially in the complex group, as there were many different subtypes. Also important to note is that not all patients might be able to be examined by this method due to an age or other complicated factors such as ptosis, in which case you cannot really examine the patients. But there are also many future directions. We would like to do a longitudinal study in the future to see if there are any progression of the oculomotor abnormalities in our patients, and we would also like to focus more on the anti-saccades as well as an additional marker of an objective marker of the cognitive impairment in our patients. So in the end, I would like to thank you for your attention, and I would like to thank my team and also our patients, because without them, none of this would be possible. Thank you.
Thanks a lot for the nice functional data, which are really novel to the field, and I appreciate very much then later on the discussion. As we heard in the session prior to that, it's always important to have certain biomarkers, whether it's MRI imaging, whether it's clinical, certain findings. And I think one of the holy grails is to find a good fluidic biomarker. And the first choice, which is Darius Ebrahimi Fakhari, reports about blood-based biomarkers. And in particular, he leads us first to the pitfalls and later gives us opportunities. Please.
Thank you. Thank you very much for the kind introduction. Let me just quickly pull up my slides. Okay, well, thank you very much for the invitation, the opportunity to speak here today. It's a great meeting, and I already learned a lot, so thank you for this. I'm going to be talking about blood-based biomarkers in HSP, and I have to admit this is not a topic I usually talk about, so bear with me. I hope this will stimulate some thoughts and trigger some questions. So I'm presenting on behalf of our team and also our many collaborators. These are my disclosures. Before I go into the talk, I want to acknowledge that none of the work that you're going to see would be possible without the many families that participate in our studies, and as you'll see, also give us blood samples to work with. So I want to acknowledge that first. In the next 15 to 20 minutes or so, I'll briefly talk about 4 areas. First, I'll give a very brief overview of what the Spastic Paraplegia Centers of Excellence Research Network in the US is doing, what activities we have across the network. I'll then switch gears and talk about blood-based biomarkers and the critical unmet need to find markers that we can measure in blood. I'll then review some of the existing data on plasma neurofilament light chain in HSP and show some of the subtype specific differences. But most importantly, I'll mention emerging work on additional modalities, including next-generation sequencing-based proteomics, opportunities to collaborate, and I'll hopefully close with some future directions. So very briefly, Stefan introduced this this morning. We have a network in the United States that is coordinating research on HSP, has been coordinating research on HSP for the last 2 years, and is going now into the next phase. The overall mission of my group, but also the network, is to advance patient-centered translational research, in our case in childhood onset movement disorders such as HSP, by linking clinical insights to mechanistic discovery and therapeutic development, accelerating a path from molecular understanding to first-in-human studies, and eventually meaningful clinical impact. Now, this is a broad mission statement. We all know that the drug development pipeline looks something like that, and the more I've worked in this space, the more I realize that this piece of the puzzle, a biomarker, is really critical to advance therapeutics beyond proof of concept or first phase 1, phase 2 clinical trials into pivotal studies. So our work in this area started with a natural history study that was initially single site at Boston Children's Hospital about 5 years ago. At this point, we've seen over 700 families with early onset hereditary spastic paraplegia. You see the genotypes listed here. Many of the families have come back for a second, third, fourth, and now 5th visit. I won't comment on the videos very much, but I think the theme in today's sessions was the great clinical variability, and I think the videos showed that very nicely. You have symptoms that begin in early childhood, symptoms that begin in adolescence or young adulthood, and you have variable degrees of motor disability. Now, this natural history study, I won't go through any of this work in detail, but just to say that I think phenotypic expansion and gene discovery has been one of the areas we focused on. But now that we're in the longitudinal phase of this program and have had the support initially by the Spastic Paraplegia Foundation, now through the NIH to extend this to a multi-site study, we're really looking to develop this into a longitudinal study that is discovering sensitive biomarkers for clinical trials. So this is the network. We have 11 sites across the US, as you've seen, and broadly speaking, we have three main projects. The first is to describe the natural history across the age span. The second is to apply state-of-the-art genomics to unsolved cases. And the third is to develop sensitive biomarkers. One of my personal priorities is to align and harmonize this as much as possible with international cohorts. I've had great conversations. I've listed some countries here. I think if we all work together, I think there's tremendous opportunities. And blood-based biomarkers might be one of the first areas that we can work on together, given that we are already collecting blood samples in a very similar way. So what have we done so far? We routinely collect at every visit DNA, RNA, this is at the initial visit, and then longitudinally plasma, serum, dry blood spots, and in some cases fibroblasts. You see on the right from our first monocentric study, we have just over 200 blood samples collected, plasma samples collected, and we're collecting follow-up samples going forward. We've used these samples in a number of ways, but I also want to point out that this is a resource. We are happy to share these samples with anyone who wants to work on this. We and others have worked on mainly looking at neurofilament light chain, as I'll show in the next couple of slides. And using the fibroblast biobank, we've developed some functional assays as well. But I think there's a lot more work that we can do, so ideas are very, very much welcome. So, I was tasked to look at biomarkers, blood-based biomarkers for HSP, and much of the literature has concentrated on neurofilament light chain. And I'll present some of the work. This is work that is looking to develop biomarkers for clinical trial readiness. As we all know, there's different types of biomarkers. Really, I think where we need to invest our time and resources is to develop monitoring biomarkers and surrogate endpoints, so markers that we can use for clinical trials that teach us about outcomes at the shortest period of time possible. Now, one of our fellows, Dr. Habiba Agyanda, has put together this very nice table summarizing all the published work on neurofilament light chain from various groups, including groups here in the room. I think in a sort of a summary, it's fair to say that most of these cohorts are relatively small. They're all cross-sectional, and they mostly use the Samoa platform and plasma. I'll go through a couple of disorders in sequence now. I'll start with SPG4. Julian has presented some of this work already. There's a nice body of work from the Tübingen group looking at a larger cohort of individuals with SPG4. You see that there is a statistically significant difference, but the difference is quite modest, I would argue. And as you can see here with age, and this is mostly, it's predominantly an adult cohort, the two curves of controls and patients are quite close to each other. So NFL does not seem to be a very strong discriminating marker in SPG4, with maybe the exception of the very young patients, Julian showed us earlier today, who have very early severe disease. So my sort of summary take, I won't go through the statistics and the many limitations here, but my overarching take here was that there's a modest elevation in serum or plasma NFL in individuals mostly with early disease onset and severe disease. And I think you'll see that as we go through the different forms of HSP that have been investigated for neurofilament light chain levels, this continues to be a theme. Now, what about SPG 11 and 15? Clinically very different from my experience. There's much more rapid disease progression following the onset of motor symptoms compared to SPG4. This is work done by our group here looking at SPG 11 and 15. You see that here the difference is much more clear between age-matched controls and our cohort of patients. There's no difference between SPG 11 and 15. They're very similar. And that difference in absolute level compared to matched controls is the greatest the younger the patients are. So in our mind, the earlier or the closer they are to their initial manifestation and this initial often rapid disease progression before there's sort of a little bit of a plateau and a slowdown in disease progression. But again, this remains to be shown in longitudinal studies. This is purely cross-sectional, or mostly cross-sectional. Similar findings made by our colleagues here from Erlangen. SPG11 tends to have higher levels than other forms of HSP, including SPG4 and 7. When we looked at the few longitudinal samples we had collected at that point in time in both SPG11 and 15, we found what I just described. So the levels are higher the closer you are to disease onset, and then tend to sort of trail off. But if you look at the graph on the right-hand side, really it's all over the place. Some go up, some go down. So it's really hard to make a pattern out of this. So my sort of, again, 50,000-foot view here is that there's a moderate elevation of plasma NFL, but mostly early into the disease. And again, we have to see how this evolves when we have more longitudinal samples we can look at. Now, switching gears from these the common dominant and recessive forms to the more rare recessive forms, and certainly disease that is more neurodevelopmental and neurodegenerative compared to the more progressive neurodegenerative types. This is AP4-related HSP. This is some work that we published a few years ago. Again, modest elevation, statistically significant. You see the area under the curve is actually not so bad at 0.87, but again, a modest elevation. And at the time, we didn't have a lot of longitudinal samples. We did look at GFAP as well, as you can see, as a marker for astrocytes, and did not find any change there. So our take was, again, modest elevation in plasma NFL, but not in GFAP. And then when we looked a little bit more carefully, we see what we saw for the other forms as well, that you mostly see that elevated plasma NFL in individuals who have symptoms suggestive of more severe disease, such as, for example, epilepsy or a GMFCS level of 4 or greater, so higher motor disability. One disease that we have a recent interest in, mainly because of our colleagues' work at Boston Children's Hospital in developing treatments, including ASO-based treatments for this disease, is KIF1A-related disorder, which has an overlap with HSP. Habiba from our group took this cohort of 46 patients that we have in a longitudinal study. We had access to matched controls, so these are both age and sex matched controls, mostly children in this cohort, and looked at NFL. And I think we had this, I think Alexander mentioned this morning, I would have bet my year's salary that the level would be sky high, and it was not. As you can see here, the NFL levels are very modestly elevated to the point where I don't think it's clinically meaningful. I thought this is the one disease where we're going to see a high NFL level, but we did not see it. And that is surprising, and I am not quite sure yet if this is a clinically meaningful marker in this disease. You can see here there's no real correlation with age as a surrogate for disease duration, as well as with SPRS scores. There is a very modest correlation there. So, yeah, really, I think surprising in many ways, given how progressive and degenerative this disease appears clinically, but that's what we found. Here, this is just some more data to say that there's only modest correlation with disability, but no correlation with spasticity in KIF1A-related disorder. So all in all, you can, I think, look at this body of work and say, okay, there's some interesting findings here. My takeaway, again, 50,000-foot view, is that we need more longitudinal data from the same individuals to really make sense whether neurofilament light chain has a dynamic that is useful as a biomarker. And I think certainly that's something that we're going to look at, but I think we need to look beyond that. And a lot of the inspiration for this work comes from other neurodegenerative diseases. I'm not an expert in this area, but again, our group has sort of developed some expertise in this area. We are looking at more broad-based ways of assessing changes of blood-based proteins that have been linked to neurodegeneration. This is taken from a recent review paper that I can recommend that looks at different platforms. Broadly speaking, there are 4 platforms. Our work has concentrated on the so-called Olink platform. We have more recent work on the new LISA platform. And then there's, of course, also both label-free or labeled mass spectrometry-based methods, and SomaScan is another platform that we have no experience with, but is also available. All of these platforms have been looked at in different neurodegenerative diseases and Alzheimer's disease, ALS, so others in the room might have more experience in that area, but we started with Olink, thinking that this is a good way of expanding our portfolio. Again, this is very recent work, ongoing work, looking at 3 subtypes of HSP, AP4-related HSP, SPG4, and SPG11 and 15. We have the advantage that we have, again, age and sex match control, so we can match every patient to a healthy control, regardless of age. And we ran a new panel in collaboration with Olink or Thermo Fisher that is looking at 48 neurodegenerative disease-associated proteins. The mechanism or the method is a little bit different here. This is an antibody-based detection, and the readout is next-generation sequencing or PCR-based, depending on which platform you use. Again, very simple principle. You have 2 specific antibodies per protein. They have little oligos that complement, and then there is an amplification step. And with this method, you have very high sensitivity, so you can detect proteins at a femtogram level, so sub-picogram level. And most importantly, you can measure them quantitatively, not only in relative or qualitative terms, but quantitatively. And we thought that was a good platform. We were one of the first users of this new panel. These are the 48 proteins that you can see here. A lot of it is informed, of course, by Alzheimer's disease and Parkinson's disease, not a major neurodegenerative diseases. But we felt there were enough interesting proteins here that it would be worthwhile to link this with HSP. And I think one other factor that I was excited to see is that Olink or Thermo Fisher has also teamed up with the UK Biobank to run a very extensive panel of 5,400 proteins across the UK Biobank samples, which at this point is about 500,000 samples. This has just started last year. There are at least 15,000 individuals with neurodegenerative diseases, including 200 with motor neuron disease, and I hope some HSP patients are included in that category. But regardless, this will be a very valuable reference data set for all of us to work with. So this motivated us to use this technology. I don't have any final results to show you, but just to share that we're doing the first experiments now. We're understanding how the technology works and how proteins cluster. We have found a couple of proteins that are differentially expressed. We found neurofilament light chain elevated, similar to what we had seen with the Samoa assay, so that was a good quality control. But we're still sort of looking at the proteins and are trying to make sense. So I have more to report, hopefully, in a couple of months. I want to share this more as sort of a new platform and one that we can all work on together if we share samples. This is again from the same review paper looking at what has been learned in Alzheimer's, FTD, and Parkinson's disease. And I think we need to do something very similar for HSP. And I would really like to focus our time and resources to add HSP to this map to really learn which proteins are differentially expressed in blood in various subtypes of HSP and how they change over time. So this is more a call for collaboration. In summary, I hope I was able to show you that blood-based biomarkers are indeed a critical unmet need in the field of HSP. We have a lot to learn from other fields. Development of sensitive biomarkers is a priority for our network. Plasma neurofilament light chain is variably elevated, so really difficult to make sense of at this present moment. There are some really exciting NGS-based proteomics platforms that have very good analytical sensitivity and specificity, so offer real opportunities for us to look at a larger number of proteins. And there are tremendous opportunities for collaborative projects linking blood-based biomarkers and detailed natural history data. So again, with this, I have to thank my team and the many collaborators that contribute to this and the funding that we had to pursue this pilot study from various sources, including EuroHSP. So thank you for that, and I look forward to your questions.
Thanks a lot for this beautiful insight into the proteomic signature of HSP, and I think we will have definitely some questions at the end of the session. More relevant to clinics is patient-related outcomes, and I think if you're talking about patient-related outcomes, it's always important to ask actually patients what matters to them, what bothers them, what kind of pain they have and what kind of impact in daily life. And I think Mirinda Artiemi from Heidelberg will manage to address that point, patient priorities, what matters and why. We need to change your name.
Thank you.
Perfect. Hello, everyone, and thank you for the invitation. I'm a clinician at training in Heidelberg, working with Rebecca Schuler, and I'm really happy to be contributing to today's session by presenting our results from a large cohort study that included about more than 600 patients, really asking important questions about what health impacts matter to them. And by health impacts, that's an umbrella term sort of encompassing symptoms as well as health experiences. And really the question behind all of this, and something that we have to keep in mind as we move in the direction of interventional trials, is if we want to, so what do we want to measure in clinical trials if we want those measures to reflect something that's of benefit and meaningful to patients themselves? And we've had a really good background, so I've spared all of you that, but I wanted to include one slide and remind all of us what key challenges in developing good outcome measures in HSP currently are. So first of all, HSPs are clinically very heterogeneous, so symptoms range from spasticity to ataxia. There are a lot of genotypes. Second of all, it's a slowly progressing disease, so it's very difficult to detect meaningful change, especially in the time frame, in a short time frame such as clinical trials. And last but not least, patient cohorts are small, and that makes designing good trials complicated. And these are really challenges that a lot of rare diseases face. And about 3 years ago, there was a review that kind of looked at the measurement landscape, and they picked up on the fact that they discovered more than 80 different outcome measures were used in different HSP clinical trials, or trials in general, emphasizing the variability and the fact that we lack a consensus when it comes to developing optimal trial endpoints. So current scales, such as the Spastic Paraplegia Rating Scale or FAST-based measures, they've been shown to kind of be good at detecting disease severity or progression. But one thing that's not really clear and that really needs to be addressed is, does whatever they measure really overlap with what patients experience? And this is a key question, especially when you look at what regulatory agencies, such as the FDA or EMA, are asking us to do when designing good trials. So this takes us to the study aim of this project, which was trying to identify patient-prioritized health domains that would be relevant in a context of a clinical trial. This was done together with the ProSpecs Consortium. So as a disclaimer, the study included patients with HSP, spastic ataxia, and ataxia. I'm only presenting results related to the HSP SPAX group, and I know that someone from Matis Synoptics group is currently working on the ataxia results. So what did they do? So together with the ProSpecs Consortium and PCOM working groups, so patient-focused outcome groups, which included patient representatives, patient partners, and investigators, they designed two surveys that were staged. They were translated into 6 different languages, and they were basically spread or collecting answers across several countries. So that included lots of centers in Europe and North America collaborating together and thereby collecting anonymous data. Survey 1 was designed to capture sort of a breadth of symptoms, and about a year later, Survey 2 followed up after preliminary analysis of Survey 2 being kind of a refinement step because the number of health impacts was reduced to 36, and participants were asked to rate severity and relevance to their daily life of those impacts. Both surveys included information on demographics such as age, mobility-related questions, in order to get like an idea of the disease stage they were, genotype, and the diagnosis. Proxy responses were also allowed. And if you think about that, so there were 2 surveys spread across, translated into so many languages and spread across different countries, that resulted in a bunch of data. And we had to think about coming up with a way that meant we could process the information in a way that was clinically meaningful, but also kind of coherent with what regulatory bodies ask us to. And when we think of a concept in the context of capturing treatment responses in clinical trials, we thought the following criteria would really make sense. So health impacts, we looked at their prevalence because we wanted to make sure that these symptoms were frequently enough in the target population. We were also interested in health impacts that were sensitive to change, so they would change with disease progression, meaning they would be able to capture longitudinal change. Thirdly, obviously, they have to be relevant to patients. And last but not least, we also looked at correlation between severity and relevance because we thought it was also important that a higher severity of a symptom kind of also translated to a higher relevance in everyday life. And that leads us to the demographics of the survey population. Survey 1 collected over 1,100 responses, of which about 600 were HSP or spastic ataxia, and about a third of these were SPACs only. Survey 2 collected over 800 responses, and of these, there were about 500 with HSP, and these encompassed 178 of the SPAC subgroup. Patients were stratified into different disease stages, including mild, intermediate, and advanced. So we collected information in different languages, but obviously, as you can see by this table, most answers were given using the English form and the German one. And in terms of genotypes, in both studies, as is sort of reported in the literature, SPG4 patients were the most common one, followed by SPG7 and RSACs. And there were, obviously, due to the nature of the disease, there were about roughly 30% reporting that and the genotype was unknown. So we first, wanted to look at prevalence and really asking the question, how often do these symptoms occur? And what you see here is a spider plot where we try to group these items into different clinical domains so that the information itself would be easier processable by someone looking at the graph. And if you look at this, it immediately, comes out that items, so the dots that are highlighted in light green, that are mobility and posture related, are very, very common. So these are the most common symptoms. If you name them, the top ones are reported by patients are reduced walking speed, the use of a handrail that's needed when walking up and down stairs, increased exertion while walking, reduced walking distance, and balance issues while walking. Another domain that's a clinical domain that's sort of also very prominent is lower body function, so that's anything to do with the legs, leg weakness, muscle stiffness, correlating to spasticity, lower limb contractures, or muscle wasting. But at the same time as something, this is something that as a clinician or someone working with HSP isn't really a surprise. You see that other non-motor symptoms were also quite frequent, of which sort of fatigue, sticks out, as well as reduced ability to work, which is sort of a concurrent theme, and bladder incontinence. And there were some health impacts that were not very frequently reported, and those were taken out sort of in the next step of survey 2 when the refinement happened. And this takes me, so as we looked at the general cohort, we were thinking about, okay, are there differences when it comes to different disease stages? And I don't want you to be overwhelmed by these 3 graphs, because obviously we don't want to get lost in the details, but if you look at them step by step, if you focus at the mild stage, okay, so roughly the way the items are numbered and labeled is pretty much exactly the same as the colorful spider plot that was there before. So you see in the mild stage, it's mainly mobility and lower limb function related items that are very frequent. And then as you move to the intermediate stage, those are still very frequent, they actually increase. But then at the same time in the intermediate stage, you see that other items start to occur or increase in frequency, those related to pain, autonomic dysregulation, or perhaps sensory function. And when you then look at the very last graph here, the advanced stage, you can see that all of a sudden, mobility is not as prominent as anymore. And if you have to keep in mind that advanced stage people are all wheelchair bound. So obviously at this point, whether your walking speed is reduced doesn't really, it's not very relevant anymore. But at the same time, a lot of the other clinical domains increase. And when you look at different patterns, and I think the key takeaway message from this slide really is when it comes to selecting outcome measures, you really need to be careful about what your target population is, and your endpoints need to be exactly targeted to what you're looking at. We also asked, obviously, patients about how relevant health impacts were to them. So we looked at individuals that reported having a symptom, and among those that were symptomatic, we asked them to rate relevance. So this is data from survey 2, and you have, so the different bars represent different items, and the colors stand for the different options you could pick, for instance, ranging from no effect to having a very severe effect on your daily life, emotionally as well as physically. And we tried to rank these symptoms by having a at least moderate effect. And if you look at these list of health impacts, sort of the top bit is kind of similar overlapping with what we saw in the frequency plot, where reduced walking speed is very important to people, reduced walking distance is sort of an issue, leg weakness, balance while walking, so anything that's sort of ambulation related. But also at the very top, among these ambulation related items, you see impaired ability to work. That's actually very highly ranked by people, and it really matters to them because it compromises their daily life. Other highly ranked non-motor related impacts include symptoms that are related to autonomic dysfunction, so urgency to empty the bladder, urine leakage, but also other aspects of life, such as difficulties with social interacting, in social life, with social activities, impaired sexual function, or fatigue in general. We were interested in, obviously we collected a lot of answers to the surveys, and we were thinking about whether there's any underlying pattern of how questions or symptoms are picked. So we used exploratory factor analysis to try to see if there were any clusters occurring, and that kind of sort of is a good way of representing or representative of the cohort themselves, because there was one factor that occurred that was ataxia. So basically most most of the patients that fall into spastic ataxia will be represented by this cluster. There was another cluster that had items related to pain and sensory. There's always obviously also a mobility cluster. Another cluster which we chose to sort of name psychosocial functioning because it had different items related to anxiety, fatigue, cognitive impairment, impaired ability to work, as well as an autonomic dysregulation factor. And one thing that kind of struck to us was, as we were looking at sort of what items were kind of associated with each other, you can see that in the psychosocial functioning group, impaired ability sort of associated with them, which kind of makes you wonder whether it is more other aspects than mobility limiting you when you interact in life and work. And this kind of leads me to one of the final figures, which I think has a lot of interesting information. So this is a dot plot where all the health items were plotted. You have frequency of the symptom on the x-axis, and you have the relevance of a symptom rated by at least being at least moderate on the y-axis. The dot sizes kind of reflect correlation, so I didn't mention this, but obviously we also looked at correlation between severity and relevance, and they were pretty high, so that was quite good. And if you look at this graph, obviously you start wonder, actually, those items that are located in the top right corner are the ones that kind of we are interested in terms of clinical trials. And if you look at that, you end up with having sort of 5 main factors that kind of evolve and seem to be very important to patients. One of them is anything related to sort of walking and ambulation. The other one is lower body function, so things such as weakness in legs, reduced muscle mass, or contractures in lower limb joints. Then you have sort of psychosocial aspects such as feeling tired, lack of confidence when sort of engaging in something new, impaired ability to work, difficulties with social activities. There's also pain that's kind of prominent, as well as autonomic dysregulation, anything that has to do with bladder issues mainly. And when you look at this plot here, some of you might have noticed that some circles are highlighted by another red, some dots are highlighted by another red circle around them. And these are impacts that basically emerge in the mild disease stage when you stratify those. And as I said, we stratified people by HSP predominant and having spastic ataxia, so you may be interested in, okay, what does it look like in the SPAC subgroup? Actually, it's very, very similar. It's not that different as you would expect. So you have, of all the dots, obviously, you have a slight shift to the right in terms of frequency, so things such as slurred speech or problems with like hands, they will be more frequent in the total group. But actually the domains that seem important overlap with the general HSP cohort. And this takes me to the summary. I basically presented to you results from one of the the largest patient-reported data set of HSP patients to date. This, importantly, was co-developed with patient representatives. We tried to make it accessible by translating it to different languages. The context of use of the analysis was obviously outcome selection for clinical trials, interventional trials, and we discovered that there were 5 core domains, main themes such as mobility, lower body function, autonomic dysregulation, pain, and social participation. And you could see that mobility sort of remained central throughout, several steps of our analysis, but other non-motor impacts were also important. And importantly, something that really needs to be kept in mind as you see a ceiling effect with mobility-related items. And last but not least, impaired ability to work clusters more with psychosocial functioning than mobility. So then you might wonder, what do I use this information for? So I included one last slide as sort of brainstorming ideas of what this data could be used for. Obviously, there's a lot of tables and things I didn't include, but it's a lot of raw data, and you could go about, okay, we would like to develop an HSP-specific PROM. So it could provide empirical data for item generation and domain selection, for instance. Another context of use could be, what about refining existing outcome measures? And there's still, a lot that needs to be done so that we reach trial readiness in HSP. A third thing I came up with was a digital mobility assessment, so it could help, the list of our results could help with prioritization of endpoints to focus. And in general, it could help also people who are working on developing therapies on what kind of aspects of the disease they should focus on. And with that, I'd like to thank you for your attention. Thank you to the Tom Wahrlich Foundation for the invitation. There were a lot of people obviously involved in this work without whom all of this would have not been possible. Thanks to Rebecca's, my supervisor, and most importantly to all the patients and patient organizations that really made this possible.
Thanks a lot for the great overview, and I think there are so many data that I think the evening is not sufficient to cover it. We need to move on, otherwise we will have problems with the housekeeping. I think we are now, after talking about patient-related outcome measures, it's always important to have certain scales which help mostly the clinician to reflect disease onset and or progression, and in addition maybe therapeutic response. And the talk will be by Jonas Morales, and it's about the spastic paraplegia rating scale and its possibility to modify it. Please.
So thanks for the kind introduction, and also for the organizing committee to invite me here. I'm going to talk to you about the most widely used clinical scale or clinician-rated outcome measure in the HSP field, the spastic paraplegia rating scale. So I think most of you, clinicians or people that works in the lab, knows SPRS, and this scale was developed under the leadership of Professor Scholz and Rebecca Schuller in 2006. And in 2006, the scale could be considered, I would say, well-validated. So like all the parameters we need to say that this was a good scale, we can say that it was met. They decided that should be focused on the disease severity of HSP, although this is an abstract concept, so right now we know that we need to define it a bit better. It was also related mainly to pure forms of HSP. It was reliable, has internal consistency, and was also correlated with some other scales that were already available at that time. So it was a validated scale in 2006 when they published it. However, the field moved, and it was not enough anymore to have like this formal validation. So the idea here is not just to have a validation of a scale, but idea that we can use that given scale as an endpoint for a trial. And for that, it was not so clear. It becomes more clear in 2022 when there was these FDA guidances telling us a bit better how regulators think about these concepts to say, oh, this scale can approve a given drug that might have efficacy for any given HSP. And then this guidance that is also followed by EMA established that we can have like this kind of fit-for-purpose clinical outcome measures. And the center here is that the patient's perspective is the most important one. So the thing is, the first step is what is more relevant to patients, as Melinda just presented to us. So we have this data right now, but in 2006 this was not available. We should define which is the concept that we want to use the scale, so the concept of interest, and also in which context we want to use, let's say for all disease stages, just for early disease. So this is something important when we're going to decide which kind of clinical outcome assessments we're gonna use. And then, depending on what we want to, like which concept and for which population, we might choose a clinician-rated outcome measure, which is, an example is SPRS. We can use a patient-reported outcome. We can use a performance outcome measure, or even a digital biomarker or a digital health technology. And so here there is a guidance on how to build this pathway for a scale become an endpoint for a trial. And this was not clear in 2006. So should we just throw away SPRS and do it again? So the answer is no. So we have a good scale. It's the most widely used scale across the field. So this is an advantage. And we can say, if it's a good instrument, perhaps you can just modify that instrument in a way that it could be fit for purpose somehow and validated for use for these regulatory agencies in a trial context. So there is different ways on how can we improve already existing clinical outcome assessments. So perhaps in my mind I can say, oh, I do SPRS a lot, and I do the same way as Marcondes is doing there, or the same way as Filippo. But if we pick the scale, we're going to see that the information is quite concise, and I might not do the same as they are doing. So it's quite important to provide really clear instructions and also to provide some training materials in a way that we have like really low sensitivity to change of the scales. We have a lot of discussions today about low sensitivity to change in biomarkers and also clinical scales. And we need to reduce also the variability of clinicians, especially in multicentric studies. So one key point here is really try to do in the most similar way the scale. Also some kind of wording or way of describing the instrument could be changed. The order of the item, so when you show some order, you should do exactly from item 1 to 13 in this order, but perhaps patient might become fatigued if you started with one or the other, so we can also change in this regard. There are some items of SPRS, like the items ones regarding to walking distance, and also the items regarding to pain and bladder dysfunction, that are somehow a PRO, so we ask the patients. And it was not clear the recall period, so we should really define what is the recall period to ask to patients. And also the idea that, if possible, we should have like a broader application and also including children. So can we also change a bit so children can also be included in the original version? It was mainly for adults. So the starting point for the modification of SPRS was mainly based on expert opinion, right? So what we did, and many of you here in this room participate in that, was a Delphi survey. So we have a first version of the modified SPRS, and in that moment we have two scales, the modified SPRS and also the MIDI SPRS that was intended for children from 5 to 16 years old. So we have sent invitations across the globe. We have a good representation. Most continents have some response for specialists, mostly physicians, but also physical therapists, and we have some people that are quite experienced with the scale and with the disease, and also some novice. And it was important to have novice because we want to be clear, even for someone that is not quite into the field yet. And the response we have was that most items regarding the instructions, the score level, and also additional information was good. But we have a lot of individual comments, so it was good, but it was not enough. Then we have an in-person discussion in the Treat HSP meeting in the end of 2022 in Tübingen. And there we have discussed this round 1 of the Delphi, and we agreed that, so it's a clean row. The concept of interest was regarding lower limb function, so not just the whole disease severity, but mainly the lower limb function. And the context of use would be in symptomatic HSP patients across all stages. It was told to be mainly for adults, but there is advice from the FDA, whenever you can use the same outcome measure across the ages, this is better. And there was like really mild difference from SPRS to SPRS MIDI. So the idea was to merge both scales in a single one. And now it's not just adults, right? It's from 5 years old and patients older than that. Then we have another version of but now just as a modified SPRS with MIDI incorporated in the scale, and we did the round 3. Again, mostly the same people were invited. We have similar results, a bit better agreement with the items, but still some comments. And just giving an example of what were the comments. So it's possible to allow for a second try in a case of unforeseen event like a fall, twist. So the idea here is much more related, like the comments, that we need perhaps a manual or a more detailed information in a way that we are doing in the same way the scale. So here you will not be able to read it, but there is like this final version of the actual version of the modified SPRS, and in the right side we can see the scale items. And in the left side, we can see the instructions, and also if the patient will need some kind of walking aids to perform that given item. And now I would like to show you, so what we have built was a video manual. So there is like a training, start of the training tool for SPRS, and likely this will be the first time that this video is being shown. So I have 2 videos to show you, so you can see what's the kind of information we are providing, and then I will continue explaining a bit more. So let's listen and watch to the videos. Modified spastic paraplegia rating scale intended for application in children age 5 and older, adolescents and adults with HSP. For some items, contextual information is added, like the use of walking aids and concomitant medication. Note factors unrelated to HSP that may have influenced the rating, like depression, lack of motivation in small children, and recent injury. Perform evaluation in a well-rested patient. Take breaks when necessary. Assess items in the order presented in the scale. Performance items 2 to 4, may be performed before or after the remaining items if this fits the clinical workflow better, provided sufficient rest is allowed. Rate what you see, unless instructions specifically demands otherwise. Prepare the settings before you start. The required items are a chair with armrests, stairs, examination bed, and a stopwatch. You may also need to provide different kinds of walking aids, step or box to adjust examiner's positioning during the tests. And then just to show you some of the items.
Items 2 and 3 assess gait quality and gait speed in the same trial. The total distance is 10 meters, including 1 turn, 5 meters forward, a 180-degree turn, and 5 meters back to the start line. Clearly mark the starting and turning lines on the floor. Use a stopwatch to record the absolute time until the first foot crosses the finish line. Provide the standardized instruction conveying the following meaning. The patient should move as fast as possible without risking a fall. Running is allowed if they feel able to do so. The patient should wear comfortable shoes and use their usual walking aid or orthosis, the one most commonly used in everyday life. Observe and rate quality and speed during the same trial. Do not repeat the test separately. Typical pitfalls to watch for are illustrated in the following short video clips. The patient is positioned incorrectly, with a walker or walking aid placed at the start line instead of their feet. The patient should always begin the test with their feet exactly at the starting line. The turn is initiated when the walker or other walking aid reaches the turning point, rather than when the patient's feet do. Always cue the turn when the feet reach the line. The examiner stops the stopwatch too late, for example after the second foot crosses the line instead of the first. Timing should stop exactly when the first foot crosses the finish line. The patient touches the wall or uses it for support during the test. This indicates loss of control and should be rated as gait quality 3. The test is performed on a carpeted surface rather than a hard, even floor, which may affect walking speed and consistency. Additional frequent errors include talking while walking or running and stopping at the turn. These should be corrected before the final trial.
Items 4 and 5 assesses stair climbing ability in 2 separate trials. Item 4 for quality at a comfortable pace, and item 5 for speed, performed as fast as safely possible. Use a conventional staircase with more than 5 steps and a secure handrail. The patient may start from the bottom or the top. Both are acceptable as long as the turn occurs on a step, not on a landing. For item 4, quality, instruct. Please go up and down the stairs at a normal, comfortable pace. Keep your hand hovering just above the handrail and use it only if needed. Pay close attention to how much support the patient takes for the handrail. Climbing without alternating steps is acceptable. However, performing sideways, backwards, or touching the stairs with the hands should be rated as a 3. Using an aid or assistance from another person likewise reflects marked impairment and should be rated as a 3. Small children who cannot reach the handrail may be accompanied for gentle support. Depending on the level of support, please rate 1 or 2 in this case. After a short rest, proceed with item 5, speed. Instruct. Now, please go up and down the stairs as fast as you safely can. Hold on to the handrail at all times. Use of the handrail is mandatory. Start timing when the first foot leaves the landing and stop when both feet reach the last step. Common pitfalls include skipping steps during ascent or descent, not holding the handrail during the speed test, start the test in the wrong position, timing errors by the examiner, stop the stopwatch exactly when both feet reach the last step, neither too early nor too late. If any of these cases happens, provide the correct instructions to the patient and repeat the test. So the idea here is just to show you a bit the instructions that we have provided in this video, and likely some of you who perform SPRS might do it in a different way that we're showing the video. So it's quite important to have really the instructions in a way that we are doing it in the same way. So right now, the advance of this, the idea is to do a training tool of SPRS, and we're going to send message to the Twitch HSP and also IEXOM, which is the Brazilian network of HSPs, perhaps also with the SP CERN, in order to get videos from patients. So this video training is just with a health person and showing the instructions, but we will need now patients' videos in all the scores of this scale in order to really provide this training tool. The idea is to deliver something similar to SARA training tool, in which an individual can go in an online platform, watch the instructions, watch patient videos, and also have a certification, and later this could be used in a clinical trial context. We are still working on the consents because the idea is that this platform would be for free for academic use, but if it's used in a clinical trial context, then there would be some kind of cost related, and we are seeing if it's possible to do that and how the consent for the patients, considering that this would be sold, let's say, in this way to companies when this is going to be used for trials. So this is still ongoing. And just to finish this, so the idea is that we need to optimize SPRS in order that it could be used as an endpoint for a clinical trial. So this Delphi process and all the things I just showed to you is this first step related to the expert input, and I would say that we are close to an end of this part right now. There is also some work to do related to data-driven, so there is this item response theory modeling that can provide a different, more sensitive way of looking at SPRS. And also Melinda's data, we don't need necessarily to anchor the PRO with the scale, but we need to say, oh, the items of SPRS are really the most important for patients, the most relevant and the most frequent ones, and this would also lead to this agreement for the regulators. And while doing this modification and optimization of the scale, it's also needed to do some validation of this novel version. And I think Rebecca in the last TTHSP meeting last year showed you this, and you have agreed, the guys that are part of the TTHSP. in which there is an ongoing study for SPG4, 11, and 15, and also for pediatric cohort. The idea is to validate different kind of clinical outcome measures, mainly SPRS, and to establish the natural history for these genotypes, and also because there was no information for the pediatric cohort. And in this study, there will be 5 different follow-ups. It's a 2-year study with 6 months periods of interval. And there is also, in the baseline visit, we also need to do this test, retest. So there is still some issues regarding the validity of the scale. This is a collaborative initiative, mainly from European centers, but also with IAXON from Brazil and CERN from the United States of America, trying to validate better the SPRS. So at the end of the day, we want with this that we not only have a clinical scale, but we have, in this case, a clinician-rated outcome measure that could be used as an endpoint for clinical trials and validated or accepted by regulatory agencies. I'd like to thank a lot to Rebecca. So she supervised me in a lot of this work, and I have the opportunity last October with Rebecca and Sabrina to record this video. So it was just one day, but we have a lot of work in that day and also editing the videos to provide this tool. And I also would like to thank the guys from IAXON Brazil, which is the trial-ready network we have in the country. Marcondes is also the co-lead of this initiative. And also don't miss the posters of IAXON that are in the bottom, in the poster sessions. Then we present a bit more of this initiative. Thanks a lot. Dankeschön.
Thanks a lot. Thanks a lot. I think this jumping on stairs and tables does not belong to the modified, but it was really a great talk. Maybe you stay here. I ask quick the other presenters up front, and I think despite the fact that we are tight to 6 o'clock, I think I would like to have a few questions, because the talks, and in particular, whether it's outcome measure, patient-related outcomes, certain variants, and so further, and biomarkers are important topics, and then I think there are questions. Please, please.
Thank you so much for the excellent talk. I have a question about biomarkers. Is someone looking at cell-free DNA? Because you look at proteins, but in my experience, DNA proves to be always more reliable than proteins. Is someone looking at that, like trying to see if you look at cell-free DNA, also methylation and also perhaps mutations, you can see something meaningful, like reflect the neurodegenerative process in the blood?
Yeah, I'll repeat the question. I think it was about looking at DNA, cell-free DNA, or modifications of DNA. I'm not aware of any efforts in the HSP space on that specific topic, in that specific area. I'd have to think very hard. I think it's a good idea. I don't think any work has been done. We haven't done any work in that area.
There, I see a question there in the behind.
Thank you. Hi, Matas Sinowczyk, Tübingen. Darius and others, great work. Just a note on NFL. There's a common misconcept, I think, in the whole neurology field about NFL. NFL is unlike any other COA, clinical outcome assessment or biomarker. Does not reflect the level of abnormality, but the rate of abnormality. And as such, is different from SARA, SPRS, or any other biomarker. And the reason why I'm emphasizing this, because it was also at least implicitly present in your analysis. Because what you were doing, you were correlating this with other markers, clinical outcomes or biomarkers, which reflect the level of abnormality, not the rate. And that was largely, I guess it was due to the nature that you just had cross-sectional data, so you had to do it. But I think, just as a word of caution, what we should do as a field is should highlight that this is not the actual analysis, but just a proxy. Because it seemed like, oh, it's not correlating with the SPSS or any other measure of disability, but it's not supposed to do this job. And this caveat was lacking from your presentation, many others. So that's just a word of caution to the whole field and any publication on NFL.
Yeah, I think the rate of change is very important, and I think we need to establish that. I think another word of caution is I think we need to use the same platform then to measure NFL. I think that's important. And still correlated with clinical changes. I think in the ideal situation, we'll have a very clean data set in 2 or 3 years where we have longitudinal clinical data mirrored by whatever blood-based marker we have. Absolutely.
Maris, just to get your idea, like, so even if it's not that sensitive to change, so the information that Darius showed, like different slopes, but if you are able to change NFL with a treatment, that would be relevant because it's slowing down. That's what you're saying?
Not the, if you're a car, if the biomarker is a car, it's not about the distance traveled, but about the speed of the car. And as all the other biomarkers are distance traveled, whereas NFL is the speed of the car. And of course, it's great if you have a car which is overspeeding, NFL being too high. If you reduce the overspeeding, that might certainly be meaningful. So it's not about meaningfulness, it's just what you look at. Reducing the speed, not the distance traveled. And every other outcome measure is reflecting the distance traveled. So that's why it's so important.
I think regarding the response, in particular for the SOD1 associated motor neuron disease, it has been established, and it's also at the moment at least predicting certain, in multiple sclerosis, certain acute phases. I think there is a big development, maybe not just one single one, but I think a different one. I believe that it will not stop by NFL, therefore this kind of unbiased assays help. And in particular, besides what you meant with DNA and RNA, I think there's still a lot in biofluidics which we need to address. Maybe other questions? I see not now, but I think all 5 will be also at the dinner. And I think they take the questions and they deserve a big applause for these wonderful talks.
So I think that Henry and Susanne will do some housekeeping remarks.
And we have some experience with it.
Yeah, thank you very much for this first interesting meeting. And just a quick note, in the foyer, there are waiting some presents for you, some personalized mugs, and they are donated by Wouter de Vries. He's the chairman of the Life for HSP Foundation, which also focuses on helping people with HSP. And please look for the name on the boxes. And if you can't find your name, there are also blank ones for you. And now we are going to the restaurant and we meet there at 7:15. It's called Wilma Wunder, and the address is?
It's Schaumburgstraße 1, but it's directly in the city center, really on the central marketplace, close to your hotel. So just return to your hotel, dress up, and then it's just 5 minutes walking or so, if you want to. See you tonight. Have a great dinner. Oh, somebody came with a car. We have free parking tickets here for you. Just go to Nicole. Have a great night. See you later.